So their population is going to start shrinking, and in particular, you can see as this bulge here starts to retire, they're going to have way more retirees than they are going to have workers in their economy. That's going to become really significant because those old buggers, and I'll eventually be joining them not too far away, even though I know I only look 29. But once we all start to retire and we age out of the workforce, we need people to take care of us. It's increasingly the case that we're going to need people to take care of us if we're not healthy. And that's where all of you come in, and that's why you're so important, because we've got a short runway to actually find a lot of cures to a lot of diseases and a lot of lifestyle issues that we've created for ourselves. And we've got to find solutions to those rather quickly. Otherwise, this bulge as it comes through and hits retirement age, and we all start to sit around in nursing homes, we're going to become a really major burden on the next generation below us. And there are not going to be enough of those people in the next generation below us to wipe our bottoms and do all those things, the really undignified things that happen, right? So keep this in mind. So this should be engendering a sense of urgency. We've never had a more, and quite literally, based on the demography and based on these numbers, we've never had such an extreme need to get all of the scientific community, particularly the scientific community working in healthcare and working in life sciences research, to be the most productive you can possibly be. And so, in a way, my call to action here, at this point, is go and position yourself to be in the place where you can have the most impact. If that means switching jobs, go do it. You need to position yourselves where you can have the most impact on healthcare and life sciences outcomes that your work is contributing to. Wake up every morning and think about how you can have greater impact and really measurable impact. So anyway, so that's sort of hopefully sort of a sense of urgency that comes from that. So the point is we all care, and different countries will care a little more than others. Now, the summary of the summary of the summary. You're all weird and rare. Cores are cheaper than you. Apart from Phil and his family, cores are also more abundant. We need you to be more productive. There's a very short runway, 10 years, maybe 15 years before us Gen X start to become, really quite a big burden. So we have to solve some really big problems fairly soon. So we need to get busy, and therefore everybody cares. So there's those 8 billion people in that photograph I showed a minute ago. We've all got something at stake in seeing all of you doing what you do, but doing it a little bit incrementally better every day. So the reason I'm here at this event talking about this is because I genuinely think that this community has switched up research in this space by a huge notch. This is a lever for increasing the efficiency of scientists, building on the shoulders of others, and occasionally reinventing wheels, but to a great degree assembling things out of the parts available and then quickly moving on to bigger and better things. And I think it's been the hallmark of this community since its inception, and I've been watching this for some time. So anyway, we're going okay. We'll get a little bit of time in for some of the nerd talk. I better, do the things that pay the bills. Now, I work in HPC engineering, so really my job is to, my job and my organization's job, we are all about trying to figure out how we can make HPC simpler and get the infrastructure simpler so that you guys can run these big workloads that you run very quickly on demand and without too much fuss. We also sort of, our approach is that we don't think that we should have people with PhDs building racking and stacking machines in data centers. The specialists in our company who work on data centers have forklift driver certificates, not PhDs. The PhDs are in an office somewhere writing software that manages all of that. And again, it's a sort of immiscible layers. Now, we talk often about this thing called infrastructure as code. And sort of, by way of context, I just want to give you a few insights to a few of the things that we've done this year that I think are sort of having a measurable impact on trying to improve this sort of, delivering an efficiency dividend so that all of you can benefit. Now, so we talk about infrastructure as code quite a lot. The cool thing about infrastructure as code is that you write down the specifications of what you want, and this thing, the cloud formation system in the cloud goes out and builds it for you, right? And so the whole idea there is that the end of it, the manageable asset that you get out of it is the recipe. So just to highlight that, it's the recipe that becomes the manageable asset. It becomes the thing you worry about, not the cluster. The cluster is really a throwaway thing. It's an ephemeral thing. You build a cluster, you get it to do a thing for a period of time. When you're done with it, you dispense with it, but you keep the recipe around in case you want to do it again. And that, do it again maybe tomorrow, maybe next week. But you get reproducible, fairly solid results all the time. Now, one of the things that we've done this year to that end is we've created this thing called the recipe library, the HPC recipe library. It's a repo on GitHub, maintained by a bunch of folks on my team. And what it's all about is actually trying to put some big building blocks in there so that we can get folks being able to build infrastructure in the cloud rather quickly without having to do all of the learning required to understand what all of those things are. And this is meant to be just a fast-track onboarding experience for people who are building workloads in the cloud and getting HPC done. HPC is hard, it's fiddly, there's a lot of moving parts, there's an incredible amount of interconnections that need to be brought together, right? So this recipe library is probably one of the more interesting things that we've done this year, which is really moving the needle in that efficiency goal. So what it does, it's built up of CloudFormation templates, if that means anything to you, CloudFormation templates and CDK scripts, and they're designed to work together so that you can take these big building blocks out of the recipe library and assemble them together, and they're designed to work together. You can have a large networking environment, a big storage environment, file caches, clusters, all of those things, and you can get it all pretty quickly. There's a lot of one-click launchers in there as well, which is basically just a super recipe built up of lots of little recipes. And what those are good for is you can take those, steal them, modify them to your heart's content, and make them your own. And then you've got a one-click thing, which becomes your one-click thing. And if you need to spin up that one-click thing in a different environment, if you're working with a collaborator and they need to copy the environment to work on, you can just hand them over those scripts. Again, it's the scripts that are the manageable asset, not the infrastructure itself. Anyway, so I think this is one of the things that my team has done this year that we've been working on, that we're very happy about, because I think it's going to have sort of like a good impact on this. Now, storage and moving data, and I heard a lot of people talking about this, in the last 24 to 48 hours. Storage and managing data in and out of the cloud is actually kind of complicated. We systematized or servicized Lustre a couple of years ago. We have this thing called Amazon FSx for Lustre. You can spin up a large parallel file system very quickly in a few minutes and have your own Lustre file system. Now, when I was last a hardware guy building HPC clusters, building supercomputers, I guess I stopped doing that about 10 years ago, building physical supercomputers when I moved to Amazon. Building a Lustre file system took 3 to 4 months. It was a big deal, and it was also a big pain in the ass when it stopped working. So the fact that we can do that with a click of a mouse is actually pretty cool. So we've systematized that. This has been a cool thing. Now, this year we came up with this, we sort of came up with another iteration of this, another kind of approach to this thing called file cache. And what file cache is actually taking a Lustre file system and using it as a front to things that are behind it, like an S3 bucket or an NFS filer. And those S3 buckets or NFS filers, they could be local, they could actually be far, far away, in another region, in your own offices, in your own buildings, in your data center, in someone else's data center, right? But you've got a Lustre layer sitting in front, which suddenly makes the entire thing very POSIX-y and extremely fast. Really good for lifting a public data set out of an S3 bucket and turning it into something that you can do an insane number of IOPS on. Hundreds of gigabytes a second, lots of IOPS, and so forth. So we are working on this, and you can unify different things. You can, one of the cool use cases is to take something that's a bit of S3 over there, a bit of NFS over there, and maybe a bit of other local stuff nearby, and make it look like it's a single file system. There's some cool aspects of that, and I encourage you to have a play. But more to the point, reach out if this is not doing what you need it to do. As with all of this stuff, you guys know how to find me. I'm on the Nextflow Slack. If there's feedback that you want to give to us to let us know that something that we built is not behaving the way you need it to behave to get that efficiency dividend, we need to know. And so just drop me a Slack, get back to me. I'll make sure that that information lands in the hands of the engineers and the product managers that are designing and building it. Now, I think batch is a wonderful thing. I'll say that. Batch is actually probably the service that most of you are using. We did a lot of work this year on the fair share scheduling in batch. So if you're using an AWS batch environment and you're sharing it amongst a bunch of different users, fair share scheduling may actually be something that you really want to look at doing in order to reduce the friction and tension between you and your neighbors that are sharing the same thing, the same resources. So have a look at that. But I kind of want to quickly get on to this next topic. So last year when I was here, we announced this, which was the biocontainers mirror in ECR public, so the Elastic Container Registry. If you're not using this and you're using AWS, you really should. If you're not using this and you're using some other system or environment, you probably should be thinking about using this as well. The cool thing about this ECR public mirror of biocontainers, it's fast, really fast. It's really fast if you're inside AWS, that kind of makes sense. But we actually fronted the whole thing with our CDN, a thing called CloudFront. And what that means is that if you're in, I don't know, Melbourne, pulling a container, the first time it'll take a little while, but that's more or less the same experience you have, say, using the biocontainers on key.io right now. But the second time you get it, it's going to be super fast because it's going to be in the edge cache very close to you in your city. It doesn't matter whether you're in Melbourne or Madrid or Milan, right? Or Milwaukee. There you go, I found another M in another part of the world. So if you're not using this thing, give it a try. It's literally a single line change in your configuration file, and you can get access to this. This is going to be much, much faster and could actually speed up some of your pipelines. But while we were working on that, one of the questions we've been asked by a lot of customers is, can we have an ARM mirror of biocontainers, so that it's not just x86 that everybody's driven to use, it's ARM. And the reason for that, and it makes a whole lot of sense, ARM is actually really cheap. Let me see, I've got a slide there. So the thing on the right-hand side is the HPC instance we launched mid this year, made up of the thing that we call the Graviton, the AWS Graviton. That's our ARM-based processor. Slightly souped up version of the architecture in your Apple M1 or M2 is actually majorly souped up. It's got really pretty incredible memory bandwidth. It's a groovy little CPU. It's amazing. Goes toe-to-toe with its x86 cousins in terms of performance, but it's about 40–50% cheaper. Carbon footprint is 40 to 50% better in terms of flops per watt, if you think about it from that point of view, or flops per ton of carbon. So it's a pretty amazing processor, and a lot of folks are moving to it now. So the question that we got asked, more or less as I got off the stage last year, people were asking me about an ARM version of it, and so we really do want to get that happening. Now, there's a bunch of different groups in the community who've been doing a bit of work to port much of what goes on in Bioconda into an ARM binary universe. But there's a group that we work with on the sort of the traditional HPC side of the house, on the supercomputing side, right? So Department of Energy, national labs in the US, where we do a lot of, we do a lot of work with those folks, Lawrence Livermore and some of their friends. They've come up with a tool called SPAC. Now this is sort of a fairly normal dependency tree for a fairly simple HPC application called Tinker. It's a molecular modeling tool. It's considered to be a mini app, so it's not considered to be something that's that complicated. But you look at the dependency matrix here, and it's just bonkers. Anything changes in there, and it's just chaos in the build environment. So these nice folks in SPAC, the open source community called SPAC, solve this, and the SPAC tool is that first 5 characters of that command there. Boy, is that doing a lot of work. SPAC is it's an HPC package manager that understands all that complexity, and it just goes and finds all of the source bundles for you. SPAC's built on recipes, and it can pull a recipe out of its recipe set, pull all of the dependencies in, build them, get the thing working, and in, I don't know, maybe 30 minutes you've got BWA ready to actually run. And the whole thing's compiled and working really well. Then we kind of pushed a little bit further forward and worked with the SPAC team, and we said to them, why couldn't we have like a giant build cache? Why couldn't we have a binary build cache? When somebody builds one of these things, spends the CPU cycles to actually compile all these things into binaries, why can't we have those binaries be available? And they said, that's actually an ingenious idea. So we all got together, and AWS actually funded most of the compute that runs behind this. My team, we sat and looked at this and thought, this is one of these places where we can just move the needle really quickly. And if it means that we no longer have scientists sitting around waiting for 2 to 3 or 4 days, pulling all of the dependencies together and compiling them, this is just a major win. So we put a tremendous amount of compute behind this, and what's come out of it is the SPAC binary cache. And for a bunch of different architectures and a bunch of different environments, including an ARM-based environment, so ARM64 binaries, there is a giant binary cache of applications. And the convergence between the x86 cache and the ARM cache is actually, they are converging over time, and we're starting to see, pretty good outcomes. But necessarily, what is hard is unit testing and optimizing. So building something is partly a barrier to many of these codes, and there's just an awful lot of x86 assumptions built into people's makefiles and build scripts and so forth. There's a lot of x86 assumptions, there's a lot of compiler assumptions. Then, of course, at the other extreme, there's, can we actually get everything not just building and running, but building and running fast, right? Because ultimately, fast is the goal. This is high performance computing. So here's what I want to kick off today. So we've been doing a lot of work with Marco, who's a new member of the Seqera team, but has been a Nextflow member for some time, Nextflow community member for some time. He sort of lives in both universes of the Bioconda world and the SPAC world because of the job that he used to have working at a supercomputer center. And Marco's done an awful lot of work to sort of take some of the ideas from SPAC and see if they can work over here. But in order to get a large-scale effort going and to bring it to completion, and really to get 99% of all of the, of all of biocontainers to be, executing happily on ARM64 CPUs, whether they're Gravitons or some others, or the ones on your laptop. In order to get that to happen, we need a few things. We need to crowdsource some of the effort. So we're going to, in the first quarter of next year, we're aiming to have, my team we're gonna try and stand up this is a crazy idea, it's called a build jamboree. We're gonna get an enormous number of people from around the world, and we're gonna get together over a weekend in a lot of cities. Probably eat a lot of pizza and drink a lot of Red Bull, and we're going to see if we can actually get an awful lot of these recipes working on both sides of the house so they just build. And we think that'll be a good, be a useful community effort. But we also need unit tests. And I think this community, particularly when it comes to codes in Bioconda, you guys have got all of the unit tests already, I suspect. They're in your pipelines. And so I do want to see if we can solve that problem. Because once we've got unit tests, we've got an awful lot of clever people over at ARM who are willing to put some effort in ARM and in AWS and in other companies as well and other places that are willing to put the effort in to see if we can optimize this stuff to make it run faster on ARM. So anyway, so that is my ask to you. Don't be shy. You know how to find me on Slack. Most of some of my teammates are there as well, and we can get some interesting conversations going. Marco is the guy in Seqera and in the Nextflow community who's really leading the charge on this, and I'd like to see if we can pull together some of the folks from around the community to see if we can all get together and clean up this particular yard and see if we can make things work much, much faster. Anyway, in the end, you all know what the goal is, right? The goal is that we've got something like a decade left to get all of you and all of the people that you support being incredibly productive, because we need you to solve some really big diseases and lifestyle issues and medical issues at an enormous scale, and the pressure is on. Anyway, thank you. I hope I didn't depress you. I hope I've kind of motivated you, but thanks for coming. And thanks for this community. And particularly kudos to this guy up the front here who's been key to starting this movement. Anyway, thanks very much.
So we are a bit tight on time, so we'll go to the next speaker. Thanks, Brandon.
No problems.
I'd like to introduce you to Julia Mipedrao from the University of Tübingen. Stage is yours, Julia.
Thank you. So good morning, everyone. I'm Julia, and I'm here to talk about how you can use the NF validation plugin for the validation of pipeline parameters and input files. So I think you are not seeing my slides.
It's not plugged in properly. That's good, so I can go to slideshow.
Perfect. So in Nextflow, we have this special variable called params, which is used to launch workflows providing different parameters or inputs and settings for every run. And also, it's very common to use a sample sheet. This is a file used to provide all the sample-dependent information, such as file paths, sample IDs, or other metadata. So Nextflow doesn't provide an integrated functionality for this validation. That's why pipeline developers should have to write several codes to validate this. In the NF Core community, we started developing some Groovy code to validate these inputs, but in order to extend this to all Nextflow pipelines, we developed the NF validation plugin. This uses the JSON schema format to provide all the validation rules. So in the NF validation plugins, we have different functions that you can import to your main Nextflow script and use them. For example, the first one we have params help, which will print a help message to the terminal when a user uses the param help. Then we also have param summary log, which will print a log of all the provided parameters that differ from a default value. To actually validate these parameters, we have validate parameters. And then we can also validate the content of input files, such as sample sheets, automatically with validate parameters or from sample sheet, which is our last function. This one can also be used, or can be used to create Nextflow channels from the contents of these files. So as I mentioned, we use JSON schema format. If you are familiar with JSON schemas, you will see that we have some particularities that differ a little bit from the standard schema. For example, with use grouping, this is useful to organize the parameters and make the outputs look nicer. And with this schema, you can validate several things. For example, the type of your parameters, if they are strings or integers. You can also specify which parameters are required. And for example, for strings, provide patterns or minimum and maximum length. And then other particularities that we have is this help text. With this, you can provide a longer help message, which can support markdown formatting to help the users a little bit better, or error messages, which will be usually more explanatory than the default error message. And finally, let me bring your attention to the last keyword schema. I'll talk about this in following slides. This is used to provide the path to a second schema file, which will be used to validate the contents of an input file provided through this parameter. So because of all these particularities and to avoid errors when creating this Nextflow parameter schema, it's recommended that you don't do it manually, but in NF Core tools, we provide some functionality with this NF Core schema build that will help you create this parameter schema using a graphical interface. So as we have seen with this parameter schema, we can validate our parameters, generate help text and logs, but on top of that, it can also be used to generate documentation. For example, that's what we use in the NF Core website. For every pipeline, we have a page describing all the parameters. And it's also useful for launching pipelines, helping the users provide their parameters through a user interface. Let's move to file validation. First of all, thanks to Nicholas, who contributed with this feature. So as I mentioned, with this keyword schema, we can provide the path to a second schema file that will validate the contents of an input file or a sample sheet. And the same schema is also used for the creation of an input channel. So you can validate CSV, TSV, and simple YAML files. And each property, in this example, we have field 1 and field 2, will validate a column of a CSV or TSV or an entry of a YAML file. Apart from validating all the same things that we could validate with a parameters schema, such as type or required fields, we have some special keywords. First one is meta. So in channels, it's very common to have a meta map, which will hold all the metadata information. With this keyword meta, you can specify that your field needs to be stored in this meta map. Then we also have unique that will validate that all the values from one field are unique. Deprecated to throw a deprecation message. And finally, dependent required, which is very useful, for example, if field 2 is required only if field 1 is provided. So let's see an example to understand a little bit better how this works. This is a very reduced schema for space purposes, but we have a sample sheet with 2 samples, and each of them has the sample ID, 2 files, and strandedness. And here in our main.nf, we are importing the function from sample sheet from the plugin, and then we are using it to create an input channel called channel input. And in this case, as an argument, we provide the name of the parameter that will be used to provide this file. In this case, it's input. So if you see below in the Nextflow command, we use input to give the path of our sample sheet. Then in this example, sample ID and strandedness are metadata. So that's here we are using the keyword meta, specifying the name of the key in the map that will store these values. And then in the output, we are seeing the channel. So you see we have a map with my sample ID and my strandedness. And then everything that's not a meta value will be added to the channel in the same order as it's specified in the schema. In this example, we have first FASTQ1 and then FASTQ2. So all this was for channel creation, but at the same time, it is also validating the values. We are validating that our values are strings. For FASTQ1, we have a pattern. Then for strandedness, we have the 3 possible values that it can have. And finally, sample FASTQ1 and strandedness are required. So to prove that this plugin can make your life easier, we are going to quickly see 2 real-life examples. First of all, in NF-core RNA-seq. So as I mentioned, in the NF-core template, we have some code already to validate the input parameters, which is this Groovy file. So when we were able to remove all this code and use the NF validation plugin, we could remove more or less 600 lines and add less than 200. And for sample sheet validation and the creation of the input channel, we were using a sub workflow with module and a Python script that was doing all this validation. So when we replaced that for the plugin, we could also reduce around 200 lines of code. In the case of Zarek was already using some Groovy and not these Python scripts to validate their inputs, but still we could have an improvement of almost 100 lines of code, and there's still margin for improving that, which leads me to my last slide, the next steps. So thank you to everyone who started using this plugin in the community and provided a lot of feedback. We already have some perspective to keep improving. First of all, our plan is to provide some tooling to create these sample sheet schemas, because now currently NF-core tools provides tooling to create the Nextflow parameter schemas, but not the sample sheets. And finally, this conditional validation, which will be very useful for several pipelines where they need to validate some parameters. So the validation of some parameters depends on the value of other parameters. And I hope you now have an overview of what you can do with the NF validation plugin. Thank you to everyone who helped with contributions and the development of this plugin. And you can find documentation and get in touch with us in both Nextflow and NF-core Slacks in the channel NF validation. Thank you.
So thanks, Julie, for the talk. So there's a question. We have time for one question from Adam saying, who validates the validators? It seems to be using vanilla JSON schema, the variable, not one that includes the Nextflow extensions that have been added. So we have anything to
so for validation, we use the JSON library, which does this validation of the parameters. And what was the second?
I think he's referring maybe to either to split JSON operator or something like this. You're using like vanilla JSON. Was not very clear.
To validate the JSON schemas?
Yeah.
So with the tooling that we use, we provide to create this JSON schema, it will generate like a valid schema, and then you can also validate that your schema has the proper format.
Okay. Thank you. So the next talk is going to be with Michael Wilson. Stage is yours, Michael. Welcome.
Perfect. Thank you. My name is Michael Wilson. Good morning to you. I'm a data scientist at Biomodal Biotech in Cambridge in the United Kingdom. We've recently gone through a rebranding like Seqera. We were formerly Cambridge Epigenetics. And I'd like to talk to you about our first product that we're putting out publicly, which is Biomodal Duet plus modC, and its application for variant-associated methylation in genetics. So we know DNA is the alphabet of life, with canonical bases A, C, G, and T. But each cell carries the same genome, yet they all differentiate into brain, liver, kidney, heart, etc., all of which serve different functions and different roles in the body. So how does this happen? So the name for this field and the collections of mechanisms that make it happen is called epigenetics, and it works in combination with cellular factors to change the expression pattern of genes in different cell types and controls how they're constructed, how they develop, how they age, and in particular how disease progresses as they go. Now there are multiple different forms of epigenetics, including non-coding RNA, histone modification, but I will focus today on DNA methylation, as this is the one that's provided by Biomodal Duet plus modC. So there are many different types of methylation that occurs to cytosines in the genome. This includes 5-methylcytosine, or 5-MC, which occurs, so if you get a C followed by a G in the reference genome, then 5-methylcytosine occurs about 50% of the time, and typically works in order to suppress gene expression. So it actively targets gene promoters upstream of genes and makes sure that that gene is expressed and allows one of the primary mechanisms by which cells differentiate. In contrast, 5-HMC occurs a few percent of the time and is mainly enriched in gene enhancers and active gene bodies, and is typically localized where genes are expressed rather than suppressed. Now, these mechanisms lead to a really interesting phenomenology. In particular, in the human brain, 5-HMC, rather than being a percent level effect, occurs 20% of the time. And this is critical for how cells can differentiate in the brain to many, many different types and forms. It's crucial for the normal development and functioning of the human brain, and in particular is associated with learning and memory and the associated diseases. Beyond that, in contrast, in the realm of cancer, it has been suggested that for primary tumors, both genetic variants, so a base on the reference, say a C, that converts to T in your own genome, can cause inactivation of a gene, but that can also be followed up by methylation of the other chromosome you carry, such that both genetics and methylation are vital to predicting the expression of, say, the BRCA1 gene, and ergo in terms of determining how cancer will progress. Finally, the picture's a little complicated. If the cytosines are methylated in your genome, they're also more prone to converting from a C to a T overall. So it's a complicated interplay of both genetic variants and methylation in its different varieties of 5-MC and 5-HMC that you need to understand in order to fully be able to describe the pathways with which these diseases progress. So why aren't genetics and methylation interrogated more often? Well, for quite a while, we've had mainstream methods that kind of work, but they have various limitations. For instance, you're required to have multiple workflows in the lab, multiple kits, in order to accurately measure both the genetic variants and the methylation that is occurring for cytosines in the genome. This is particularly important when you have low input amounts of DNA, such as when you take a blood sample that gives you cell-free DNA floating around in the bloodstream that one would hope to be able to use for early diagnosis of cancer. It also uses harsh chemicals that damages the samples and leads to reduced accuracy for the calling of both variants and methylation. The data analysis is, by extension, pretty time-consuming, can be complex, and at a technical level, you're only resolving 3 bases rather than 4. So when you try and align to reference genome for short reads, you end up with more mapping inaccuracies than you would otherwise. So I have to convince you today that Biomodal is a better solution. It's one that uses just a single workflow. It requires minimal amounts of input DNA. It does not damage the DNA. It has high sensitivity and specificity for calling both SNPs and methylation. It does so without data loss, and in terms of what most of us here are interested in, it works as a pipeline with a click of a button that does state-of-the-art analysis. Not least, it enables new applications that combine both genetics and methylation for variant-associated methylation to give us new insights. So with that motivation, I'd like to talk to you a bit through the chemistry of how this actually happens and how we identify cytosine modifications. So we take the original strand of DNA, we litigate on a hairpin construct at the end of the strand. We then apply enzymes that create a copy strand. In particular, on the top here, you can see the red scenes, which carry a cytosine modification, so either 5MC or 5HMC, that we use enzymes to protect these and otherwise apply deanimation. So this turns unmodified cytosines into Ts and leaves the modified cytosines alone on both the forward read and the backward read. We then use next generation sequencing in paired end mode, such that read 1 maps to the forward strand and read 2 maps to the copy strand. At the end of the day, this gives us the bases on read 1 and the bases on read 2. They come in pairs. There's then a lookup table that you can see at the bottom that shows that if you have a pairing of A and T on the top and the bottom strand, then the resolved base that you've measured is A, one of the canonical bases. In contrast, if you end up with CG pair, then you've actually measured modified C with 5MC or 5HMC. There's also various error suppression modes or states where if you have a pairing of AC or TT, this can only occur from inaccuracies in the PCR amplification that occurs in next generation sequencing. So we can suppress these and by a result get better SNP calling. So obviously this is a pretty involved pipeline. We take our protocol in the lab, we run it through a next generation sequencer, generating raw fast queues. We use read resolution to take these 16 states and map them into both the canonical bases and modified C. We do 4-letter alignment rather than 3-letter alignment to create BAMs to a reference genome. We call variants. We quantify the available methylation for both 5MC and 5HMC. And we use these to enable novel analyses that are an extension to the pipeline associated to the impact of variants and methylation. Of course, this all happens with Nextflow and all of the mainstream cloud providers that are out there, including AWS, GCP, Slurm for universities, and Microsoft Azure. So the result of this work is that we get uncompromised genetic accuracy. So our calling of SNPs is comparable to what you get from whole genome sequencing with, say, Illumina. We have the additional error suppression that we utilize in order to achieve this. In particular, compared to the other methods out there, they're not able to distinguish methylation from C to T variants in your genome. As a result, they're not as accurate as we can be for calling SNPs. And this is true of both bisulfite and enzymatic methods. So for variants, we do well. We do as well, if not better, up to even low coverage. We also make more accurate modC calls than existing methods. So this is particularly true of specificity, but also sensitivity, which is better than a lot of the methods that are out there. And we can enable new analysis, such as variant-associated methylation. So you carry chromosomes in your body. For each one, you have a pair. Typically, this is derived from both the maternal and the paternal allele that you inherited. Now, in this case, we have one allele that is consistent with the C on the reference on the top, and this is fully methylated for reads for the cytosines on these reads. In contrast, your second allele is consistent with a T variant at the same position and is entirely unmethylated. Now, because other methods cannot identify C to T variants, they could not have been able to distinguish these physical read groupings. And by extension, they give you an average methylation signal that misses what's really going on and really predicting how the disease is progressing. This has been suggested, for instance, that the PLAGL1 gene that acts to suppress cell growth and is implicated in certain cancer cases works by exactly this mechanism of having allele-specific methylation. In contrast, because we can identify these SNPs, we can identify these read groups and call the differential methylation exactly. Now, obviously, we understand that for individuals who carry two X chromosomes, they would have double the amount of gene expression than those that carry XY. In the body, this is actually controlled by a mechanism called inactive X, which applies global methylation to one of the X chromosomes for individuals that carry both. Because we know in these cases that one of the chromosomes is methylated and the other one is not, we would predict that we would see a higher instance of allele-specific methylation than on the autosomes or non-sex chromosomes in the body. So we went and looked for examples of genes that have been predicted to be inactivated on the X chromosome and compared them to the ASM calls that we are making, and we saw exactly this. So for genes that are inactivated via methylation, we see an enrichment of the ASM calls that we're making. On the control genes, on the autosomes in the body, we see many less calls that's validating the method and showing that this is prevalent in the human body. And for a class of escape genes that have leaky inactivation, if you like, it doesn't work perfectly, we see an increased number of calls, but nowhere near the number we would expect of inactivated X. So in summary, Biomodal duet plus modC has many benefits. So when it comes to epigenetics and genetics, you no longer need to choose. You can obtain both simultaneously with one sample and one analysis pipeline. This is particularly important for low input DNA, such as cell-free DNA used for liquid biopsies, where we can go as low as 10 nanograms. This is true of genomic DNA, cell-free DNA, and also DNA that is being kept in long-term storage using FFPE. Our technology is able to retain C to T mutations that others cannot. C to T is actually one of the most common genetic base changes in the human body. And unlike other mainstream techniques, this means that we get the full picture and we do not obscure what's really going on. And this impacts the SNP calling and sensitivity of methylation that I've shown previously. Finally, its bioinformatics tools included. Duet modC pipeline comes with its own read resolution module and incorporates seamlessly into current workflows. It also enables novel analysis, such as variant-associated methylation, that you can get for the click of a button. I'd like to also highlight, finally, that we're employing or hiring data scientists and computational biologists at the minute. So if you would be interested, please contact me, and I'd be more than happy to talk to you about the positions. And with that, I'll leave you with a summary slide for Biomodal duet plus modC.
So thanks for the talk. We're going to move you on to stay on schedule, but there are some questions on Slack
No worries.
to answer them directly. So Jacob, the stage is yours.
All right, good morning, everyone. I'm Jacob. I'm going to talk about how to use data pipelines in machine learning for material science. I'm a PhD at UCL, University College in London. I've also applied my research at Spotify, so your music sounds better. And since 2018, I've been working also in material science, working at the Army Research Laboratory on epoxy glass composites. Nowadays, I spend my time on something called Matterhorn Studio, which enables instant access to machine learning pipelines that are cutting edge and they're really great for lab automation. And so today, I'm going to talk to you about lab automation, why we do that, then why we need machine learning for experiments, and then ultimately, why we need pipelines for that machine learning. All right, so materials do rule the world. You're sitting on chairs that are made of materials, the computer's made of semiconductors, and right now we need to kind of figure out how to make these materials in a more sustainable way that can be recycled. And biosynthetic materials are quite attractive for that. So for example, with yeast cells, we can actually produce fuel. We can also produce beer, which you might know. And there's also biosynthetic materials that can help with drug and pharma production lines. So, of course, we want to maximize the output from these yeast cells. So what kind of processes can we go with that? And so, for example, here's a process at the Technical University of Munich. And they're working on these yeast cells. They have a whole bunch of complicated steps from discovery to scale up. But today, we're just going to talk about that middle part here, which is a loop where we have some yeast cells and we're going to feed them vitamins and trace minerals, the same way you kind of feed things to humans to make them strong and produce better science or more fat or something. So why do we do lab automation and research, for example, for yeast cells? You see here some machines. There's an OpenTron, costs about $10,000, up to maybe a Beckman for $500,000, and then custom designs for multiple $1 million. And the great thing with these lab automation systems is they give you high throughput, so they just produce a whole bunch of data. More importantly, they're also reproducible. So instead of having 3 scientists do the same experiments and getting 5 different results, you have basically a robot that will do it almost the same way every single time. And so that leads to the scientists being freed to just do the science, which is what they sign up for in the first place. Now, specifically, what you want to do in these kind of setups is that you have a closed-loop experimentation system. So for our yeast cells, for example, we're going to have a first step where we're just going to grow them and feed them stuff. That takes maybe 18 hours, and then you're going to move them into a measurement stage, so step 2 here. And then finally, when you have that data, you have to kind of make the decision of what do I do next? What do I tell the robot to run next on the experimentation machine? And so this automation is actually happening across all kinds of disciplines right now, physics, chemistry, biology, who are trying to figure out better alloys for faster aircrafts, chemicals, paints of combinations, plastics that are more stable but more sustainable, and concrete that doesn't require, a kiln firing, which is really, really energy intensive. So here you see different robot systems that have been put together over the last 5 years. Now, why do we need machine learning to run these experiments or to plan specifically these experiments? And the central question there you want to ask is, what experiments should we run next? And so in this example here, again, we have our yeast cells. And so we're going to look at just temperature, just one parameter. And so we maybe look at anywhere between 10 and 30 degrees. And so right now we have some historical data points here. So these black axes are previous experiments we ran in the lab. And right now, 25 degrees is the best top runner. It produces most of the lipid yield, so the cell performs best at 25 degrees. And in the middle, we're missing data, right? So what should we do next? How should we just explore this kind of parameter space? The first one kind of strategy is random. So you just throw some dice, or you throw some if you're bad at darts, I guess you throw some darts at the dartboard, and you just hit some different temperatures, you put them through your lab automation setup, or you just do it manually in the lab. And it's actually a statistically valid method, it's just not particularly efficient. And so maybe you want some more structure. So with a grid approach, you just kind of chunk up the parameter space in even spaces, and you just explore it that way. So that way we will get a better structured idea of how these yeast cells behave. And then finally, we have design of experiments. There's a show of hands, who knows design of experiments? Wow, okay. Different crowd. Right, so design of experiments is the established method to run experiments, for any kind of experiments, really. So specifically, of course, we want to characterize behavior of any kind of natural process. But for example, with these cells, we might think that there's some kind of red line that characterizes that behavior. And design of experiments helps you to pre-calculate which experiments you need to run and to get the best kind of line of fit. Now, we're moving into the stage of machine learning nowadays, and so there's actually a fourth method now called Bayesian optimization. Let's try this one, Bayesian optimization, show of hands. All right, that's a bit more, good, I'm happy. So I'm gonna explain to you Bayesian optimization now in 3 minutes. And basically, here's a red line, which is kind of nature. So this is the true yeast cell behavior. Of course, nature never tells us, and we kind of have to play with nature and run those experiments to get some data on the yeast cell behavior. So obviously, I just created that red line. I know the truth now, so I can show it to you in this example. So here, we have some historical data again, and we can fit a machine learning model here with the blue line. So the blue line is guessing right now quite well what the data is. But then in the middle part, we don't have data, and so the guess is kind of regressing just to the mean of what it sees in the other data. And on top of this specific machine learning model here, called a Gaussian process, we also have an uncertainty measure. So you see the confidence band is quite tight around the places where we have data points. And then where we don't have data, so in the middle part, it's quite wide. So that means machine learning model says, I don't really know what's going on, don't trust me too much on my predictions. And so with the predictions and the uncertainty measure, we can now apply kind of acquisition functions or expectation gain functions that will help us decide what experiment to run next. So that looks like this here. So we have this orange line now. That's the mathematical calculation of the maximum statistical information gain for this specific point. And so right now, the machine learning model thinks that running actually around 25 is a good idea. And that kind of makes sense because 25 is the highest performing one. So let's explore a bit more around this space. So we go in the lab, we run the experiment, we put in the data, and so this is our new kind of understanding of the world of yeast cell behavior. And so again, the expectation measure here in orange now suggests for us to move closer to maybe 22. So you go in the lab, you run the experiment, you put in the data point, and now we see we kind of split up the space already in a high uncertainty region on the middle left, and then another one on the middle right. And so a machine learning model is getting a bit funky here with the blue line, and it's suggesting that we actually go back to the 25 degrees. So we go back into the lab, and we explore that. And now we put in the data point here, and now the machine learning model, as you can see, is slowly squeezing out the uncertainty in an efficient way. And furthermore, we're actually moving to the optimal point at 18 degrees Celsius. So a few more steps. There's 18, there's 19, there's 16 Celsius, and then finally at 18 degrees Celsius, we have reached the maximum point. So machine learning with optimizing around these uncertainties eventually will converge to the optimal temperature. Now you're going to tell me like, well, Jacob, I can see this obviously. It's right there. It's a temperature and I just explored. But the fact actually is that in high dimensional spaces, Bayesian optimization will just be a better choice to explore these things. And so especially when scientists need to deal with 4, 5, 6, 7, 10 dimensions, it is much better to use these kind of methods that are efficient to search this space. Okay, well, I'm glad we got through this. You can try this again on matterhorn.studio and play around with the demo I just gave to you and get a bit more experience with that. Now, here's an overview again, why Bayesian optimization is a good choice. Specifically, random is simple, but it's not particularly smart. With the grid, we have a bit more structure, but it's just too expensive on high-dimensional spaces. Design of experiments is a great choice to make your interpretation and make a first step. but it's complex and not particularly efficient in a lot of applications. And with Bayesian optimization, we're able to most efficiently navigate the space, but it's also complex and it's data hungry. So data hungry, well, we just kind of try to produce most data, for example, flow app automation. And then the complexity part, we're trying to fix that with Matterhorn Studio. So that's the little sheep here. So why do we need data pipelines for machine learning is the final step here today. So there are challenges in Bayesian optimization, of course. There's no free lunch. And so the first one is that we need to find the right algorithm. So different machine learning algorithms that are specific and perform better for certain materials. And then you also want to collaborate with an expert on it. So you don't want to leave statistical efficiencies on the table, but you want to integrate them into your machine learning model. And then finally, of course, it's science, so we want it to be reproducible and transparent and interpretable. So it would be great to have some kind of like structure and standard, right, to standardize this. Well, so we put together something called the opt store, which is an optimization store. And it's basically just a place where you have access to all of these algorithms. There's a REST API. It's based on Docker containers. And of course, as you can imagine, it's using Nextflow pipelines to structure all these algorithms and put them in one central place. And so right now, you can just click on a button here, click on Run. If you set up your data, it will create a draft. You click on Start here, it will go into running mode, and then it will give you the next experiment to run. Either you pipe that into your lab robot with REST API again, or you just go and do it by hand. Now, what's happening in the background here is actually this is just a Nextflow pipeline. So this is the pipeline. You can look it up right now on GitHub. And this pipeline has a few modules in it. And the one that's kind of like, the moneymaker is a Python file where we have, this is a platform called Blowtorch, another platform, a package called Blowtorch by Facebook. And it's a very popular Bayesian optimization method. I just put that into that Python file. And the pipeline pulls the data from our platform, runs the Bayesian optimization, and pumps the result back onto our platform. And that's all just very easily done with Nextflow, because this is the workflow we're going through here. In the Op Store, you kind of choose an app, and then the Nextflow tower will just schedule it on the AWS batch, and then that one will run it on an EC2 container, whatever app you chose, and that will put the result back into the platform. And to support all of these efforts, we kind of created a forum called OptCore, which you can see obviously is inspired by NFCore. And so that place, we're trying to specifically answer questions to machine learning and Bayesian optimization and how to make these work with the Nextflow containers. So here, example, there's a category, Blowtorch single task GP. That's a specific machine learning model. People just can click on that and share about how they use it. And so, of course, because these are Nextflow pipelines, we can do all kinds of interesting things. First of all, they are reproducible and transparent because, well, you just go in the Git repository, and if you just download and have the same container, it will always give you the same result. Then with the OptCore forum, you can actually discuss these efficiently. And then finally, in terms of collaboration and making sure that as a community we're all moving forward efficiently, you can just fork the pipeline, make your own changes to it, and discuss those results with the community. And I think that's better than the chaos right now that's happening with, hundreds of pipelines, hundreds of Python packages being put out there and not being, discussed or collaborated on in an efficient way. So putting it all together, so machine learning helps run the best experiments for material science, but in general anyway. With the Opt Store and OptCore, we're trying to help people find the best algorithm and make that run. And then with Nextflow, we've really found a solution that helps us to collaborate in an open science way to make these pipelines work. All right, thank you so much.
So thanks, Jacob. We have, I think, time for one question. Do you have any plans of trying to put any of these pipelines on NFCore, for example, maybe some material science pipeline that you think could fit?
Whether they would be part of NFCore?
Like, for example, you said you have some pipelines, you create OptCore, right? But have you thought of maybe submitting some of the pipelines to NFCore? Just a question.
Yeah, so I discussed it with Phil. I think it's something that I would love to do. It's about how to integrate these communities. We're trying, so our goal is to support people to really get the Bayesian optimization right in the best way. And of course, obviously, predominantly bioinformatics right now, but if we could find a space to put these together, and I think it will happen, not just for material science or bioinformatics, it will come to other areas of science too. I think, in a way, it's just a matter of time until these things merge.
Totally. Great. Thank you very much.
Thank you.
Jose from CRG, the stage is yours.
Thank you. So, good morning to everyone. My name is Jose Espinosa Carrasco. I work as a bioinformatician in the Comparative Bioinformatics Lab at the CRG, the lab where actually Nextflow was created. I'm also an NF Core team member and from yesterday an Nextflow ambassador. And today I will present the NF Core protein fold pipeline, which implements several AI-based methods for the prediction of protein structures. So maybe if you don't know already, you are wondering why it's interesting to get these protein structures. And the thing is that once we have one of these, when we have the protein structure, we can easily infer the function of a protein. And this way, for instance, we can design drugs for targeting the protein or stabilize the protein structure to use them in biotechnological applications and so on and so forth. It turns out that you can obtain a protein structure using experimental methods, but this is very time-consuming, and also you have to spend a lot of money. And that's why it has been a long-standing problem in the computational structural biology, the problem of how, from a sequence of amino acids, obtain a protein structure using, or infer a protein structure using computational methods. And actually, there is even a context, the CAPS competition, that were there, the computational structural scientists gather and they compare their methods, and the one that is the best performing one is the one that wins. And it was actually in the framework of one of these CAPS competitions where AlphaFold 2 shown for the first time that actually based methods for the prediction of structures can outperform all the pre-existing methods and actually achieve almost experimental accuracy. So here I have the schema from the AlphaFold 2 workflow that I will briefly summarize, just highlighting the points that are important for the pipeline. So the workflow takes as input a sequence, and it searches in genetic databases for homologous sequences in order to build a multiple sequence alignment. You have to keep in mind that this is a quite computationally expensive process, since these databases are in the order of terabytes. At the same time, it also searches in structural databases for structured templates in order to populate the so-called pairing matrix, which contains the information of the residues contacts. So once this is obtained, this is then fed into the neural network architecture. It consists of two blocks. The first one, very roughly speaking, just optimizes these two representations. It's a so-called evoformer block. And then the second one actually gets the actual position of all the residues and gets the prediction, the 3D structure of the protein. And this can be done, so actually this is done several times so that the accuracy it's better. So in our lab, we actually use AlphaFold to predict protein structures, and we use these protein structures to show that you can actually improve multiple sequence alignment. So this was published in this paper, and if you have, you want to know more details, maybe you can discuss with Leila or Cedric that are in the audience. And the important point here, the thing that I want to highlight, is that for this, for doing this work, we developed an Nextflow pipeline that actually was scalable, portable, and reproducible, leveraging the Nextflow features, right? And it turns out that this pipeline got some attention from the community, and actually discussing with Harshil, we thought that it was a good idea to make it publicly available as an NF-core pipeline. And actually, as you can see here, this pipeline was first released in February 2022. The main work was performed by Athanasios Baltsis, which was a former PhD student in the lab. Also, Leila and Luisa helped in some parts of specific parts of the pipeline, and I took care of the standardization of the pipeline, so to say, I take care of the NF-coreization of the pipeline. And you can see in this typical NF-core method map that it has two main workflows, the AlphaFold workflow and the CollabFold workflow, and each of them is subdivided in two other sub-workflows that I will explain later on in my following slides. Actually, we have kept working in the pipeline since its release, and this is the current state of the pipeline in development. So actually, apart from fixing some bugs and issues reported by the users, the main improvements that we have done to the pipeline is that the databases and the model parameters have been updated to the latest version. We also add the ESM-fold workflow. Also, the databases and the model parameters that now are hosted in S3 thanks to the AWS Open Data Sponsorship Program. And finally, this is the theme that it's still work in progress, is that we are trying to run the full data sets in AWS and powered by the Seqera platform. This is still work in progress because in the previous version, we actually have some reduced versions of the databases that we are not really running with the real data that now we have in S3. Okay, so next I will explain the three main workflows. So the first one will be the AlphaFold workflow. So you can choose to run this workflow by using this parameterization. The first thing that the pipeline does when you run this mode is it checks whether you are providing the databases and the parameters in a directory with a given structure, and it checks for the file integrity. If you don't provide these databases, then it will download them. But you have to keep in mind that this is a quite computational expensive process because databases are in the order of terabytes. Then now we can choose between two sub-workflows. The default one, which runs the native AlphaFold, the one that you can find in a DeepMind GitHub repository. And the problem of, so to say, the problem of this sub-workflow is that it runs both the MSA building step and the prediction step in the same process. This is not very convenient because of, for instance, if you are running the pipeline in the cloud, you will be running a process that does not need GPUs in a GPU environment. And that's why we decided to split these two processes to split them so that you can choose what environment you want to use. And apart from this, we have other options. So for instance, you can choose between using the reduced databases provided by the AlphaFold team, or also you can use several model parameterizations of the model. Then there is the second workflow, the CollabFold workflow. Actually, this framework was developed by a collaborator of us, Martin Steinegger. He was a former visitor in the lab, and he took part in the main effort of the AlphaFold 2 paper. And the nice thing of CollabFold is that it optimizes the search in the databases using the tooling, the MMSX tooling developed also in the lab of Martin. So as before, you can choose with this parameter to run the CollabFold workflow. Then we again will check for the integrity of the files, or it will download them. And again, they are in the order of 2 teras, so it's quite big. And the good thing that I didn't mention before is that you will run this only once. And then, in this case, we have 2 sub-workflows. One of those, the first one, the web server, allows us to run the MSA building step in a web server. By default, it's the one provided by the MMM6 team, but you can also customize and provide your own web server. And then we have the collab-fold server local, which in this case runs both the MSA building step and the prediction step in the same in your machine. And you can, in this case, you can also do the first one in a CPU environment and the second one in a GPU environment. Here we have, again, an option to set the different parameters that we can use for the model. And then here we have the possibility of using templates or not, structural templates. And finally, the SM-fold workflow. In this case, we are not using, we are not searching in big data, genetic databases, because this framework is based in language models. So you don't need these databases. You can see actually that you are only giving the parameters to the pipeline, and these are not so big as before. Actually, they are claiming that the efficiency, the accuracy, is as good as in the case of AlphaFold. So you can also give it a try if you are interested. We are preparing a paper that will be submitted once the release of the pipeline is ready. And I don't want to finish without mentioning that in the lab we are contributing a lot to the NF Core community. So Luisa and I'm involved. I am also involved in this NF Core multiple sequence align pipeline that will allow to perform multiple sequence alignments with several tools and also evaluate which of the tools is doing it better. So if you want to know more about it, just visit the poster 14. Also, Bjorn and me have been involved in the framework of the WordPress project, which is the annotation of the functional annotation, of the bovine genome in the framework of the Orofang consortium, which is the functional annotation for animal genomes. So in this framework, we have been mainly involved in these two pipelines and also in other pipelines. And the thing is that in that community, we were trying to push the use of standard pipelines, and that's, of course, NF Core pipelines. And the story is that we were quite successful, and actually many of the groups are now using NF Core pipelines or even developing NF Core pipelines, some of them that are already released. And we plan to also submit a paper explaining this, so to call a success story. And finally, Susan Jean, it's implementing a log ratio-based analysis trying to add this workflow to the differential abundance pipeline. Sorry. And just finish. Thank you all the people that was involved in the project, especially Cedric Notre Dame, my PI, also the core facility of the CRG, Martin Steinegger, of course, and also people of Seqera Lab, Harshil and Jordi that helped me a lot in deploying the pipeline in the Seqera platform. And of course, I would also like to thank both the Nextflow and the NF Core community. So there are some resources of the pipeline in the NF Core page, and there is even a bite-sized talk summarizing the first release of the pipeline. So thank you very much.
So that was a great talk, Jose. So we have a few questions here. So one of the questions is, GPU seems to come up frequently for these types of workloads. I'm curious on any current finding around CPU only versus with GPU performance for NF Core protein fold. Have you tested with GPU or only CPU?
So you can actually run the pipeline using only CPUs. It will take longer, but it's possible.
But you've been using GPU?
With CPUs, yeah. Only that it's not so optimal, let's just say.
But you haven't had the experience of using it with GPU?
Yeah. No, of course. So we are developing it using GPUs, yeah. Okay. So we are developing it.
So is the process to, another question, is the process to the collab-fold web server a standard one? Does it make a call to a URL to wait for a response for the collab-fold web server?
I'm not completely sure. I will have to look at that in the details. So we are leveraging the tools provided by them, but I'm not completely sure.
Okay, thank you very much. So now we're going to have the cough break and then the first poster session. So feel free to go there, visit the posters and ask questions. Thank you very much.
Oh baby, I wanted you to be here inside my arms where the night goes down. Oh baby, I wanted you to be here when the sun comes up and the day goes by. Oh baby, I wanted you to be here inside my arms where the night goes down. Oh baby, I wanted you to be here when the sun comes up and the day goes by.
All right. We're going to get started again. Hope everyone had a chance to check out the poster session upstairs. We're going to start off with Harshal.
Awesome. Thank you. Again, this is amazing. Compared to last year, where, you know, just the fill out in this auditorium is stupendous. I think we may have to consider hiring out the new camp next year, and possibly, you don't want to see me play football though. Yeah, thank you all for coming. It's been absolutely great talking to a bunch of you, meeting you in person, older faces from last year. And yeah, this event just keeps on getting better and better, and hopefully next year is going to be even more stupendous, especially after last night and what we've got planned potentially tonight as well. So yeah, lovely to see this. What I want to talk to you about is how to set up automation on the Seqera platform. So before I do that, I just want to set up some audience participation here. Whenever I say tower, I want someone from the audience to correct me and say Seqera platform. I know you're thinking it. I need to wipe out 2 years of muscle memory overnight, so I need your help. Some positive reinforcement might be good as well. Okay, so I head up at the scientific development team at Seqera Labs, and we do various things. So we're typically bioinformaticians, but we're sort of Swiss Army knives in a way that we interact with various departments and do various things, and it's awesome the team that we're building at Seqera to do various things, whether it's developing the product, the people team, the marketing, engineering. Everyone is just been awesome to work with, and we're quite a fluid team. We still are quite heavily vested in open source and community contributions, like Evan alluded to yesterday, and so we're still contributing quite a bit back to NF Core. Our primary goal also is to keep our customers happy and build relationships, and also to help them be successful in their environments. So just a brief overview of the Seqera platform. Some of the benefits Evan showed you yesterday. There's also a ton of other stuff like cloud expenses and various other things that really help to collaborate and run pipelines in a single environment. But as I said, I want to talk to you about the automation aspect. As bioinformaticians, we love automation, and it's something that is really important to me. It has been also with a lot of the stuff that I've helped implement on NF Core and various other places. If you can automate stuff, just do it. It makes your life so much easier. There are various use cases for automation. So UIs, wet lab scientists that may not necessarily know how to code, just want to click a few buttons and run a pipeline on something like Seqera platform. APIs, if you've got existing databases and various other things that you've already integrated into your own infrastructure, and you may want to leverage an API to automate that process, that's another option for automation. CLIs, bioinformaticians tend to love CLIs because you install things on a command line, it's easy to test, it's easy to develop, and it's also easy to interact with. And so these are sort of the use cases that you typically have. But it's not necessarily one or the other, it's more of a continuum. So I, for example, may be incredibly lazy and want to use the UI just because it's convenient. I may also want to use the CLI to do the more mundane tasks that I have to do all of the time. And so there are various use cases for automation. As Evan showed you yesterday, you can obviously add a pipeline to Seqera platform and run it via user interface by clicking on a few buttons. So that's just simply a UI type launching. Via an API, so Seqera platform, I haven't said tower yet. So on the Seqera platform, what we have is an API, and so it's essentially composed of 3 components. You need to understand the basics to get how all of this messaging and communication happens in automation. And so what we have at the back end of Seqera platform is a database. Now, that database holds all of the information that you see when you open up the user interface on a website. It holds all of that information in a database. So that can then be accessed via API calls, which is one layer of automation. And so here you can see there's sort of a Python way of posting a request via an API call. It's not my favorite thing, but it works, where you need to integrate API calls into existing systems and that sort of stuff. And hopefully this is just mostly to show you the flexibility that you have on offer. That API then speaks to the front end. So you've got a database at the back end, you've got a back end container that is making all of these API calls and speaking to the user interface. So whenever you see a pipeline running in Tower or other various metrics, that's all happening via API calls behind the scenes. We've made that open, so you can leverage that for your own automation. But what we've also built on top of that is the Seqera CLI. And so the Seqera CLI behind the scenes is also making API calls, but it's doing it in a very nice user-friendly way. And so there may be instances where you want to launch a pipeline via the command line. And it is behind the scenes, it's chaining together multiple API calls, so you don't have to. And so it's a convenience function that just makes it a lot easier for you to interact with the Seqera platform. Another nice thing about it is that what you can do is you can import and export entities in Tower. So everything that you create in Tower, when you click a button and say add, everything you say in Seqera platform, when you create an entity, is essentially just a bit of code behind the scenes, right? And so what you can do is you can import and export that out of the Seqera platform and store it as code. This is also nice because you've essentially now got infrastructure as code. Everything that you've done and created in the Seqera platform is stored as code. You can version control that, and you can update it, and you have better provenance as to exactly what you've done and how. To take it a step further now, what we've built as a team, right? So it's a bit of a cascading thing. So you've got the API, the CLI is built on top of the CLI, and us as a team have built Seqera Kit, which is built on top of the CLI. And so essentially what Seqera Kit is, it's an interface to the command line. And so all of the options you typically would see when you run the command line are exposed in a standard YAML format that you can then specify to create virtually anything on the Seqera platform, as I'm about to show you. You can also now, because this package has been written in Python, you can import Seqera Kit and hence the CLI functionality directly into Python for you to leverage your own automation. And so now this is opening up the ability to automate your internal processes via Python, which I think is going to be very powerful. There's tons of stuff that you may want to do dynamically via scripting that you can now do and don't necessarily need to do it via user interface or via an API. So I'm gonna show you, give you a live demo. You only live once. I'm not sure how I'm feeling about it right now, but the fire exits that way, by the way. Okay, so what I've done is essentially I've installed the Seqera CLI locally. I've just created an alias. This still needs to be, we haven't rebranded it yet. We're in the process of doing that. We're going to do it this quarter. But I've just created a local alias that essentially aliases the TW option to Seqera. And it has a number of options, right? So it's a CLI, so you can do virtually, as I said, virtually anything that you want to interact with on Tower, you can do via the command line interface. Thank you. Right, so that's the CLI. Now, what I want to show you is there are ways that you can essentially abstract away things like workspace names, organization names, users, and everything, and create a standardized format to define a set of variables. So these are the things that you most likely will be changing whenever you create a new organization in Tower, a new workspace, or launch a new pipeline. There are some things that you need to change all of the time. Other things remain constant. And so the way that we've developed Seqera Kit is that you can specify just standard environment variables. You can, in this case, I'm going to source some secrets here as well. I don't want to air my dirty laundry here on stage. And so these are in a file, and they're things like AWS permissions and various other stuff. So all I really have to do is source this single file. And all of these variables are now, or should be available to me in the executing environment there. What I now want to do is take a YAML that defines everything that I want to create in the Seqera platform, all the way from an organization, from scratch, adding teams, workspaces, participants, credentials for all of these different, in this case it will be for AWS Batch, adding a compute environment and utilizing the batch forge mechanism that we've got on Tower that will essentially go and spin up everything that you need to do without you even needing to touch the AWS console. Using Fargate in this case as well to spin up these instances very fast. Big thanks to the engineering team who released the CLI with this functionality for us this week as well. It's going to be really useful to show that. And then once you've got the compute in the Seqera platform, you then want to add your pipeline and run it, right? So what I'm going to do is I'm going to just run Seqera Kit, which I've got installed here. Just check it is. Oops. Did I spell it wrong? There we go. So it's installed there. So now all I have to do is provide this YAML to Seqera Kit. What it will do is it will work its way through that, and it will go and create everything in the platform for me. And so you can imagine this YAML file being a source of essentially infrastructure as code that you can maintain. You get a bit of dynamic, the dynamic aspects by using environment variables. So things that you want to change, you can encode in a different way. At this point now, what's happening is Seqera Kit is going in and it's creating the AWS compute environment for me in the workspace. Let's go and have a look at what that looks like. I'm just going to log in. So here's the brand new organization that I've just created from the command line. I've also added Adam and Isha. I'm going to give them higher roles, so if anything breaks, I can blame them. And then if we go into this workspace here, this compute environment is now available. The pipeline hello world is just being added. So if I refresh this page again, the hello world pipeline's been added, and it's also been executed on AWS Batch, all within one flow. What I want to do is take it a bit further now and also replicate what Evan done via the user interface yesterday. So what he done was used this fetch NGS pipeline with a set of IDs from a study that he found on the SRA. I'm going to use the latest version of fetch NGS, which comes with all of the NF test magic that we implemented at the hackathon just to top it off. And so what I'm going to do is I'm just going to run a separate, smaller YAML that just literally just adds a data set. And in this case, that data set is just a few IDs, database IDs. It will add the data set for me as well as the pipeline directly into the user interface. So again, all I need to do is that. If I refresh, I've now got the fetch NGS pipeline here. I've got that data set. I can now launch this pipeline. So this would be like a UI type launch. So let me just rename this. I'll provide as input to the data set that I just added. So this would be how you would launch this pipeline from the user interface. Oops. Give me permissions. Okay, so because I've just created this organization from scratch, that's probably why I've got these restrictions on here. There's nothing really I can do about that now, but hopefully it highlighted to you by pushing up these assets directly to the Seqera platform. It just makes it a lot easier for you to manage and maintain this overall. And so what I can do is I just attempted to show you how to launch it via the UI. Normally it would work when you create the organization and you've been given the appropriate permissions. What I also want to show you is essentially doing the same thing via a Python script. So here, all I'm doing is specifying the workspace that I want to run, the dataset name, the one that we just added into the user interface. This is just going in and fetching the dataset ID directly from the Seqera platform. I'm creating a temporary parameter file for these parameters within Python, and then launching this pipeline. So this is most likely not going to work either because of the workflow limit, but I just want to show you what it looks like. Oh, you did? Beautiful, thank you. There we go. Such a great team. Right, so here now, what we should see is we've just launched the same pipeline from Python. I'm gonna go and launch it now just to complete the circle via the user interface. Right, so hopefully this shows you that you can automate your processes via a native CLI. You can have YAML, infrastructure as code. You can even have Python. This entire end-to-end process in terms of going from nothing to running a pipeline and having everything you need from credentials, users, a potentially complex compute environment in the cloud, adding a pipeline, running that pipeline. I think it's probably taken about 2 minutes for me to do that end-to-end. If I were to share this, maintain it, and I want to spin up additional workspaces in the future, it just makes it a lot easier to do that. So to highlight that, what my lovely volunteers in the front have done is as soon as I've created this organization, they've created workspaces and have triggered exactly the same things on Azure as well as GCP. So these pipelines are now running on multiple clouds within the space of about 10 minutes. So that's really the big advantage of this, and I hope it's useful for you to see exactly what benefits you can get from this sort of thing in terms of having infrastructure as code. You can push this up to GitHub. Anyone can run it, and it sets up everything you need directly within the Seqera platform. Thank you for listening, and I hope that was useful.
Good thing we have some time for questions, because there's a lot of questions. So first of all, you mentioned GitHub. Would any of the examples that you showed right now be available on GitHub?
Absolutely, yeah, sorry, I forgot to mention that. So on the Seqera Kit website, what we're trying to do is, what we've done essentially is we've added a bunch of templates here for you to start adding your own YAML files with some documentation. This is likely to change depending on what happens in the CLI, but we'll try and keep on top of that. It's in beta form, but please come and use it, break it, give us your opinions. We want to improve it. We use it every day. If you find any benefit in using it, then we'd also be great to hear your feedback on how it's working for you. We've also got some end-to-end examples here where the Python script that I just showed you, this is just a simple hello world example of that you can use and build out your own processes, as well as a YAML that shows the entire end-to-end process. And actually, Ish has also created a pull request that adds these templates that we've run for you today via pull request into this public repository, so they'll be accessible for you to go and browse and play around with.
Another question from Geraldine. Could you use this to set up GitHub Actions to automatically launch a pipeline after, for example, committing some changes or releasing a new version?
Absolutely. So because it's in version control, it's in a GitHub repository, we can set the GitHub action to do virtually what we want. And in fact, we've got a workflow here that runs the end-to-end testing workflow YAML file that I just showed you. It's, again, injecting all of these environment variables that we need to change that might be variable. It's injecting all of these into the environment, and it's creating everything for us. So we can absolutely use it as part of CI/CD, and we have been doing that. It's great. The plan is potentially in the future also to maybe roll this out on NF Core, because we're using Tower for a lot of We're using Seqera platform for a lot of our CI/CD on NF Core as well, because it's just a lot easier to create, these cloud resources very easily. And in the future, the plan is to have these simple sort of templates that we can use with NF Core at some point. So hopefully it'll be there soon.
Another question came from Stav. Can you do environment variable substitution on the YAML with Seqera Kit?
Yeah, so if I understand that correctly, if we look at this, this YAML that I've just used, the organization name is actually exported into the environment there, which is in this case Seqera Kit. So if we do echo organization name, it's Seqera Kit. When you run Seqera Kit, what it does is it scrapes your local environment for all of the environment variables that you've put into there, and it will use them and substitute them wherever it needs to within this YAML. And so in that way, you can do other things. You can have a time variable that is just bash returning the current timestamp when you're running this pipeline, and you can even substitute time variables to have dynamic parameter naming for your output directories and various other stuff as well. So yeah, absolutely.
Another question from Raul. Is it possible to access Tower Forge functionality through API calls so that one could have code to define one's compute environment?
Yeah, you should be able to create compute environments via API calls as well. So anything that I'm showing you now that I'm able to do via Seqera Kit, you can do via the command line interface. Anything that you can do via the command line interface, you can do via the API. And so it just bubbles down in terms of functionality.
And there was another question. Can you take use the Seqera Kit to take down the infrastructure after pipeline is complete, so you don't end up with compute environments from previous test runs?
So that's an excellent question, and actually Isha has just recently added a delete functionality to this. So where is it? It's here. Here we go. So yes.
Perfect. I think that's it we have for questions for now.
Awesome. So yeah, I'd just like to thank Isha, who's mostly been working on this. So take a bow, my wonderful volunteers. And everyone at Seqera, yeah, it's just been awesome. I know that I'm not going to name drop everyone, but this just wasn't our work. Everyone was involved, engineering. Again, other people within the company were involved to get this over the line. So yeah, thank you all again for the effort you've put into this.
Next up, we have Sabrina Krakow.
Thanks. Yeah. Hi, everyone. Hi, everyone. I'm from QBIC, the Quantitative Biology Center, which is the bioinformatics core facility of the University of Tübingen. And I'm super happy to share with you our recent work on a new Nextflow plugin called NF CO2 footprint, which is to estimate the carbon footprint of pipeline runs. So why is this important? First of all, because in general, data analysis, but also in particular bioinformatic analysis are contributing considerably to the human emissions of greenhouse gases and thus also to global warming. And I would like to give you an idea about the impact of this. So I would like to show you a few examples of a recent publication by Grayley et al. And they showed, for instance, that a de novo assembly of the human genome from Illumina reads generated roughly 15 kilograms of CO2 equivalents. So maybe a brief explanation about this metric CO2 equivalent. This is used for a better comparison between different greenhouse gases. So if other types of greenhouse gases are generated, this is converted to the amount of CO2 which would have the same impact on global warming. So back to this 15 kilograms of CO2 equivalents, this would be comparable to driving 68 kilometers by car within Europe. The authors further estimated that a metagenome assembly from 100 soil samples generated an amount of 77 kilograms of CO2 equivalent, and this would be comparable to driving 440 kilometers by car. So this should be addressed somehow. And in order to do this, we, on the one hand, need to increase the general awareness about this, and importantly also to deepen the understanding of which are the most influencing factors here. So for this, we need to estimate the carbon footprint based on an estimation of the energy consumption. And since actually tracking the real power consumption is coming with its own challenges, I will focus here on a method which estimates the energy consumption. And in this context, I would like to highlight the Green Algorithm project. This is a pretty nice project which was developed by Loïc Lane-Longue from the University of Cambridge, Jason Grayley and Michael Inhoye. And they developed an online calculator, which is called the Green Algorithms Calculator, which you can see here. And you can provide the resources which were used by your computation or your algorithm, such as time and so on, and also specify some details about the underlying hardware and where your computations were run. And then it estimates the energy that was needed for this and also the carbon footprint. And since these numbers are relatively difficult for most of us to interpret, they also provide some nice equivalence values for a better interpretability. So for example, how long does a tree need to sequester this amount of CO2 again? How far could you travel by car and how far by train? So this tool is pretty nice. We used this at the beginning of the year already to estimate manually the carbon footprint of some Nextflow ones we had. And then it became apparent that it should be somehow implemented and used for Nextflow as well. So first I would like to show you how the actual underlying method works. Here you can see the equation. So one can compute the carbon footprint or the amount of CO2 equivalent by using the energy that was needed for the computation and then also the carbon intensity of the respective energy production or power generation. And the energy needed, in turn, can be estimated with this relatively simple formula. So we first need the running time. Then the formula also contains a term describing the power draw of the whole processor. This comprises the number of cores which were used, the usage factor, and also the power draw for one individual core. And these values or approximations for this can typically be retrieved from the manufacturers. Additionally, the formula contains a term describing the power draw for the memory. This comprises the actual available memory in gigabytes as well as the power draw per gigabyte. And in this context, it might be worth noting that there's no usage factor included in this part because from the power consumption of the actual memory, this is mainly independently of if the memory is actually used or not. Additionally, the formula contains a term called PUE value, which stands for power usage effectiveness, and this describes the efficiency of the data center. This is important because the data center does not only require energy for your actual computations, but also for cooling down the whole system, for example. And last but not least, it's also including a pragmatic scaling factor, which includes the fact that most of the algorithms are run multiple times before the actual final results can be generated. And when looking at this formula, we can see already what are the main important factors which are contributing to this. So first of all, the efficiency of our computations, so how much time, memory, and how many cores do we need, but also the requested resources. So for example, if we over-allocate memory, this has a bad impact. then the efficiency of the data center is important, the PoE value. So larger, more optimized data centers usually have a lower PoE value, and also the behavior of the user is important, of course. If we look at the actual carbon footprint which is resulting, for this also the carbon intensity plays a big role. And these carbon intensities depend on the actual sources of the energy mix which is used. And this hugely varies between different geographic locations, but also within time. And there are different data sources contributing or having such carbon intensities over different regions. And the green algorithms calculator, for example, uses a relatively or a bit more static version, since this is also only applied after the actual computation. However, there's also a source called the electricity maps, which provides real-time data and also some nice visualizations, as you can see here. And when I checked a couple of days ago, just to show you an example how big the difference of these values can be, in Sweden there was a carbon intensity of 12 grams of CO2 equivalent per kilowatt hour, while at the same time the value in Queensland in Australia was 682. So this shows how huge the difference can be, which is mainly caused because Queensland relies on coal while Sweden uses mainly renewable energy sources. So with this we have seen the underlying method and which type of data sources are important for this context. So we made use of these methods and implemented this into a new Nextflow plugin, which is called NF CO2 footprint. And this is basically using the same underlying equation, just that we use this for each, or compute this for each task of the pipeline on the fly while making use of the Nextflow trace matrix. So which matrix are we actually using? So we use the real time, then the number of CPUs, the percentage of the CPUs which are used by the tasks. Then we added also a new entry to this trace matrix called CPU model, which contains information about the actual model, which can be used to retrieve the model-specific power draw values. For this, we make use of data which was provided by and collected already by the Green Algorithms project, and we also make use of the memory which was requested. So these are the trace matrix, which are the values of the plugin which are automatically obtained during the run. Then the plugin also comes with some configurable parameters. First of all, the power draw from the memory per gigabyte. This was estimated to be, in the past, to be a relatively constant value across different types of memories. So for this, we can simply use this default value. Then the efficiency of the data center, so the PoE value is important. There it might make sense to retrieve this value from the actual data center. And further, the users can also provide a carbon intensity value, which is in case they have custom one, or apply or provide a geographic location. And in this case, again, data from the Green Algorithms project is used in order to then retrieve a location-specific CI value. So this is the main basic concept of this plugin. It then generates multiple different output files, importantly an HTML carbon footprint report. So this provides some general information about the whole pipeline run, for example, the total CO2 emissions of the whole pipeline run, the energy which was consumed, and similar to the Green Algorithms calculator, also some equivalence values. So how many tree months would this correspond to, and how many kilometers by car, etc. It also comes with, includes a few plots, for example, a plot showing the CO2 equivalent distributions across different processes, so that we can see on the first view which processes have the main impact in this case. And of course, it also provides detailed information about each individual task. We also applied this already and tested this for the NF Core Mag pipeline. This pipeline is for metagenome assembly and binning, and we run this on our local compute cluster while requesting different numbers of CPUs for the metagenome assembly step, which was performed with Spades. And you can see here the resulting, first the resulting carbon footprint, then the real times and memory values, used memory values across different numbers of CPUs. And what we can see here is that when we look at the carbon footprint, the value is first going down for an increasing number of CPUs and then going up again. And the reason for this is that the decrease in in the actual running time does not compensate anymore for the higher energy consumption due to the higher number of CPUs. So this shows that this can be and should be used to optimize also the pipeline settings in order to reduce the carbon footprints. And with this, I'm already slowly coming to an end. So to conclude, once this will be released, this will be super easy to use, and we hope to increase the incentive to reduce the carbon footprint of pipeline runs. And we think this will also help the user to optimize the pipeline settings, to carefully select the data center, and to reduce the number of runs, at least where possible. It's already available, and we also set up some brief documentation already about the important parameters and how you could already test this even prior to release in case you're interested in. And also there's a Slack channel in the NF course workspace in case you have any questions, feedback, or if you want to contribute. Beside the release, the next steps would be to include a more fine-grained real-time carbon intensity data, such as you showed you for the electricity maps. Then to add also support for cloud systems. And finally, it would be also nice if this will work for the Seqera platform to monitor the carbon footprints for multiple ones, and also to recommend eco-friendly pipeline settings or even to automatically optimize this in the future. And with this, I would like to thank you for your attention. Importantly, Julia Mir for contributing. So both of us are the main developers so far. Then Loic and his colleagues from the Green Algorithms project for previous work and sharing the data. Then Phil and Paolo, of course, for many contributions, help, and suggestions. And thanks again.
We have a few questions here, and thankfully a couple of minutes for that. Would it be possible for this to support GPUs as well, not just CPUs, for the plugin?
What?
For the plugin, does it support GPUs as well?
It does not support GPUs yet, so this is an open to-do. It was also, since recently, the Nextflow trace matrix did not support GPUs also in the past, so, but yeah, it's a to-do.
The other question coming from Chris, would it make sense to take into account in the formula for the energy, the cost of manufacturing the processor instant machine type?
The embodied emissions for the hardware, or what?
Yes, so the formula to include, to calculate also like the type of machine or how expensive it was to manufacture it.
Yeah, so far this is all embodied emissions are not included in this formula, and also now, for example, storage is also not included because this is really focusing on the actual pipeline one. But it could, of course, be extended in the future for different factors.
The other question is kind of in similar mode. Does this support different architectures, like ARM versus the x86 and like different architecture of the machines?
Yeah, so there are powered raw values. For example, like for this, we need the information about the hardware. And if there's no if there's no information about this provided, then we need to fall back to some reasonable default settings. And this is an open to-do also to include more data about this in the future.
The other question comes from Max. Would it be realistic to move computations to low CO2 intensive regions? And what do you think are the possible opportunities and hindrances in this?
So the first part.
So if you are to use this to move the computations to the low intensive CO2 regions.
Yeah, this is of course like one conclusion one could get to see, okay, in which regions would this be more efficient to compute, while also of course taking into account the PoE of the data center. So these are two factors which should be considered together for this aspect.
Another question is, what is the cost of carbon footprint when using the NF2 footprint?
Of running the actual, it should not be too high. We tried to make it as efficient as possible. I mean, as I said, it's using the already recorded metrics, which are already there, and just summing them up and using a relatively easy equation. But we could measure this, of course, in the future.
Adrian is asking, in your experience, what are the least and the most carbon-intensive pipelines?
I did not apply this to too many pipelines, but the meta-genome assembly pipeline, for example, I mean, it depends on the input data. I mean, I run it on the Mac pipeline using the full-size test data set, but this is still compared to the most real data, still relatively simple. But if one would include, for example, as in the introduction said, 100 soil samples, this would be massive.
Great, and let's go for maybe one last question. Give me 1 second, sorry. A lot of it is about, like, a lot of the questions coming through are about whether or not we can use this for the Seqera platform as well. So I think that's a question not directed at you, but I think we've gotten most of them, so we're good.
Okay,
Thank
thanks
you.
a lot again.
And we have Victor Perez up next.
Hello, everyone. First of all, thank you to the organizers of the summit and the hackathon. It's my first time in the summit, and it's been a very enjoyable experience. So I want to talk to you about an image analysis pipeline that is used in multiplexed imaging and spatial omics. But before going into the topic, I would like to thank my colleagues and collaborators in this project. So my colleagues from the Institute for Computational Biomedicine in Heidelberg, external collaborators in Tübingen, in Seqera Labs, and as well to the original development team of MC Micro. So as I said, I want to talk to you about an image processing and analysis pipeline, but I also want to show you the context in which this pipeline lives. So I will make an overview of what multiplex imaging and spatial omics is. Then I will guide you through the pipeline step by step. And finally, I will show you the work that has been done to transfer this pipeline into NF-Core. Okay, so what we're seeing here is a small region of interest in tissue. The first goal of spatial omics is to study cell interactions, not in small regions of interest, but rather in whole tissue. Yeah? So the second aim is to investigate the interactions in space of different cell types. And for that, we need to phenotype these cells. That means we need multiple markers to assign the cell types. And this is done through the technology called multiplexed imaging. So in multiplexed imaging, or multiplex imaging is a family of methods that allow the cyclic staining of samples. One can have multiple cycles in a single run, and within each cycle have 3 or 4 markers. So at the end, one ends up with 20, 40, or even hundreds of markers in the same sample. Just as a personal anecdote, I was in the field of microscopy 5 years ago, and back then people was happy with 4 or 6 marker stops, yeah? So the progress of multiplex imaging, it's just tremendous, yeah? So the data set that I showed before, it's a 20-plexed image. That means it's composed by 20 channels or markers. And now I'm going to show you what is the general framework of spatial omics. So we take our multiplexed image, we use the channels staining the nuclei and the membrane to perform segmentation of cells. Segmentation means locating the cell and also getting to know the area that occupies in the tissue. Once we have isolated the cells, we use the expression patterns of the markers across all channels to assign phenotypes to these cells. Yeah? Okay, so this is more or less the framework of spatial omics, but I also want to show you what is the relevance of the field. I will change the sample, just hold on there. So I'm showing you now tumors in breast tissue. Yeah? There are 3 tumors here. They're classified as mixed, compartmentalized, and cold. Yeah? There are 2 types of cells there, immune in orange, and tumor cells in blue. So the classification has been done using the information acquired from the spatial organization. For example, the mixed, if we only look at composition, the mixed and compartmentalized tumors look basically the same, yeah? So there is no extra information there. But thanks to the spatial organization, we can see how the cell types interact and create new classifications of these tumors. So the relevance of this is that it opens the doors to patient stratification and also potential custom treatments. Okay, so this is the framework where the pipeline exists. Now, what do we need to implement it? So we have a bunch of algorithms, and each of these algorithms have its own required libraries and packages. Yeah. We also have to deal with the different output formats, yeah, from all the new technologies that are coming out there. So even if we have all these variables, there is a blueprint for this pipeline. And I'm going to go through the pipeline step by step. Yeah? So let's imagine that one of these devices produces tiles as raw data. As I said, we are interested in investigating tissue, whole tissue images. So we need to stitch these tiles to have a single image. Now, as you might see, the image gets dimmer in the edges of the tiles, and this is due to the inhomogeneity of the illumination. This is just a limitation of the microscope, so we need to correct for it. Yeah. And once we have applied the illumination correction, we go to the step called channel registration. So we need to remember that we have different channels in a data set. These channels are acquired in different cycles, so it's very likely that the sample will drift during acquisition. Yeah? So we need to correct for this drift and align all these images in the XY plane. Once we have the channel registration, we can also enhance the contrast of the image by correcting the autofluorescence. Yeah. And finally, once with the enhanced contrast, we can apply segmentation algorithms. For that, we require the nuclei channel. So we segment the cells. And finally, we can extract quantifiable features that will help to do later the spatial analysis and the phenotyping. So this pipeline, the blueprint of this pipeline, has been already assembled in Nextflow. This is a paper from 2021. It's Dennis Shapiro, my supervisor. Yeah. But now we want to go to the next step of standardization. So we want to have our pipeline curated. We want to have it tested, and we also want guidelines to include new modules in the pipeline. So the natural step is to go into NF Core, and we've been working lately to do this. What we've been doing so far is first include the modules that are already marked with green. Yeah? These modules are already NF Core modules, and we are using them to assemble the whole pipeline. This is currently work in progress. We will be using the sample sheet to handle multiple data sets in a single run. We also want to have optional modules. That's why it's MC Micro, multiple choice. Yeah. But also we want to take into account the devices that are, the acquisition devices that are out there. So we would like to include staging modules before injecting the data set into the pipeline. Yeah. So I would like to have this, my aim is to have this ready by December, yeah? The pipeline and at least one of the staging modules. And before ending my talk, I would like to do just some shameless promotion. My colleague Krešimir Bestek is presenting a poster in stand 9. Look for him. It's also an image analysis pipeline. He's in the very corner. And if you're interested in image analysis pipelines, just reach out and let's connect. Yeah? Thank you.
Thank you very much, Satish. That was fascinating. And we have a few questions out here. One is, what was the most challenging in bringing the non-genomics pipeline to NF Core?
In bringing what, sorry?
The non-genomics pipeline to NF Core. What do you think were the biggest challenges in that?
I think it's finding the right algorithms for it, yeah? Because you could add as many modules as you want, but yeah, we want to keep this blueprint simple, yeah? So not to have so many tunable parameters. Yeah? So it's conceptual. The challenge, it's more conceptual, yeah?
The other question is more specific. What is the limit on number of channels? How many colors can be distinguished?
Well, so far, I've worked even with 100 channels, yeah? I can just talk from experience. I've seen 20, 40 channel data sets, but also I've worked with 100 data sets, yeah? 100 channels data sets, yeah.
We have a question from Ben. Are there any potential Nextflow features that would make it easier to develop and run your image processing pipelines?
Any Nextflow features? Mm-hmm. Well, I don't know if, so as I said, the pipeline, it's already assembled in Nextflow, yeah? We were working with it for about a year, but we realized that we need guidelines for it, yeah? And so those tools are currently provided by NF Core more than Nextflow, yeah?
What is the most challenging step in the pipeline in terms of algorithmic complexity?
In terms of what, sorry?
Algorithmic complexity.
I mean, all these modules do a different thing, yeah. I won't be able to only mention one. Yeah. Each of them make their own task, and they do it well, yeah. But what is more complex, it's difficult to rate that.
Is it possible to run the same analysis manually on a researcher laptop? Or what are kind of benefits of performing this sort of headless image analysis with Nextflow?
Well, I mean, that will depend on the size of the data set, right? And the size of these data sets can be up to 1 terabyte, yeah? So it goes from 500 gigabytes to terabytes. Yeah? So I'm not sure if you can just simply perform that in your laptop, yeah.
And then, are there any of the tools used by the pipeline optimized for GPUs specifically?
No, we're working on implementing that. Yeah, we're working on implementing that because not all modules require GPU, so we will have to configure the process that really require GPU. Yeah.
Great, and maybe one last question: is there any other pipelines already available or packages to do the same?
Not really. No, as far as I know, there is.
So that's the first one.
Yeah.
Great, thank you so much.
Thank you.
We have Satish up next.
Thank you. Hello, everyone. So good to be here. So today I'll be talking to you about harnessing our platform, which is called Memory Machine Cloud, or MM Cloud, for efficient Nextflow pipelines and tapping into cost-effective and dynamic resource management. we all love Nextflow because of its scalability. Nextflow was designed from ground up so that you can run it on your laptops, on HPC clusters, and on the cloud with a simple change of configuration. And this is possible because of the decoupling of the configuration that Nextflow allows for. And currently, the platform Seqera supports different cloud providers, right? So you have AWS Batch, Azure, and Google Cloud. So for people who are still not using cloud, I've just listed out some of the chief advantages and disadvantages of cloud here. You can see, so the chief advantages of the cloud is that you pay for what you use, and also you don't have to maintain or own these resources, so it's not your problem. You just pay for the service. But with these convenience, there are also disadvantages that come along with it, is that there's a lot of wasted cloud resources, and we just heard from Sabrina and just bringing about the awareness about our carbon footprint of our pipeline runs. Running jobs on cloud can become quite expensive very fast with some unforeseen compute. You left some pipelines running that can add up and cost you a lot. You have long-running jobs at the risk of disruption. And there's a steep learning curve for adopting the cloud, so it's not a simple drop and replacement as well. So with these, also for people who are not familiar with the cloud offerings, you have two different kinds of instances that the cloud providers offer. Those are the on-demand instances versus spot instances. So on-demand instances are you pay a fixed price for a guaranteed service, whereas with the spot instances, you have these hyperscale cloud providers auctioning off their excess capacity at a fraction of the price, and usually these discounts range between 50 to 90%. So we just launched a tool on our platform, and we call that the Wave Radar, where you can clearly see the difference between the current pricing estimates of on-demand and spot instances. So the dilemma here is, do you opt for a stress-free premium of guaranteed on-demand instances, or do you take or play the gamble with spot instances? Sorry, I forgot to mention the spot instances, which are cheaper, come with a catch. They can be taken back by the cloud providers on a very short notice, usually 2 minutes in case of AWS. So do you want to pay more for the on-demand instances, or do you opt for spot instances but handle that they can be taken back by the cloud providers at any time? So it's always this dilemma, but managing costs is a big issue when you are running your workloads on the cloud. Currently, Nextflow allows for these error handling mechanisms. By default, if you're running on spot instances, Nextflow has a spot retry attempt parameter that is defaults to 5 times. It'll try for different spot instances before the task fails. But the essential point here is that if a spot instance is reclaimed, the Nextflow is basically going to rerun your entire job. So imagine if you had an alignment job that was almost finishing, and there was a spot reclaim, and essentially it's going to retry and start the job from all over again. So considering the carbon emissions, it's a waste of time, it's a waste of resources, and also not friendly to the environment as well. Also, not only just spot reclamations, we all have seen processes where they fail due to insufficient resources. They're running out of memory, or the CPU utilization, CPU cores aren't enough. But Nextflow also has dynamic retry strategies, right? So where you can set the amount of resources that it needs to retry again based on the task.attempt. But this is, again, essentially, it just adds up to your cost, because if the instance has been reclaimed, on the next task attempt, you are basically asking for double the resources, which is like twice the cost. So it quickly adds up when you consider if you're using these cheaper instances, but every time it's just adding up to your costs. So with that, we would like to introduce our platform. It's called the Memory Machine Cloud. It's basically a container orchestration platform for running data-intensive workloads and also an interactive applications on the cloud. Currently, we support AWS, Google Cloud, and also Ali Cloud and Baidu Cloud on the Chinese market. So to dive into what are the features that are available on the platform, we essentially provide a CLI to interact with the cloud. So we call this the float. Float is the CLI and also a job management service. So with this CLI, you can submit jobs to the cloud, track them, manage all these resources through the CLI. Also, we have something that's not currently available on AWS Batch and also on platform Seqera at the moment is real-time monitoring of CPU and memory usage. So you can clearly see the CPU and memory usage of a particular job in real time using the Wave Watcher profiles, and also carbon footprint. So we are not providing on a granular level that Sabrina has shown along with the NFCO2 footprint plugin, but we do track them on a workload basis, on a higher level basis. So offer these Wave Watcher profiles. So the core service that we are offering is what we call as the Spot Surfer. So this is basically, just to take an example, imagine you have a pipeline running, say RNA-seq. I have a job up shown up there. It's called the coli map. It's one of the processes in RNA-seq. So what you're seeing here is the memory usage. And it's almost, as the graph shows, imagine it's almost finishing up and a spot reclamation has happened. So now coming to the secret sauce that Memory Machine Cloud offers is that whenever a spot reclamation or a spot interruption notice is provided, we can take a snapshot, freeze the container, take a snapshot of the application that's running, move it to another new instance, and restore the application on a new instance. So in this way, essentially, you are not losing your progress. In the application's perspective, nothing has happened. And in this way, you don't have to rerun the entire job again. So in this way, you can take the advantage of running on cheaper spot instances, also have the insurance that you won't lose your progress of your workflows, even though a spot reclamation happens. And you can see all these. We also have a user interface, but you can also monitor these via the CLI too. There are some exceptions, though, like when you have really large memory-intensive applications. Usually, the 2-minute warning that the cloud service providers provide might not be enough to dump everything that's in memory onto disk. So what we are coming up with is incremental snapshots that will be where periodically snapshots will be taken of your application so that you don't lose your entire progress and you don't have to restart from the beginning. Also, with this function, another feature that we offer is what we call as the Wave Rider. So with this, you can dynamically right-size your instances in real time. So this is because we are monitoring the CPU and memory usage in real time, imagine if an application is ever running out of memory, we can take that as a trigger, freeze the application, snapshot it, and move it to another instance. So what you're seeing here is an application where it started off on a 4 GB machine, and it was running out of memory. So the application was frozen, and then the next bigger instance is found and is deployed onto that instance, and it continues. Again, you can see as it starts hitting the upper limits of the resources that are available, the migration mechanism is kicked in, and it'll dynamically move your workload onto an instance that is a right fit for the workload. And the converse is also true. If your application is not utilizing all the resources that are available on a machine, you can configure it to make sure that it can migrate to an instance, a smaller instance, where it will be able to finish the workload there. So in this manner, you can make sure you're providing just the right resources for your workloads. And I believe this is in the interest of also raising more awareness about the carbon footprints of our jobs, because raising awareness is one step, but what are we going to do about it? So this is one way where you can make sure you are using just or getting the right resources that are required for your workloads. How do we connect with Nextflow? So we have developed a plugin. We call this the NF-Float plugin. And with this plugin, you'll be able to add on a simple configuration changes to your Nextflow pipelines. So as you can see, you just need to specify the plugin, the work directory, an S3 bucket, and provide your credentials. And all the jobs will be now routed to Cloud. So it's basically a replacement for AWS batch service. While Cloud is handling, by default, all the jobs that will be checkpointed will have the insurance of checkpointing and restore, and also being able to select different types of VMs, be it spot or on demand. So the two main features I want to talk about are that you can set, mix and match, or set individual VM policies to your individual process as well, using the with name selectors in Nextflow, right? So if you really have a job that, a huge job that you really can't afford on a spot instance, you can make sure that that particular job runs on an on-demand instance, while the rest of your workflow runs on spot instances, essentially giving you the mix and match configuration advantage. And also, as you can see here, for what I have in the second process, you can set the migration policies, really fine-grain, fine-tune your migration policies, where you can set the upper bound and lower bound utilization parameters for CPU and memory utilization, and set those either by default for the entire workflow, or you can also customize it for individual processes within the workflow as well. So just to provide a high-level overview with the plugin, the way we are deployed right now today is through AWS Marketplace. The operations center of Cloud will be deployed on an instance within your VPC, and all you need from there on is the additional config specifying the plugin that I just showed you, and also as you can see, once you have the plugin and the operations center set up within your VPC, all the jobs, the operations center will take care of spinning up the worker nodes, finding based on the user policies, VM policies that you set, the operations center will take it on from there. So because we are deploying within your VPC, data privacy is built in, so there's no sharing of the data essentially with the outer world. So now I want to talk to you about extending Cloud capabilities further with different file systems. So this is something that we have been working with Seqera, is genomic applications essentially are, or let me put it this way, cloud is not essentially the best platform for running genomic applications, which can produce gigabytes to terabytes of data, because these are stateful applications. You must all have seen the breakthrough fusion report that was put out by Seqera, where you can clearly see that the fusion file system has offered a more optimized and more highly performance-driven alternative for the current existing file systems that are available. So what we are trying to do is also extend this capability with Memory Machine Cloud. So currently we support 3 different file systems, the default S3FS, FusionFS, and JuiceFS. Currently, the checkpointing and restore software or feature of Cloud works only with S3FS and JuiceFS. We are currently trying to get it to work with Fusion. So hopefully we can, once we have that working, we can integrate directly into platform Seqera. And because from the perspective of all these 3 different file systems, Fusion file system is the most efficient and also the least resistance that requires the least amount of setup when compared to the other 2 file systems. So I just want to show some comparison between a recent benchmark study that we have done comparing AWS Batch versus Cloud. So what you're seeing here in blue is AWS Batch, and in orange is Cloud. And we ran the NF-core RNA-seq using the testful profile on using the full-size test dataset. And you can see clearly Cloud outperforms AWS Batch in terms of wall time, CPU time, and also in cost. So what you're seeing here in the third graph is comparing on-demand versus spot instances. So if it costs you $97 on AWS Batch for using on-demand instances, the same if data set that is the same pipeline, if you run it on Cloud using JuiceFS or Fusion, it costs you a fraction of what you would be paying for the on-demand instances. So we believe this is highly crucial where so that you don't rerun, so that you can make sure you utilize these cheaper instances, but also have the guarantee that you won't lose your work while we are performing our computations. So with that, I would like to acknowledge the Seqera team, especially Ben for helping with the plugin, Jody for helping with the Fusion integration, and Paulo for all the discussion and encouragement on bringing this platform into the Seqera community. And of course, for all these awesome pipelines that we use every day. And also, I would like to acknowledge the core developer team from Memberge for their hard work on bringing some innovative features so that you can interact with the cloud via CLI and also the plugin. With that, find us on Nextflow Slack. We have a channel called Platform Memberge. Drop in there and we can chat more. Thank you so much for your attention.
Thank you, Satish. We have a few questions here, but maybe we'll go for one and then break for lunch. Do you know if this approach would be transferable to GCP's preemptible instances?
I'm sorry, could you repeat that again?
Would this approach be transferable to GCP's preemptible instances, which are similar to the spot ones?
Yes. We do support preemptible instances on GCP. So the issue with GCP is on AWS, you get a 2-minute warning for spot preemption, but GCP is not so kind enough. Usually it's 30 seconds or less. So that's exactly why we are bringing up the incremental snapshot support, where you can set a periodic snapshot interval timer that will take incremental snapshots so that you don't lose the entire progress. You'll lose some, but that's a better trade-off in our view.
Great, thank you. The rest of the questions are in Slack for you. We'll see everyone back here at 14:30 after lunch.
Oh baby, I wanted you to be here inside my arms where the night goes down. Oh baby, I wanted you to be here when the sun comes up and the day goes by. Oh baby, I wanted you to be here inside my arms where the night goes down. Oh baby, I wanted you to be here when the sun comes up and the day goes by.
Excellent. Hopefully everyone got some delicious lunch. I see there's still some people coming in. We're going to start in just 2 minutes. We've got an awesome, jam-packed set of sessions for the afternoon. And first up is Paolo. So see a couple people still coming in. Lots of seats in the front. And we'll get started in just 2 minutes. Okay, so we're getting ready to start the first session of the afternoon, starting off with Paolo, covering converged computing with the Nextflow ecosystem. I'll turn it over to him. Yeah.
Thank you. Hi, definitely quite exciting to see all these people growing more and more every year. So first of all, thank you for joining. Super incredible event. And also this year is a very important year because we are celebrating 10 years since first Nextflow Commit. So it's very exciting. It was quite a long journey. So quite good opportunity to say thank you everybody who was involved into this project since the beginning. Pipeline developer, Nextflow developer, committer. We're just using Nextflow, so really big thank you because this is possible only by you at the end. So it's a collective project. It's not something driven by a few people. And also it's quite amazing to see how this idea managed to grow in such big movement, pipeline developer, adoption in the industry. And and the point is, the core idea is in Nextflow to enable the deployment of scalable pipeline in a portable manner in different infrastructure, and above all, enabling scientists to focus about their work, that is science. So all the goal, the vision Nextflow is to try to keep away all the complexity to deploy the pipeline at scale in different environments. So this is what is driving the technology. We want to continue to deliver with this technology. And the point is that computational data pipelines are becoming bigger and bigger, more complex. There are more cloud, more computational infrastructure, more services, Kubernetes. So to tackle this challenge that is growing, we want to evolve this technology from a single tool to something that is more complex. It is an ecosystem of technology. So also Evan was mentioning this yesterday, and the idea to have specialized component that take care of very specific aspect of this challenge. Today I'm going to talk on a few of them. So of course about Nextflow, and then Fusion, and way how these components enable the scalability of this pipeline in the Vision Nextflow. The first one, is Fusion. This operating system, not yet. File system that we designed to enable the scalability of data pipelines in the cloud for data cloud native pipelines. And why we need a specialized storage for data pipelines? Because to scale pipelines, the problem is not computing. We have plenty of computing. It's just enough having a credit card about the computing, but the point is how we can use efficiently the computing power. And the bottleneck in data pipelines is, not surprisingly, the I/O. This pipeline ingests terabyte of data, produce terabyte of data as well. So it's very important to be able to use the storage in an efficient way. There are a lot of file systems, distributed file systems for the cloud, but the point that they were not designed having the cloud in mind. So they tend to be, or they are expensive, or they have fixed location, or both of them, and above all, add new services that you need to maintain. So it's not what we would like to have. Ideally, it would be much better to use object storage, like S3, that is a real file storage for native cloud. But the point is that there is a mismatch between the interface of object storage and the tools that we use in data pipelines because they are not designed to work with object storage. And here we come with Fusion. Fusion is one thing that is doing is creating a transparent interface to allow to use object storage into your pipeline without having to reinvent the wheel every time. So just to give you a very simple idea, how does it work? This could be a basic task without Fusion. Essentially, if you want to access a file over S3, you need to use the AWS S3 command line to copy the data in your computer or in the container. Then you can run the tool read this file, for example, and then produce another file, and then you need to copy back these into the cloud, in the object storage. Basically, this is what Nextflow is doing for you when you use Nextflow with AWS Batch or S3 behind the scene, but even if this is done automatically, it's very inefficient, does not scale well, produce a lot of redundancies. If you are using thousands of files for each of these commands, there is a separate authentication, so it's not efficient. Instead, what we can do with Fusion is transparently mapping the data that is in the bucket to a local path. So if the data is my bucket some files, we can access it from the local path, from the local file system using /fusion/s3, my bucket, and then the complete path. Behind the scene, this tool is taking care then to translate this to API call to S3 for you. So one side is much more simple to manage the data because delegated to this driver, but also what is important that is more efficient. And this is a simple representation of how could run pipeline use AWS batch maybe, and then using the Fusion driver. So Nextflow launched pipeline job into the cloud, is run into container, and then there is your job. Here there is the Fusion driver. The Fusion driver run within the job container, is not installed into the compute node. So this is already a difference compared to other technology. And so essentially, this piece of software is creating this abduction layer between your job and the S3 storage. But the other important thing is what happens in here. The Fusion driver takes care to create a cache using the node instance storage. So if this node use a very fast instance storage, like could be NVMe disk or solid state disk, Fusion driver is able to speed up greatly the performance of your pipeline execution because essentially what it's doing is working the local disk in a transparent manner and then moving the data when needed to the object storage. And so, again, there are plenty of different implementation Fuse driver. We are seeing also this morning GSFS, SF3, SFS, S3 SFS, I don't remember the name. But the point, there are so many, but the point is that Fusion is optimized for data pipeline, and above all for Nextflow data pipelines. The most critical part is this idea to have a pre-fetching algorithm that download the data before it's written by your by your tool. So this greatly speed up the access to the data. In the same manner, there is a asynchronous parallel algorithm that upload data in background, so making it much faster compared to other solutions. And then there is another trick that apparently is simple, but is a game changer. I will show you later why. The ability to create symbolic link over object storage, something that is not natively supported by the object storage. And then finally, what is also very important, that there is really almost nothing to do to use this technology in your pipeline. Few lines, really 2 lines in your Nextflow configuration file, and you don't need to store or configure the cloud. You don't need to configure compute node, almost anything. And also remove the need to have a custom MEI when you want to deploy Nextflow and run into an AWS batch, because usually you need to create a custom MEI to configure AWS command line tools. Using Fusion, this is not needed anymore. You can use a stock MEI. So we wanted to compare, because this is a continuous recurrent question, how is different Fusion compared to other similar technology? We made a benchmark comparing Fusion with Airclone, is a popular technology that's doing something similar, and also Amazon AWS Mount Point that was recently introduced the past year. Also this tool mount the S3 bucket over the local file system. We use FIO, this is a popular file system benchmarking tool, to compare the different performance across different operations. Reading file, sequential manner, and random manner, small file, large file, also writing files. Let's see the reading of small files. Here we can see in both cases, these 3 tools have similar performance. Here, megabyte per second, so this is the throughput, how fast is the reading operation. The bigger is better, and the best one is Airclone, 90,000 more or less megabyte per second, both in reading, sequential manner, and random manner. Then there is Mount Point, and then there is Fusion that is 60,800 megabytes per second. So here what we can see that all the 3 tools benefit from the caching of the file over the operating system. Basically, since the file is very small, the Linux operating system is able to keep this in memory, and all these operations essentially are over the memory. So this is why they are very similar to this number. Also, Fusion is a bit slower because it managed a second layer cache. So this adds a bit overhead. But we see the benefit of this caching system here. There is a huge difference when we read big data files that are very common in our pipeline. So when we need to read in a sequential manner 100 megabyte files, Fusion is able to read at 6,000 megabytes per second. Our clone, 85, so it's a huge difference. Mount point, 3.28, so barely usable, I would say. Same figure, more or less, for random access data, large data file over S3. Fusion is able to do a 2,200 megabytes per second, Aircolumn 1.48, so there is not really comparable. Same things for mount point. Again, why so fast? Because Fusion download in parallel manner in a prefetching the data, storing locally. Your tool works basically with the local disk. So if you know that's very fast local disk, your pipeline is going to be very fast over S3. Something similar is happening also for sequential write and random write. Fusion is able to write a file with 1,200 megabytes per second, Airclone 137. Mount point we were not able to access, it was crashing in our test. Finally, random write, also similar figure, Airclone does not support, and not even mount point support random write over S3. So here I think this should clarify why this tool is different from the other, because it's optimized to access large data file over S3, something that is not happening with other solutions. And we want to also to make a benchmark when what happened when we run a real-world pipeline. We benchmark NF core RNSE pipeline using Amazon Batch, using on-demand instances with S3, Amazon FSX Cluster, so a shared file system in the cloud, and Fusion. Also, we use the test profile of NF core pipeline. This means that basically this is a data set that is somehow similar to a real-world use case. And here we are reporting the CPU hours, so how long run the pipeline. With S3, it's the slowest one, and we are expecting this more or less 100 hours to run this pipeline because there is a lot of data transfer, so tend to be quite slow. And using FSX is quite fast because we remove all the overhead on transferring the data, and this is expected. And we can see that Fusion can be at par with FSX, even slightly faster. So both of them are twice faster compared to the use of just S3. But even more surprisingly, the result when we look into the cost of these three different technologies. Here we're reporting the cost to run the pipeline, so the compute cost, and then the cost of the storage to store the data for 1 month, because maybe you want to retain the data over time, of course, or the result of the pipeline. And with the S3, we spent $124. With FXS, quite much more, because it's a separate service, tend to be quite expensive, and the cost was $293, and with Fusion, only $69. Here, it's not. No, but wait, there is an interesting point that how is possible that this guy is cheaper than S3? Because we say that Fusion is just an adapter layer of S3, so it should be the same. So this is something on which we spent some time to understand why it was going to be cheaper. It's cheaper because Fusion uses symbolic links. So basically, you create much less redundant data in your pipeline. It is avoid to decrease the cost of the storage. And why this is happening? Because when you use S3, you have the same input file that is used by, let's say, 10 tasks. You have 10 copies of the same file. If you are able to create the link, basically, you are just moving the symbolic key reference of the same file. So we have much less duplicated data into the pipeline. And also to show better this mechanism, we compare different pipeline And we measure how much data we're producing. So RNA-seq S3 was more or less 1 terabyte of data using Fusion, 90% less. scRNA-seq, 32% less. ATAC-seq, 44% less. SAREC is incredible. I was so surprised that I wanted to check twice the result. It's 61% less data. It's incredible. Okay. Yeah, I think that the message is quite clear. I think you should not use S3 without Fusion in your pipeline because you are just wasting money and time, essentially. Availability, this technology is available for all 3 major cloud providers. We consider Amazon S3 production ready. We are still working on Google Storage and Azure Blob Storage. It is available as a preview feature. And both of them can be used both using Seqera platform or Nextflow command line tool, just adding the configuration in your Nextflow pipeline. And OK, so this is a Fusion optimization of the storage. Then the other point is about optimization of the container. Container are a pillar in our data pipelines. And Nextflow was one of the first tools using container in data pipelines because allow us to capture all the dependencies into the workflow application and easily distribute to avoid all the mess to configure this software in the target computing environment. So it's super important, but also changed dramatically in the last year how containers are used into the pipeline. In the beginning, it was just one container and a few tools. Pipeline also, they were more simple. Now, pipeline much more complex, and then there is this pattern to use a different container for each task. So in a pipeline, you can have 20, 30, 40 different containers, and then maybe you need the container from Docker and the container of Singularity, and then maybe you need also the container for ARM. Also this morning, the presentation Amazon was mentioning about this. So it can become very complicated to manage all these packaging on container, maintaining a line, upload it to the registry. They have different credentials, so it's quite a mess. And somehow it's a paradox because containers were introduced to simplify the deployment pipeline. Now there is this new burden to maintain the container. Instead, with Wave, what we are trying to push is a different idea. Instead of having containers that are monolithic assets or images built by somebody ahead of the pipeline execution, we want to push the idea that the container has a disposable resource that is created on demand so that we can also optimize the container for a specific job in your pipeline. Maybe you can create a container with a specific tool and for a specific CPU architecture where you want to run your container to get better performance. Also, define the container by specifying what I want in the container instead of thinking how to build the container. I don't want to think how to create a Docker file, all the dampness to tricky bash command. I want just to say, I want this tool into this container. That is. And so this is more or less the schema. How does it work with? You can see here, OK, this is a reputation Nextflow pipeline working into a compute node. And this launch job, to run the job, needs to some container. And basically, what it's doing, asking to Wave, I need a container for this tool, for this platform, and the user that is making this request. Then the container and Wave collect all this information, interact with the Wave to authenticate the user, and maybe also to authenticate the private repository that you may need in your pipeline to put this container. So removing all this problem, managing the credential, and then can decide to augment a container on demand or create a new container using conda packages or Spark packages. This send back the target container image name, and then Nextflow use this name to pull the container. Here, maybe the most important thing is that Wave is not a registry. It's not hosting images. It's not copying your container, but it's just a mediator, a broker that collect the information and decide which container should be used in your pipeline. Then the pipeline pull the container from your registry, essentially. And there's quite a lot of new things when we from since when we introduced this technology the past year, what we call freeze container, the support for singularity, security scan to check all the vulnerability in your container, a new command line tool for Wave, but I won't focus on the last two, the support for multiple architecture. Again, also this morning, Brendan from Amazon was mentioning the need to have the ability to support new architecture, in particular ARM, because they provide a more cost-efficient way for computing. Problem what is that to run data pipelines on ARM, we need containers that are compiled and built for this architecture. And so this is a huge work, because imagine all biocontainer, all conda tools need to be recompiled from one day to another. It's a huge task. Instead, what we are promoting here, a different idea. Instead to compile all these from one day to another, we can build on demand. If you need one tool with Wave, you can say, I want this tool for this CPU architecture, and Wave build this bioconda recipe for you on demand. And okay, targeting different architecture, of course, this requires having bioconda recipe or conda recipe that are compatible with ARM, that compile on this CPU architecture. Not all of them are ready, but this is also why we are, I jump into another slide. We create a Seqera channel on Conda that provide Conda recipe dedicated for NF core pipeline that compile on ARM. And these are available through Wave. Second point, also to bring the support for package manager to Wave and Nextflow then. So why Spark? Also, again, this morning, the Amazon Brendan, was talking about this. Because Spark is somewhat similar to Conda, allows you to create a compiled packages specifying some package name, but it has a capability different. It is able to compile the tool for a very specific CPU architecture. So not just say for Intel or for ARM, but very specific CPU set. This is make it possible to optimize the tool for a very specific CPU micro architecture. So if you are wondering how much you can gain, there is this interesting research paper which they show that if you compile the tool and the container for a very specific architecture, you can gain up to 2 times the performance for that tool compared to a generic platform. So now this is also integrated into Wave, so you can use Spark to create container on demand into this Wave. And finally, the demo, the most exciting part. Okay, this is going to be a bit boring compared to Evan's demo because he was having a nice interface. This is completely be on the command line, but for some of you, it's going to be still fun. And what I wanted to mention also that now we have a command line tool for Wave that you can find on GitHub, bigger. Here you can see this makes it possible to use this service not just from Nextflow, but also like any other command line into your terminal. You can find the releases page. There are pre-built binary for Linux, Mac, Intel, Mac, Silicon, and Windows. So once I install this, I will use virtual machine for this. Let's clear. Okay. Yeah, Wave to this computer. And the most basic operation that I can do, let's see the in Wave demo. Oh, not super comfortable. What I can do with Wave, the most basic operation to access a container with Wave. To do this, I have the -i, the stands for image, that allows me to specify container name, like, for example, Ubuntu, and this gives me the container name that make it accessible through Wave. So I can use this with Docker like any other container, Docker run this guy, or just put this case. Oops. This is just pulling the container, so nothing magic is happening. Obviously, I can pull also with just from Docker instead of using Wave. But why should do this? And not super. Why should do this? But because if this were a private container, I could have been able to pull through the credentials stored into Seqera platform, because Wave is federated with Seqera platform, is able to use your credential in your account to access your container. Also, an interesting thing that you can see that independently, if I access this container from Wave or from Docker, I have exactly the same checksum. So this is to show that if you don't say to modify the container does not change at all. It's just a different name to access exactly the same container image. But what if I want to really change this container, add maybe some script that I need to be used in the container because I want to run a task with this. Let's say that I create a directory, which I have my scripts, and then I want to create a script, my script, hello.sh, and then I just say hello echo, look, I'm talking at Summit. Okay, then what I do? I should build the container and then upload to a registry and then pull from data pipeline. Instead, what I can do with Wave is -i, Ubuntu, and then I want to specify that I want to use this directory as a new layer into my container. I can do this with -- layer and then my script. That's it. And this gives me the container name, and now I can take it, Docker run. This guy is that one, yeah. And then sh-c/hello. Okay, this is doing exactly that thing. Oh, it didn't give execute permission, perfect. My script, but this is a good example to show how is fast the iteration with this. Yeah, I have a new container, Docker run shbc hello sh. And again, syntax error. Again, you want it running. Finally. Okay, again. Docker run. sh-c/hello. Finally. Okay. Part the mistake, but this is also now getting apart, show how it's simple to iterate, because we are not building the container. So when I run Wave -i layer, it's not building the container. The new script or all the content in this directory is added during the pull. So this is the big difference. So why is better? Because imagine that you have 10 containers that need the same script. You need to build 10 containers just to add the one file or two files. These files are added every time this container is pulled by the pipeline. But maybe sometimes you really need a new container, and you can do this with the usual Docker file. So here, Ubuntu, and I want to add hello.sh in user local bin hello.sh. And then I want to execute hello.sh as default. Okay, now to build this, I can use another option that is wave -f, Docker file, and then specify the context, the building context that is always this directory. Now we are really building the container. I have, again, a new name for the container, Docker run. This, you see, and was super fast. It's exactly the same because it was too fast. Yeah. So if I jump into my email, I have also an email that confirms the fact that I just built a container, and there is the ID, the user, where the container is stored, the format, the platform, the time, what I use, the log, and also the security scan I was mentioning you before. I can jump here, I can see all the security vulnerability in my container that I should try to fix, etc. So this gives you an idea that I can modify on demand existing images, but also I can build on demand new images with the usual Docker file. But we say that it's not cool anymore using Docker file. We want to just use Conda packages. Conda is a fantastic package manager, 1,000 of tools that are available. I would like to just create a container saying, I want to use the Conda package that is bad tools 230. And this is creating the container with bad tools. Docker run this and then what? Bad tools version. I made a mistake, repository. Yeah. Here. Okay, so that is. But somebody could say, okay, but why should do this when there is Bioconda, no? There is already a collection of all Conda Docker images for each Bioconda tool, because they made automatically. Well, the answer is that first is only for Bioconda. There is one channel in all the Conda packaging system. So with this system, I can take any Conda package and create a container on the fly. Second, with Bioconda, a biocontainer, there is one-to-one mapping. So one tool, one container. In this case, I can create a container with any combination of tools that are available in Conda. So I can do Conda package that is bad tools, but also some tools, for example. Version, I don't know, 117. And again, I have a new container, and I can run like before, Docker run. Some tools. So this is much more flexible because you can create container with any combination tool that you may need to use in the task. You are not limited to the existing container. And again, so we back to the point of heavy multi-platform container. I could create a container. Now we are creating container for Intel, because it's the default, because I'm running into an Intel computer, but I could specify that I want a container that is for ARM64. And this is enough to trigger the build for this container using Conda, a Conda package, and ARM architecture. I could try to run, but if this is not working, it's a good thing because I cannot run ARM container on Intel, this computer. And exactly, saying exactly like that. The requested image platform is Linux ARM64, but we are running on Intel, so they show that that container is for ARM. So there's a great flexibility into this tool. You can combine and create container on demand, Dockerfile, Conda packages. Also, we added capability to do the same with Spark and Spark packages. And one more thing is that you see that every time they run this, I have basically two numbers, that is the checksum of the build, so what represents the identity of the container, and this number instead, that is the identity of the request. Every time I run this, I have a different number, that is because of different requests, but the container is exactly the same. So some users said, okay, this is cool, but I don't want to change every time the name of the container. I want a predictable name for this container so that I don't need to modify my pipeline configuration every time. Actually, you don't need to modify the pipeline configuration, not even with this system, because Nextflow is able to use this transparently. But the point is that how we can have stable name for this container created by Wave. It is what we are doing with the freeze option, the freeze container. So essentially, you are able to say, create the container and push to my own repository. So I can say build repo, and you can specify your own repository, docker.io. I don't know, in my case, is PD Tommaso Wave demo. Oops, there is W more. And again, here it is. So basically now you have the container that is your own repository. And you can repeat this as many times as you want. It's built only the very first time, the second and third time, of course, is cached. It is always the same container. So this makes it possible to have a predictable name for your container you can take, and then also maybe create a profile in your pipeline, always use this container name. One more thing is that we say that we are able to build container with Conda, multiple platform, but there are also different container runtime. Docker is more popular, Podman is compatible, but there is Singularity. Singularity is very important for HPC world. Singularity is a completely different organization. Does it work the image file? How does it work the specification for the build of the container? So maybe not everybody knows how to use, but the beauty of this that I can just say Singularity and create a native Singularity image file, push to a Docker registry, like if we're Docker container, but we are just using a registry to store the native image file, and then having Singularity and then use it on demand. So the only difference that you see, that basically the name of the container that is returning as this prefix is Horace, the Singularity knows how to know. Maybe I should clean up a bit the screen because it's Singularity, exec, Horace docker.io, and then this is bad tools. Here we go. So, okay, I already tried this, so it's usually a cache local image. But again, here the important fact that we are not creating a Docker container and converting locally to Singularity, but it's done transparently by the building system behind the scene. And okay, we show how flexible it is to this technology. We have a command line tool, but the main point to use this with Nextflow pipeline. This is perfectly integrated into Nextflow. And we also add a new command into Nextflow to make this process a bit more transparent and to see what container are used by your pipeline. Let's say that I want to see the container used into NF core RNA-seq NF that is not the NF core RNA-seq. That's a much smaller pipeline that we use for demoing. And in this case, the spec command tells me the container which this pipeline is using. This always use the same container for all the tasks because it defines the main configuration file. You see there are 4 tasks always using key.io RNA-seq container. But what if I want to use instead the Conda tool that each task, each module define for this into this pipeline? I could use Nextflow config, and I can say Wave enabled, true, and then Wave strategy, Conda. So this is enough to tell Wave to use the Conda directive when building the container. And then I can repeat Nextflow inspect RNA-seq NF, and now the container images become become always the same. There's a typo on the config name. It's a bit different on typing on the podium. Okay. Take a bit more time. And now we have a separate container for each different task. Okay, Salmon, first 2, and then multi-QC task, fast QC. And then also I could enable the the freeze functionality. We freeze true. We build repo c3 docker.io with demo. And again, this is basically does the same thing, but use the freeze functionality to have always the same container name. We have also that command line option that is format that allows you to create this output into Nextflow configuration file format so that you can take a copy as a profile and always use this container name. And okay, I think this shows also the integration with Nextflow. I could just use Nextflow run now to run the pipeline with this configuration. And what else? Jumping back here, as of today, we have this service that is up and running available through web.seqera.io. In the last year, we served nearly 100 million containers, so it's quite stable, working pretty well. Also, as of today, we are announcing the availability of this tool, an open source project that you can download from this GitHub repository and even contribute if you want. And we are also to make it available as an enterprise distribution for customers starting from next year. So plenty different way to use Wave. There is no more excuse to not use it. Let's jump into the last part. Of course, we didn't talk much about Nextflow so far. That is the pillar technology into this ecosystem. The last month has been we work hard to smooth and make much more interoperable this technology with Wave Fusion. So not surprisingly, now we have also in Nextflow the capability to use Spark packages into Nextflow. There is a new directive that is Spark that is very similar to what is doing Conda. So essentially, you can define the dependencies using Spark, and then you can run locally, and the build happens locally, or you can build this with Wave, like I was mentioning you before. Along the same manner, there is support for the architecture directive, so you can define what is the target architecture for the task, your pipeline. And not just the architecture, but also the microarchitecture. For that discussion we were saying before, the capability to compile for a very specific CPU set your pipeline task. And again, we take care to create the container on demand for this. Fusion is now first-class file system into your Nextflow pipeline, supported by almost all Nextflow executor, the most important one, Cloud Executor, AWS Batch, Azure Batch, Google Batch, Kubernetes in the different distribution, even local executor you could run using remote file system. This can be used for maybe running a pipeline into a EC2 instance and scaling vertically. Also, we have the support for this to SLAR, LSF, Grid Engine, HPC batch scheduler. This is quite interesting. So this brings to this possibility to use Fusion also on-prem. And this is a reference also in the title of the presentation, converged computing. So this idea that cloud is becoming more efficient and becoming more HPC, but at the same time also HPC needs to become more similar to the cloud. It is a good convergence, it's a good intersection using the object storage, maybe on-prem S3-like object storage, as a temporary storage for your pipeline execution, like we can do into the cloud using Nextflow and Fusion. We also have the capability to tag scratch files when using Fusion, so that all .command star files have this tag metadata, and then all temporary data is tagged with temporary. This makes it possible to have some automatic cleanup rule to remove all this data automatically from your object storage. Cloud Cache plugin, I think we are running out of time. I can skip. Few words roadmap for next year. We are working quite a lot, interesting stuff. We don't have enough time to see one by one, but the main idea, the general vision, is to make Nextflow much more static type-oriented programming language, or DSL, that is the most correct definition. Because now Nextflow is quite powerful. but it's completely a dynamic programming environment. And this can be a bit complicated, maybe to predict error, and then maybe it happens during the pipeline execution. Instead, we want to make this experience much more solid using some static type compilation to have a precise error reporting, and also to have much better integration with ID, so that when you're writing the pipeline code, there is the auto suggestion can tell, okay, you can do this, you cannot do that. Add the support for custom data type, and also to have a well-defined workflow definition for the input and outputs of your pipeline, so that will be possible to compose also different workflow in a semi-automatic manner. So I hope that give you a good overview of what is happening into the Nextflow world. And above all, yes, the overall idea is to have a unified ecosystem, and above all, a unique mix of technology to deploy pipeline at scale in a portable manner. So always with the original vision to allow scientists to focus on their work, and we remove all the complexity of this pipeline. Just a few seconds to say a big thank you to all the people involved into this technology, Seqera Compute team, CyDat team, Infra team. Big thank you, guys. You are fantastic. It is an honor to work with you. Okay. Done.
Excellent. There are a lot of questions in Slack. We might do just one, even though we're over time. Would a Fusion CLI be available in the future?
Fusion CLI. But Fusion is a CLI. But the point is that Fusion, to work properly, need to run into the container. So I think the question is more if you could manage independently Fusion. Maybe it can happen at some point, but the point that we really care to smooth all the integration. So the best way to use Fusion is somehow automatic manner using Wave. But in some specific cases, it's also possible to use in a manual manner.
Excellent. Well, give it up again for Paolo. And up next, we have Simon coming on to talk about scalable pan-genome graph construction. Please welcome Simon to the stage.
Hello everyone, I'm Simon. I'm a PhD student from the Quantitative Biology Center in Tübingen. And I'm going to present you cluster scalable pan-genome graph construction with NF core pan-genome. Okay. So Eric Garrison yesterday already told you something about pan-genomes. This will be just a quick repetition to quickly get you up to date. So what we usually have is a batch of individuals, and then we compare these against one single reference genome, and then we can observe certain kinds of variation, like insertions, deletions, or other single letter changes. However, what we miss is the variation between all of these individuals, and so we are driven by a reference bias. And one solution here is that we can actually take all of these individuals, align them versus each other, and then present them in a graphical model. So that this graphic model then contains actually all the variation as before, and all the sequences that these individuals share are then compressed in several nodes. Now, when you want to explore the pan-genome, we also need cool visualizations, and I'm happy to tell you that one way to visualize Pan-genome actually is also in a tube map, like we do in NF-core. So you're already familiar with this. Now, how can we build such Pan-genome graphs? What you see here basically is the workflow of the Pan-genome graph builder that Eric showed you yesterday. So first we have an all versus all alignment step, WF mesh. Then Seqwish actually takes these alignments and introduces these into a variation graph. And then we smooth the graph and remove some redundant nodes. And basically, this is the core workflow. And on top of this, what we can also do is we can call variance to any kind of individual in such graph. And also, what we do as like to get an idea of the quality of the built Pan-genome graph is that we can actually translate this graph into the so-called OGI format. And OGI is also a tool, a variation graph tool. And then we can generate the statistics of this graph, or we can actually produce certain 1D visualizations and 2D visualizations, which you already saw yesterday. And all of these things are then collected in one multi-QC report. Yeah, and this is basically what's happening in this Pan-genome graph builder pipeline. However, it is written in Bash, and so I took the opportunity to actually put this into NF-core using Nextflow, and that's now the core workflow of the NF-core Pan-genome graph pipeline. Yeah. Of course, now when I port this to Nextflow, we want to scale better, because now we can make use of nodes of the cluster. And so what we can do here is that we can apply WF mesh and just do the all versus all approximate mappings. Then we can take these mappings and split them into chunks, let's say, for example, 100. And then for each of these 100 chunks, we can execute the base pair level alignment, for example, on 100 nodes in parallel. And so this helps us to save a huge amount of time. What is also possible is that thinking about human, for example, we have several chromosomes, and usually the idea is when we did the mappings, then we want to detect the communities like Eric did yesterday with the acrocentrics. And so the idea is that later we will have roughly one graphical component for each of the communities. So one chromosome corresponds to one graph component. And this is what's happening here. So we can detect the communities. And then for each community, again, this core pipeline steps are executed in parallel on nodes on the cluster. Once we have all the communities, we can combine them again into one final graph, and then we can do, again, the statistics and visualization steps again. So here I have one example of when I build a Pan-genome graph of Dodoromyces elongispores. It's a yeast fungi, and also it's an underestimated pathogen. So old people that have a weak immune system actually can die within days if they got infected with this yeast fungi. So it can be pretty severe. And that's why it's interesting to study. It has roughly the same gene length as yeast, and it comes with 8 chromosomes and some mtDNA. And doing a Pan-genome workshop this year, several colleagues actually came up with 11 assemblies from nanopore and Illumina data. And 2 of them were fully assembled, and the other ones were not fully assembled, so they still consist of contigs, and some of these contigs are actually really small, like only thousands of base pairs. And as we will see later, this can be then quite a challenge to build the Pan-genome graph from. So first I executed the workflow as seen before in this community mode. Now the cool thing is that actually we get these 9 communities, what we were kind of expecting, right? So we have 8 chromosomes plus the mtDNA. And what you can see here are actually 2D visualizations of the Pan-genome graph. So most of these chromosomes are actually linear. There's not so much large structural variation going on. However, these thin tails that you can see here or here, for example, this is unmapped sequence. So that's unique to maybe 1 or 2 strains in this graph. So that's not really good. And also, chromosome H and chromosome B look kind of messy. So what we did then is that we took another approach to this community detection. And what we did there is that we took one a reference strain, and then we mapped all the contigs to this reference strain, because from the reference strain we already know into which sequence corresponds to which chromosome. And that's what we did here, and now for each of these communities, again, we run the NF core Pan-genome pipeline and obtained these graphs that you see here. Now we have much more linear chromosomes. Even chromosome B looks beautiful now, but chromosome H is still a mess. And so we went back to the people who generated the assemblies, and we actually took a closer look at the 2 fully assembled strains. And so what we then came up with is that we actually need to put the sequences and contigs of basically of these communities that we had before of chromosome C, chromosome H, and chromosome G into one graphical component, and then now we see this beautiful Pan-genome graph. So we have very linear chromosome A and chromosome B. Then we have this large graphical component of chromosome C, H, and G. And then we have the other chromosomes, chromosome D, E, F, and the mitochondrial DNA. And this chromosome community detection algorithm in the beginning here maybe didn't work as well as we expected, because the contigs are of such a large, different size from several million base pairs to just 1,000 base pairs, and so the cluster algorithm might have had difficulties there. Now, how does the pipeline scale? Next, I built a Pan-genome graph of 1,000 chromosome 19 genomes, and chromosome 19 has a length of around 59 megabase pairs, and here you can see some timings. So double F-mesh map took 9 hours, then I split the output into 100 chunks, and then each of these chunks actually took 2 hours. The sequence step took 1 day and 13 hours, and SmoothXG finished within 15 hours. So that was actually surprisingly doable. What you see here is basically an excerpt of the final multi-QC report. On top, you can see the graph statistics. Then there's a 2D visualization here, followed by a 1D visualization, which I will explain in a moment. And so what was really puzzling in the beginning when you take a look at the statistics is that we have actually 3 billion Ns in this graph, but chromosome 19 is only 59 million base pairs, and that was really confusing. And so I was calculating, I divided 3 billion by these 1,000 genomes, and then we end up at around 3 million, right? And the 1,000 genomes project is not the newest data, and so if you take a step back to HG19, it also has around 3 million Ns in it, in chromosome 19, and so the numbers actually fit. And that's because during the Pan-genome graph construction steps, we don't align the ends, and so they just get added up, up, and up, and so that's why we have so many ends in there. And that's what you can also observe in this 1D visualization, basically. So here, we arrange the nodes actually in 1D from left to right, and then we color the nodes by the path coverage. So if a huge amount of paths actually support that node, so that certain sequence, then we color it in blue, and if only there is a low number of paths that support that coverage, we color it in red. And so because we accumulate in 1D all these nodes with Ns, then of course we mostly see this red color here. And the interesting parts where actually some alignments happened are actually only here really tiny and here really tiny. Yeah. And so basically maybe it's a better idea to remove the ends before you build such a Pan-genome graph. Next, I also built a Pan-genome graph from around 2,000 E. coli sequences. And here, the quadratic all versus all line problem actually really becomes a huge problem, even with these parallelized steps. So WF-mesh map was relatively quickly. However, WF-mesh align took 1,000 times 20 minutes, and that's really quite a lot. And in the end, I got PUF files of around over 600 gigabytes on disk. And then Seqera was really unhappy because it only had 2 terabytes of scratch space, and this was not sufficient to even start the index building. So I thought, okay, maybe I can revert to our large network storage, but it was so slow, it didn't complete it in a reasonable time at all. So fmesh actually comes with a parameter where you actually can set the percentage of mappings that should be retained for more further downstream analyses. And so I used this, and so this allowed me actually then to get much, like the alignment problem, fmesh alignment problem actually became much smaller, and so this time it only required 100 times 5 minutes to do the base pair level alignments instead of 1,000 times 20 minutes. Still, Seqwish was unhappy because it didn't have enough RAM, and so there's a parameter which allows you to just lower the number of bases which are processed in one batch. It takes longer, but still you can run it, and so after that, it only took 5 hours for the Seqwish step, and for the final smoothXG step, which normalizes the graph, this took 62 hours, and I was only able to do 1 round. Usually we do 3 rounds because it was just so much data. Here you can see the results. On top, again, the statistics of the graph, and the 1D visualization now on top with the 2D visualization on the bottom. So in this 1D visualization, I didn't explain yet, I just realized, these black things here, they actually are the edges of the graph. So they represent the topology, but in 1D, it's actually super hard to understand what's going on. That's why we usually also have these 2D visualizations. So here we see that the huge part of the Pan-genome is actually covered by most of these sequences. However, some part of the Pan-genome is unique to some of these E. coli's. And we know that E. coli have lots of bacteria sex, and then genes get added, genes get deleted or rearranged. And that's why this Pan-genome graph actually is so super complex here. And that's also why here in this 2D visualization, we have this large blob, because basically, if you have so many sequences, every sequence somehow interacts with another sequence, but at different places, and that's why it looks so complex, and that's why Phil yesterday, I think, was so scared of this huge blob, because it's crazy, but that's just reflecting biology. What you see here, oh, sorry, there's the mouse. What you can see here on the left-hand side are some extra graph components, and apparently they were not similar enough to be put into this huge graph. Yeah, that was really quick, I think. That's already everything I wanted to tell you, and I'm happy to take questions.
Excellent. Thank you very much, Simon. There are 2 questions in the Slack, but we're actually running a little short on time, if you don't mind answering those in Slack. And coming up next to the stage, we have Raquel.
Hello, everyone. My name is Raquel Manzano. I am Spanish, but I come from UK. I am a part-time bioinformatician and PhD student at Cancer Research UK, the University of Cambridge. And today I'm here to talk about my story or my adventure with Nextflow and the development of this pipeline. I want to give a bit of background because I work in cancer research, so I just want to make sure that you understand why I'm actually doing this. So in cancer, mutations are quite important. You can do a lot of type of analysis, and they can be more or less sophisticated. So it's very important to identify drivers, so that can give us an idea of maybe what kind of drug treatment can be good for the patient. or what sort of things we can use, and then the mutational signature analysis that tells you the biological processes that might be driving the tumor growth. And then you can be very sophisticated and go for a subclonal deconvolution, and you can do this even with bulk sequencing data. And that means that you can actually go into your sample of the tumor and try to deconvolute what kind of different cancer cell populations you have. And just another thing that I want to say, so it's clear, in cancer we talk about germline and somatic mutations. Germlines are the mutations that you are born with, and then the somatic ones are the ones that you acquire later, and these are the ones that you want to study in cancer. So how do you do this? So it's fairly straightforward now to do this kind of analysis. We have JTK, the Genome Analysis Toolkit, which is great. It provides best practices to do so. And of course, we have Sarek, that you should use because if you're here for variant calling. And actually, a great part of my pipeline is based on that. But for RNA, because I wanted to do more, right? Like everyone is doing variant calling in DNA. Now I want to take it a step forward. That is what you do when you're doing a PhD, try to do something else. And I said, okay, I want to call mutations in RNA sequencing because we have a lot of data. We have very big data sets that have DNA and RNA. And I thought, well, it would be cool if we can combine that information and we can know and we can do some sort of integrated analysis, even doing the subclonal deconvolution that I was talking before, and see if you can know a bit more about that intratumoral heterogeneity, if those mutations are being expressed or not, how does that work out. But RNA is more challenging. And it's challenging for technical reasons and for biological reasons. So we have an equally distributed coverage. Of course, we have upregulation than regulation. We have higher levels of duplication. And then we have very cool biological things, that is alternative splicing, polyadenylation, and RNA editing, which happens normally in our cells. So it's something normal that we can find. Well, that means for the purpose of a bioinformatician that you will find a lot of artifacts. If, for example, you do a DNA analysis and you end up with 100 mutations, in RNA you might find yourself with 1,000 mutations looking at the same. So it's like, okay, so now what do I do with these 10x more mutations that I find in RNA? Are they worth it? Are these artifacts? So while I was working on this, then this paper came across. So this paper is from Gadi Getz's group in the Broad Institute. And they developed this RNA mutex pipeline that was very simple, but quite straightforward, where you can do your variant calling with RNA using the match normal. They were using mutex, so mutex 1, not mutex 2. And then they were doing some post-processing for the RNA, where they were doing some filtering. And this filtering included all these steps. They did a realignment. They would check on the known RNA editing events, because there are databases out there. They would annotate them so you can remove them, so you end up with a nice and clean set of mutations. And some other filters. If you are interested in any of these, just let me know, and I can go deep into it offline. So my goal for my PhD was actually to, what I said before, right? I wanted to use DNA and RNA to call somatic mutations. In my case, I work in a breast cancer research group and look at intratumoral heterogeneity patterns. So my roadmap was straightforward. So I would reproduce what RNA mutex did. I'm going to update the pipeline a little bit because I felt that using mutex 1 in this moment was, there is mutex 2 now, and there are tools that do more. So I wanted to update that and tune a bit more the parameters. And another thing was like RNA mutex was a bit scattered. It was like MATLAB script, Bash scripts. So for me as a bioinformatician, it was a bit difficult to make it work. And I felt, well, if I'm going to be using this, I may as well use my time and do something more reproducible. And then use this pipeline to analyze the cancer samples that I wanted to. Easy, that is what I thought. Done, I will do it. So I did develop a Python pipeline with this, and I did nothing as close as Nextflow, but I did some, it was working very well with my HPC system, and I could resume as well and do some stuff, but again, nothing as fancy as Nextflow. I didn't know yet what Nextflow was, and then I updated the workflow, I did the analysis, everything was working, and I said, oh, I did this maybe too fast. So then, right, I arrived to the stage that now I have to do the analysis of all these samples, and yeah, I need to make it scalable and reproducible, right? And I was thinking, yeah, I have my system. I know it works in my setting, but then, you know, I felt this itch where, but if I want to publish this, are people going to be able to use what I have done? And yeah, I thought, probably not. So I started trying to search what I can do about that. And that is when, okay, I started, of course, Googling scalable and reproducible research, bioinformatics, blah, blah, blah. And that is when I went down the rabbit hole, and I found Nextflow. I think the first hit was the RNA bar pipeline that already follows JTK best practices, which you say, oh, great, I can do this, but I actually want to use mutex and other variant colors. And that is where I found Sarek, and then I read a bit about Nextflow, and then I clicked on the Slack join, and then, yeah, that was it. I think the first, I wrote in the new pipeline, like, I want to do this, would that be okay? And I think it was Maxim, the first one that answered, and he said, of course, yes, we can do this, it's gonna be fine. I said, awesome. So 1 year after, because I thought, oh, I can do this fairly fast, but yeah, 1 year after, I have my TÜV plot as well, my TÜV graph for my pipeline, and now I have developed this DNA and RNA somatic variant calling. It's called DNA and RNA bar. It is in GitHub now. It is still in development, but I'm really hoping for a first release next month. That is what I've been saying for several months, but I trust this is going to be the good one. And part of the pipeline, again, I said, is a lot of Sarek, but I didn't need, because Sarek is very fancy and has a lot of features that I actually, at the moment, I didn't know what to do with them. So I just said, okay, I want just the variant calling superflow, and then for RNA bar, I take the best practices, and I did my little Frankenstein of all these fantastic NF core pipelines. And I did add quite a few things, because I said, well, now that I'm doing this, and it's going to be scalable and reproducible, I want to do something end-to-end. I want to be that person that say, just press that button, and then you get the results. I can go on holiday, and everything is there. So I'm going to talk a little bit about the new sub-workflows. I have restructured things a little bit. I welcome a lot any feedback, criticism, because I've been trying to do things a bit differently, because as a good bioinformatician, I can be opinionated, and I like certain structures, and I have done some extra sub-workflows of the sub-workflows, because I felt the first time I looked at Sarek, I felt, wow, this is a lot of code just in the main one, and I felt a bit lost, and I have done some restructuring of that in my pipeline. And I have included things that I think that are interesting. So for example, when you finish with Sarek, you finish with a bank of VCF files. So I have included a new sub-workflow that is for the normalization of the VCFs. If you are familiar with variant calling, you might find yourself that when you are trying to put everything together for SNVs, so for single nucleotide mutations, it's fairly easy to just compare them. But if you run into indels, it might be a bit more complicated, because they can be annotated differently between the colors. So I have a small sub-workflow to normalize all that, so everything should be in the same coordinate or left aligned, et cetera, et cetera. And then I have also implemented a consensus, so you end up not with a bunch of files that you don't know what to do, but with an actual final file that is in a MAF format. MAF format is specifically for cancer. It was designed by ICGC, the International Cancer Genome Consortium, and it's a mutational annotation format. So it's a table file, but it is standardized, and you can add there all the columns that you want. And then it is very easy to play around with it, because it's a TXT file. Then I have also included some map filtering sub-workflow, and I have tried to do this as flexible as possible. In my opinion, I have put the filters that I think everyone should be doing in cancer, like NOMAD, maybe some VAFs. You can tweak that if you want, and again, I very much welcome any input on that. If you think that something should be added, we can talk about it, and I can do it. And then I have also done this realignment step that RNA Mutec used to do. So this is more related to what I was saying before about the restructuring and doing things in a way that can be as reusable as possible, because since I was putting a lot of effort into this, I actually wanted to make it good that you can just take this and take what you like and run it. And everything is similar to SARX. You can start from FASTQ files, BAMs, CRAMs, VCFs, and MAP files if you want. So you can just do the filtering if you have your MAP file and check how it looks like. And just in case you're wondering about the X that is there in the middle, it's just because if you have DNA and RNA and you want to leverage that information, in the consensus step, you can use the DNA and RNA. Well, the way that consensus works in variant calling, sorry I didn't mention that, is that I have my 3 callers, and it's a very simple voting system. So if 2 of the callers are calling that mutation, they say, okay, this is a good mutation. And in the case, if you want to do what I call a rescue step, if you also have the DNA or the RNA, that is sort of orthogonal validation. So I add also that, so it gives an extra vote there. And if it is called in DNA and RNA, it should pass. So what's next? So I did say at the beginning, first release coming very soon with a preprint that is also coming very soon, working very hard on that. At the moment, what I've been working, I was in the hackathon, which was fantastic. Apart from doing my first NF test and doing a PR to SAREC that was successful, I also had time to work on my test profile and things. And it was great because at the end of the hackathon, I did get the message that the pipeline completed successfully with the test profile. So, wow, that was nice. So all this pending of review and pushes, but it will get there. That's what I'm saying. It's coming soon. And now after this, I will finally do what I should have done a year ago, testing the scalability and run my analysis on breast cancer data sets. And then of course, I am a multitasking person. I like to do things on the side all the time. So I did like a lot NFTs. I think it's great. So I would like to implement this in my pipeline as well, maybe for a second release. And also the sub-workflow recycling. So I want to, I've been talking with Maxime and some other people about maybe you can put this in SAREC as well. Or for whoever thinks it's interesting, again, come and talk to me. And then I am also very eager to spread the Nextflow in Cambridge. So I'm part of the bioinformatics training unit. And I'm trying to prepare it. You don't always have time to do all the things that you want, but I'm really hoping that I can get this done fairly soon. And finally, just a very big thank you. This is my lab there in Cambridge, to my supervisors, Carlos and Oscar, and to Gadi Gates, he also gives the input. And then, of course, thanks a lot to Seqera Lab. I have Maxime. Maxime has been great. He is my official supervisor because I've been part of, well, supervisor mentor. I've been part of the mentorship program at the Nextflow, NF Core, and that was fantastic. If anyone is thinking about doing it, do it. It's really great. And I also want to say a big thank you to the Open Bioinformatics Foundation because they found me to come here, and it was great. And the biggest thank you to the Slack community because you guys are great. You won my heart, so thank you very much.
Awesome. Thanks again, Raquel. We caught up a little bit on time, so I'm going to choose one question here from Adam. Can you compare multiple RNA samples versus one DNA sample? And are there any plans to add detecting fusions?
That's a great question, Adam. And the answer is I don't know. I would have to test that, but I think that's actually quite interesting. Yeah, we can talk about that. And there was a second question?
And are there any plans
fusions.
to add detecting fusions? Yeah.
Fusions. Yeah, not at the moment, because we need a first release first, but I think, yeah, that would be great, and that would be a great collaboration with the RNA-seq pipeline as well, because I think that, yeah, RNA-seq with fusions, it would be great to have it as well.
Awesome. Well, thanks again to Raquel. And up next, we have Maxime coming to the stage. Give it up for Maxime.
Can you hear me well? Perfect. Hello. It will be hard to follow like such an interesting talk, so I'm not talking about Sarek today, so that's like I think one of the first time I'm doing that, so bear with me. And yes, so I'm going today to talk about a tiny side project that I had, and like it's been an idea that was like running into my mind like for years, and I finally have been able to do that. So I will briefly like start with like a short history, how I created like this AWS open resource, how to use it, how to add data, how to updating, and what are the future plans. So a brief history. For me, and I think that's why I use like Nextflow, and that's why I'm like in this community, like reproducibility is central. And like we deal with the software, like with the container, like I'm pretty sure like with Wave we will do that like even better. We deal with the parameters in the way that we build like our Nextflow pipeline, and we can deal with that like fairly good. And the main, for me, the next issue is that what we do with the reference file. So for most of our reference file, like at the moment we use like igenomes, which work like fairly well, but then what do we do with the annotation? How do we deal with that? And that has been like one of the main issues that I've been like struggling like in building Sarek like for the last like 8 years. So that's my only slide like about Sarek, and I will keep it short. So many years ago, like all of the annotation like was done using like the local cache that was available on my institutional cluster, and that was the only way you could use like annotation. As we started like expanding and like having like other people using Sarek outside of like Sweden, that became like quickly a mess. Also, locally, it was a mess to try to update the cache. I think I still have one email like requesting like an update on the cache, and they replied to me like 1 year later, and I think they were still not updating that. So now they've become like way better, but yes, at that time it was not working fairly well. So I had one of my like worst ideas ever, and I said, why don't I build like containers with pre-downloaded cache inside? So people don't do that, seriously, that's a bad idea. I've been blamed by several people, but yes, it was working, it was reproducible, and I was happy with it. So at some point, I gained like access to a new cluster. I had like admin access, which is another bad idea. You should never like give me admin access to anything. And I made like full usage of like the local cache like for running Sarek. It was like working well, and I was like happy again with that. I was still maintaining like the containers with the pre-downloaded cache, and that was not optimal at all, because I managed like to to build like automatically like some of the container, but sometimes it was not working, so I was building all of that on my computer. I had like plenty of script to help me like automatize that, but I was still doing that on my computer. So when I was doing that at home, it was taking like the whole night because like bad internet connection, so I was always going to the lab to do that, and that was a mess. Recently, we added back into Sarek like the option like to download the cache with Sarek, which was like great, and also to directly, as soon as you get the cache, you run Sarek, and then you annotate with the cache already. So that's amazing, that works well, and I figured, but why don't we like push that like on the cloud? And that way, we have like we could have like a resource and like be able to use that for everyone that is running Sarek on the cloud. Because for me, if you're running on an HPC, you should download the cache like locally, and that's good. But if you're running on the cloud, then you need to have like a resource that is devoted to that. And I was like greatly inspired by AWS iGenomes. So I talked with some people at AWS, and that's how I created annotation cache. This is like a non-meme like in the NF Core community. Every time I try to explain like how to deal with the annotation cache, like how to deal with the different like DB cache version, species and stuff, like no one understands what I'm explaining, so I'm hoping it will be better today. So how to create like an AWS like open resource? It's actually fairly simple. You can skip like most of the steps. Actually, like you just need like the 5 first steps. I decided like to do the rest like with Nextflow, but I will come to that later. So first, you need like to have an idea. My idea here in that case like, was like to create like a cache that was like a resource that people can use for annotation. Second step, you need to grab like other people to help you build that, because if you're alone, then it's a bad idea. You're building like an open resource, you should be with other person. So grab some friends to discuss about it, and like make them like work with you even if they don't have time or even if they don't want to do it. Second, make a request to AWS open resource like to create this stuff. And for me, that was like the stuff that took the most time. It took me like 2 weeks just to write like a 5 line and ask the stuff. So yes, bad planning, but yes, it's fine. Second, like once you got like agreed to that, you get like all of the credentials that you need like to create an AWS organization. That was like super fast. And then you just need to create the infrastructure that you need to host your resource. So for me, I just needed like one S3 bucket, which is this one, like annotation cache. And then the rest, you can do it like by yourself on your computer, but I want to be reproducible, I want to be like in a community, and I want other people to be able to do what I'm doing, because I'm thinking, yes, at the moment I'm doing that, but what happens tomorrow? Maybe someone else can take over and like do that for me. And I will be super happy if someone can do that, but yes, it's another question. So we already have like some scripts in Nextflow, written in Nextflow, which are like in NF Core module that we can use to download the cache. So that's simple. I'm fairly lucky like to work at Seqera, so I know that we have like this wonderful platform called the Seqera platform that we can use to actually like create a compute environment like for me on AWS. I did that like once by myself. I think that was still while I was at NGI, and I can do it by myself, but it was badly configured. It was such a mess, and I don't want to do that ever again. So I'm like so happy that I can do that with just a single click, and that's super nice. Then I just like add all of the scripts that we have like on the platform. I launch the script, and it's already everything published to the S3 bucket, and it's all done, and I can use it. So that's amazing. There was like one tiny problem that I discovered like while I was like creating that. So both of the tools, like SnipF and VIP, they are like amazing tools. They work like super well, but they work super well when you work like on a local HPC with a file system. They work like super well because they take like as an input the root cache folder, and then the whole structure inside, it's a whole architecture of folder and subfolders, is organized by like cache version, genome build, species, and like it's a whole mess, and which is where like the meme is coming from. And the tools itself, like themselves, they resolve the path internally given the input. So I'm just showing it. So for example, with SnipF, I'm using here like as one of the input like of the tool like the path to the root folder of the annotation cache, like for SNPF. And with the argument that I give to SNPF, which is here like the DB argument, it corresponds to this subfolder. So if I'm running on GRCH38 with the 105 like version of the cache, I get this one. If I want like another cache version, it has to be like another DB. If I want another species, it has to be another DB. So SNPF is fairly simple. Then if I look at like VIP, it's an extra layer of complexity. We still have like the same system where we have as an input like the root folder, and then we have like first the species, so C. elegans, Homo sapiens. Then we have like the cache version, so 110. Both of the versions like look similar, but like they are different tools, so they don't relate to each other. And then which is the genome. And that's how you use like the cache. So for me, if I want to use that on the cloud, I cannot point to the root cache folder, because if I do like a S3 copy, it will copy for me like the whole infrastructure inside of my like where I work, and that's bad, and I don't want to do that. I just want like to copy a single infrastructure. So I had like another bad idea, and I decided that I'm going to do what the tool we are using. I will like organize everything the way I want, like in the bucket, and I will let the tool that I launched like to use annotation cache to handle all of that logic for me. So that's what I did. I added like an extra layer of like of architecture, and inside like each folder acts as its own like separate instance. And I deal with that whole logic internally within like within Sarek. So as an example, within Sarek, what I just do here is that I specify like what is my SNPF cache with that, what is my VIP cache with that, and I deal with everything like with the whole like subfolder and architecture within Sarek, and we don't have to take care about that. Of course, it still works like locally with your local cache, and that's perfect. I'm not going to do a demo. I think you had like enough demo for the summit, but yes. Adding data. Adding data is actually like fairly simple. So I have like two scripts, one to download the cache for SNP, one to download SNPF, one to download the cache for VIP. I have one config file where I list like all of the different like parameters that I need like for to build and to download like this reference. I have, of course, like one Seqera platform and one AWS bucket. So I just choose like which genome I want to build. I push one button, and it gets downloaded. If I want to update, it's fairly simple. That's the same slide again, but because that's the same logic, I update the script, which are tied like to a version, and then I can update like whatever I want. And more or less that was all. So for me, what are like my plan for the future? So to deploy that like in other NF core pipeline, I think like the pipeline that we just showed, like that Raquel just showed, is like a good idea for that. I would love to have that also in RNA-var. Maybe I think we can talk with RAM and the rare disease pipeline to see if that could be of interest for them as well. I would love to see like some deployment like outside of NF core as well. Of course, with all that, we will need like better docs, a better website. I haven't shown any pictures of the website because I designed it myself, and it's super bad. I would want also a real dedicated like channel for communication, because at the moment I'm just like hiding like in a Slack channel on the NF core Slack, and I want like to have like a proper channel like for communication. So I will like sort this out. Of course, being like at Seqera, I know that we have developed like within the team this amazing tool like Seqera Kit, and I'm pretty sure I can use that like to have like some smart automation and help me like populate all of these resources. And maybe at some point adding some more annotation tools and like expand like the reach of this resource. of course, I want to thank like everyone that helped me and no, everyone that I dragged like really into this project, especially Rike and Adam. I'm sorry for all of the people that I annoyed and that I'm not mentioning here, but yes, mainly I want to thank like the whole like NF Core community. And if you have any question, I think now is the time.
Excellent. We don't actually have any questions in Slack, but thanks again to Maxime for a wonderful talk. After this, in the main hall, we'll have the poster session 2, as well as coffee. I think we're coming back here in about 45 minutes. So we'll see you all then for a great round of sessions. Thanks, everyone.
Oh baby, I wanted you to be here inside my arms where the night goes down. Oh baby, I wanted you to be here when the sun comes up and the day goes by.
All right, everybody, welcome back. I hope you're well caffeinated. We're going to start up again for the last session today with Chris, right, who's going to talk to us about real-time sequencing analysis with Nextflow. Thank you.
Okay, just to start with the kind of obligatory company slide. So Oxford Nanopore, lots of you will have heard of us. Everyone we're cool, everyone wants to use our tech. The one thing I'm going to talk about today is actually on this kind of corporate slide, which is why wait real-time analysis. I won't bore you with more corporate stuff, go talk to the salespeople. So just a bit about my background. I'm kind of in customer bioinformatics now, but I was at Nanopore for 10 years. My role in the first kind of 8 years of that was on primary and secondary analysis, so base calling, variant calling, consensus calling, a bit on assembly, but not as much as Eric. Go talk to Eric, not me, about assembly. But in the last 3 years, I've kind of formed a new group around customer bioinformatics and really driving the adoption of Nextflow within the company. So my group works to standardize and automate the authorship, testing, packaging, distribution of Nextflow. Some people still use SnakeMake in the company. We don't hold that against them. My group, a lot of what we do is actually take SnakeMake and port it to Nextflow. It's really boring. I just try and stay away from doing that. It's fine to use SnakeMake if someone's already got SnakeMake. This talk's kind of an extension of something that I gave at our own conference in the summer, so if you want, you can go listen and be bored more by me as well. My group makes this desktop application, Epitome. This kind of like a desktop version of Tower. It looks very similar, you'll see as this plays through. The idea is that the company motto, sequencing anything, anywhere by anyone. The anyone and any where part of this is key. We take devices into the field. We don't have a HPC cluster nearby. We want to analyze it on a laptop. So we have a desktop application that takes you through running Nextflow workflows all the way through to, here you see JBrowse being run at the end of an analysis. So as I say, this is supported on Windows, Mac, and Linux. As Evan said yesterday, science is done by scientists. So this is kind of taking what bioinformaticians are good at and giving it to the scientists without needing the bioinformaticians to get involved all of the time. So on to the meat of my talk. Why would you do analysis in real time? It's not really the normal sequencing model of collect a sample, send it off for sequencing, get the results back maybe a few weeks later, or maybe quicker if you're lucky enough to have your own sequencer. I'm going to give one example. So TB is still a big killer, but predominantly in India, Southeast Asia, some parts of Africa. And taking the experiment and the analysis close to those samples is kind of key to be able to action anything. So we want to make tools, give those to people in the field. And actually, Harsha will know this, the UK was very good at doing this during the pandemic with COVID. We're very good at collecting samples, analyzing them, sending them to Edinburgh for them to build phylogenetic trees. And so something very similar can be done with TB. Moving slightly outside of just the specifics of TB and where my talk's going to go now, we want to be able to do metagenomics in real time. So we want to be able to take a sample from the environment, detect what's there, and then provide some kind of insights. This is something that a number of years ago, our marketing people gloriously called what's in my pot. Some people changed the pot for ruder words, I'll say. There's two forms of this. You either do it on a whole sample, or you might do it with amplicons, so targeting RNA genes. And from this, you can say things about infectious disease, not just tuberculosis, but meningitis. You can do flu typing with this. Gen Z like to know what's in their microbiome, so whether they need to eat more kimchi or anything. Also, you could do, it's not just infectious disease, but also environmental studies. So one thing, again, to take an example from the UK, the water boards often get in trouble for releasing effluent into rivers. People want to monitor that and fine them and whatever. Also, in industrial food production, you might have a production line making, I don't know, pasties. You want to know if those pasties have got beef in or if someone's put pork or dog or whatever, right? So that's something you can do as well. For those not familiar with metagenomics, the basic process is you collect your sample, you do some DNA extraction, sequencing. You take those sequenced reads and then you classify them. So you say this thing is from whatever organism. For long reads, we use Kraken 2. It's kind of well benchmarked, it's fast, it's accurate. It's probably the lowest memory implementation of this sort of thing. After you've got those single read classifications, you want to try and estimate the abundance of things in your sample. So that's not a simple thing to do. There's some processing in there. And the important point here is that all of those three things to the right-hand side can be done concurrently or in real time. You don't need to wait for the sequencing to finish to perform read classification. Just to kind of go a bit deeper in this, sequencing is sampling a distribution. There's a certain number of things there in your sample, and you can take some measurements, you can observe what you've seen, and then you could react to that. So this animation is showing as a function of time, like sequencing time, we observe more and more unique species or organisms. And there's two examples here. The top one, we have a kind of flat distribution of organisms present. That's not very realistic. You don't really tend to get that. But what you see is the number of unique species that were observed kind of saturates very quickly, whereas in the bottom case, there's more kind of, some things are present to a high abundance, some things are present to a low abundance, and you kind of see this curve that kind of more slowly accumulates over time, and that's because some things are rare. You need to do more sequencing, more sampling of that distribution to detect those things. Practically, this means that if you can see that this saturation is happening, you can stop your sequencing. You don't need to sequence more and more and more stuff. You can have some confidence that you've seen everything that's present. So how do we do this? Well, we need some form of data streaming. We don't sequence, stop, do the analysis. We need to stream the data off of the device. So in the kind of Unix philosophies, here's an example. You take Minimap, you pipe it through some various processes. And Nextflow channels are very similar to this. In particular, though, there's this kind of slightly esoteric thing, watch path, which allows you to start a stream before any data is present, and then watch and monitor and see new things come. And I'll highlight this point here, which is that this functionality is one of the major reasons we chose Nextflow over other workflow managers. There's a difference here, though, is that Nextflow doesn't handle streams of data, it handles streams of files. So in the context of this metagenomics application, we have a file of reads, we put it through Kraken, we get out some classifications, we take another file, and so on and so forth. So we have from a stream of input files, we have a stream of classifications. This isn't great. Each one of these Kraken processes would take upwards of 64 gigs to hold the database of all the species. Just loading that data into memory can take 30 to 60 seconds. And the other thing we want to do is we also want to accumulate these classifications over the time. It's not the stream of classifications that we want, it's the accumulated result. So we've got two pieces of the puzzle here to solve. One is how do we keep the database in memory? The obvious way to do this is to use a kind of server-client type architecture. So we load the database onto a server. We have clients that kind of can query that server, multiple clients if we want. The way we actually implement this is to have a Nextflow process start a server and then have other Nextflow processes start clients that talk to that server. So the servers and the clients, they're not talking over Nextflow channels, they're talking over kind of network protocols. The way we actually implemented this was something called gRPC, if anyone knows what that is. The server and the client we have this as open source software. You can use this independent of our Nextflow workflows. So if anyone wants to go, right, redo NCBI Blast with a more modern tool and wants to make Kraken, like, I don't know, NCBI Kraken, that would be the tool to do it with. The second part of this, I have to say, a kind of thank you to Paolo for doing this one evening. I think it was probably a Saturday night. Was after explaining exactly what it was I wanted from him, I'll get to the end, but he didn't do exactly what I wanted. But he made something that we could work with. So there's these scan processes. We're all familiar with a channel. We have an input and an output. And these recursive scan processes can take the output and feed it back into the input. So along comes another input, and we can combine that with the previous output. So in this simple case, the combination is just adding two numbers together. So 4 plus 3 is 7, 1 plus 7 is 8, 1 plus 8 is 9. That's a very simple example. In our real-world example, the input here is the classifications from each batch of reads, and the output stream is the accumulated classifications. As I said, Paolo didn't do exactly what we wanted. The implementation in Nextflow doesn't just give you the last output, it gives you the accumulating stream of previous outputs, so the entire history, which is a bit cumbersome. We only ever want the last one. And also the way tuples are handled in just the semantics and the grammar of the language don't really fit well with this scanning recursive ability. Right, so just to kind of finish and wrap this up, putting all of this together, we can run this in our application. So here we are just starting a metagenomics run, telling it to do it in real time, loading a database. This isn't with a real sequencing run. You'll see a bash window pop up here, and that's me simulating a run. So it will inject some, I think, TB reads come first. 20 seconds later, we inject flu reads into the system, and then 20 seconds later, we inject a more kind of community of things in. To start off, what happens, we see the Kraken server starts. None of the clients start until we see some reads. And then when you see some reads come into the system, multiple clients start processing those reads. And the kind of key part of this and the result that we want is to have these reports generated cumulatively over time. So first, those TB reads make it through the system. We generate a report just from those. Finally, the flu reads come through the system. We can generate a report from the TB and the flu. And then that larger community eventually comes through as well. And we kind of get our final report of all of these species. So we're progressively creating a larger and larger data set. We haven't waited for all of the sequencing to finish before we started doing this. This is obviously only a toy example. Right, so if you want to read more about this, we have a blog post here. We actually did this work quite some time ago. Just to conclude then, we have a sequencer, it works in real time, but until now we didn't really have a way of making and exploiting that. The largest nanopore sequencer can generate 64 megabases of data a second, so that's a lot of data. You can do something with that. And then Nextflow allows us to analyze this data and do something with it. So thank you to Paolo for kind of spending his Saturday evening implementing that for us. It was something that we wouldn't have been able to do this without that, at least in Nextflow. And with that, I'll say thank you.
Thank you so much for the very cool talk. I had no idea that was even possible, so that's very cool. We don't have time for questions, but if you can maybe put the link to the blog post in the Slack channel, I think that would get a lot of interest. Thank you so much. Next, we have Rika, who is going to talk about this little, not very well-known pipeline that's kind of new, and I'm sure we'll all discover something. It's SARIC. So Rika is going to talk about SARIC. Thank you very much.
Nothing happens. Perfect. Okay. Hi, everyone. I'm Rike. I'm a PhD student at the Quantitative Biology Center, and I will talk today about SARIC, so to catch everyone up that didn't know about it earlier. And we were just talking about it before the talk. SARIC is actually named after a Swedish national park, and it predates NF-core, which is why it got to keep its name and didn't have to get a descriptive name like every other pipeline. The pipeline is used for germline and somatic variant calling. Just to briefly catch you up here, so basically you have samples, tissue samples or blood samples, they get sequenced, in our case with short read technologies. Then the reads get aligned to a reference genome, and then we are interested in the differences between our reads and the reference, and we can do this for the tumor sample or the normal sample, or also look at the difference between both. And Sarek does this basically in 3 big blocks. The first one is the pre-processing, which is based on the GTK best practices. We have the alignment step, so aligning the reads to the reference, currently with BWA, BWMM2, or DragMap. Then we do duplicate marking and base quality score recalibration, run a bunch of QC tools, and then have converted our input FASTQ files to CRAM files. And then the next big block is the variant calling. So here we have 3 tracks. The blue one that you see is for the normal samples. The red one has tools for tumor-only analysis, and then the blue and red track for paired calling. And you can see we have a bunch of different tools implemented at the moment for SNP and indel calling, structural variant and larger copy number calling, and microsatellites. And you can mix and match these tools however you need. You can run a bunch of different SNP callers all in parallel, or just one, or one SNP caller, multiple structural variant callers. And however you need for your analysis. And then the last big block is the annotation part, so assigning knowledge and also predicting protein effects and so on. As you can see, we have several of these entry points, so these file icons here, to facilitate easier reanalysis. Let's say a project runs for a long time, and you were initially supposed to do some sort of SNP calling, and then throughout the project, somebody also would like to do structure variant calling. Then you don't have to do this really computationally heavy pre-processing, but can just start from the variant calling, and you don't need to keep the work storage around the whole time. And also we have round and then little squared station in this metro map overview, and the round stations are all optional and only the squared stations are mandatory. So Sarek is essentially just a huge wrapper around BWA, and you can turn off tools or turn them on to tailor it to your needs. The pipeline supports whole genome, whole exome, and panel data analysis, and is for short, paired, and reads. The pipeline was so originally designed for human and mouse samples, and with that we just mean now that basically for those organisms we ship all the reference genomes already, but it's been used for other organisms like pigs or tomatoes or bees or so, and you at least need a FASTA file for this, and then maybe need to provide some of the annotation files or so manually. Right, so Maxime already mentioned that Sarek was started a long time ago, 7 years ago, and several institutes in Sweden. Was ported to NF Core in 2019. And then I somehow joined the team and showed up, and we started porting Sarek to DSL2, and then released the first version of it last year. And while we were porting Sarek, we figured, since we have to rewrite the whole pipeline from scratch anyway, touch every bit of code, we should try and optimize it here and there a little bit. Also mostly motivated by some of my own research projects that didn't quite work out because of some bottlenecks. Yeah, and at the moment, Sarek is mostly supported by people from Cubic, Seqera Labs, the Danish National Genome Center, and many other volunteers in the community, of course. Right, so one of the major or main things people complain about is that Sarek uses too much storage space. So the work directory gets too big, it's a bottleneck, and it's then hard to use. So we figured, why not try using CRAMFile? It was on the roadmap for a long time, and there was always something preventing it. Not enough tools were supporting it, or there were other issues. But then 3 years ago now, actually, we tested all the tools, and it worked fine. So that's what we did. We converted the whole pipeline and then used CRAM files everywhere. And it happened, like the thing we expected actually happened. The work storage got massively smaller when we took the same pipeline with the BAM and the CRAM implementation. Also a bit faster. We did use more memory for some of these processes. In practice, this wasn't actually an issue because it was minimal per process, and then memory, of course, can be reused by further processes down the road, but the work storage just keeps accumulating. Right, and then the other thing that we looked at is how does one sample travel through the pipeline? Nextflow, of course, parallelizes across samples, but what does one sample do? And we start from the FASTQ file, and what we then added is that for each FASTQ file that enters into the pipeline, we'll send through a tool called FastP that splits the sometimes really big FASTQ input files into tiny baby FASTQ files, and then we run the alignment step in parallel. And this made the whole alignment step much, much faster. We did double the storage space for the FASTQ files because they had to be written once to the disk, but it was still worth it. And then duplicate marking takes care of the merging, and it didn't have any further time punishments or so on. And then the other thing we investigated is the big step-based quality scorer calibration and variant calling. So GATK recommends a scatter-gathering approach where you parallelize across genomic regions. So it can be multiple chromosomes or even parts of chromosomes. And especially for the human reference that we are using, there are about 370 regions specified. And SARIC kind of tries to be smart and groups these regions together to process them in multiple logs or groups. And we just had some number that has always been there, some type of grouping, and then we figured we should investigate. Is this actually a good grouping? Should we maybe do it a bit differently? And turns out we really should have done it a bit differently. Yeah, so let me show you the results here. So here I again ran the comparison on 10 samples. On the left you can see the runtime always for the longest running group. And then on the right side, the accumulated work directory per sample, work directory space per sample. And in SARIC 2 and very early version of SARIC 3, we for some reason just had about 124 interval groups. It was just the case. And the whole point of the scatter-gathering approach was to speed up the analysis, but as we can see, we don't actually speed up much further, or we don't get much faster after 21 intervals, but for some reason the work directory just keeps on growing massively. So we scaled this down. So now with the new default, it's 21 interval groups. And this again then reduced the work storage by about a factor of 4, and then also reduced other computational resources. Right, and then one of the last things we were interested in how do the cloud costs, or did the cloud costs develop over these different implementations? Here we checked it on AWS and US East using spot prices, the EBS autoscale, and then the old Fusion mount, so none of the fancy new things that Paolo showed today. And in SARIC 2, we ended up with about $70 per sample, quite expensive. Then in SARIC 3, we reduced it to 25. And then when we reduced the number of intervals further, we are now down to about $20 per sample. And I'm quite curious to see what all of these memcloud and new Fusion things and so on will bring. Also, the pipeline got faster. We also fixed a bunch of things where we had accidental bottlenecks in the pipelines and so on. Okay, and then before I come to the end of my talk, I just wanted to highlight some of the other cool new features that were implemented this year. So Anders spent a lot of time adding sentient support for mapping, duplicate marking, and I think as of this week, various variant callers. So it's a license-based commercial tool that can be used for these steps that is supposed to be much, much faster. Not only did he add it to SARIC, but he managed to add it to NF Core modules by getting a test license so it can be used by other pipelines, also without the developers needing the license themselves. Big here added joint calling for modC 2. So when you have multiple tumor samples, you can now do joint calling. Maxime, of course, added the annotation cache that he talked about earlier, as well as an option to only build the indices. So you can pre-run SARIC before you do anything, build the indices and then share them, for example, at your institute if you run on one of these organisms that are not currently in iGenomes. And then the last thing we looked more into is benchmarking with genome in a bottle data, looking at Illumina reads, of course, but also other technology together with Lasse and Edmund. The results look more or less as expected, and we are trying to now automate this on every release. So I got to rope in Edmund this week to help me with some issues, and we basically want to push the VCF files and then compute these F1 scores automatically and display them, because I could do it manually on release, but I know I won't and I will forget. And we should really have this on every release to make sure everything works as intended. Okay. This, I'm at the end of my talk, and I also want to use the opportunity not just to thank the usual people, but especially everyone who keeps contributing, who keeps answering issues and just helping us making Sarek more awesome and better and fixing everything. Thank you so much.
All right, awesome work. Well done to you and the whole team. We have a few questions, and we should talk about cost optimizations, by the way. A few questions. One, is it possible in Sarek to merge BAMs but turn off duplicate marking?
To turn off duplicate marking? Yes. Yeah. Yes, then SAMtools will just merge all the BAMs, convert them. SAMtools will merge everything then, and you can circumvent it, the duplicate marking, yeah.
Nice. A kind of a bigger scope on the second question. What are your recommendations for developers of other pipelines to increase speed and decrease resource usage, especially for developers that start from scratch?
Ooh.
It's a big one.
Bring a lot of time, I think. That's at least what I did. I think one of the most surprising things for me is, or that I found out, is that just cutting things up and parallelizing more and like running things more in parallel is not always better. So, and we try with small data sets. I tried with big ones in the beginning, and I wasted a huge amount of time on this. So,
yeah, I would echo that. It's definitely worth taking a little bit of time to see like how you chunk things up, depending on what kind of infrastructure you're running on as well. It's worth doing that and benchmarking, because it can make huge cost savings if you're on cloud, and speeds, savings, etc. Anyway, sorry, this is a topic near and dear to my heart, so don't let me start talking. It's not what I'm here for. Okay, well, thank you very much. I think that's the question. Oh, wait, sorry. Oh, there's actually a lot more. One more, and then we'll go to the next talk. How can we help new developers to work on a massive pipeline like Sarek?
Come and ask us lots and lots of questions. I think we all really want to help, and sometimes maybe we are not fast in replying because other things happening, but please keep asking. I really don't mind being reminded 5 times to help, and I don't know, we get on Zoom calls, we help each other out, and yeah.
All right, thank you very much. And now Lucas is going to talk to us about NF-test.
Hello, I'm Lukas Vorer from the Medical University of Innsbruck. And thank you very much for having me here and to give us the opportunity to talk about NF-test in front of such an amazing community. And all of us are loving Nextflow because it's a powerful and flexible platform, and it helps us to create scalable and reproducible scientific pipelines. However, when we create complex pipelines, then we are confronted with new challenges. Ensuring that it works correctly, our pipeline could be a very complex task. Adding new features without breaking existing ones could be non-trivial, could be time-intensive, and also maintaining such a complex pipeline requires a lot of time and could be sometimes a frustrating task. And on every change, we are confronted with the question, does our pipeline still generate scientifically validated results. And software testing is the process of evaluating and verifying a software product or a pipeline so that we can ensure that it does what it is supposed to do. And also automated testing is for scientific pipeline development essential because it confirms the pipeline's functionality, it ensures accurate processing and data analysis, and it helps us also to optimize performance and also cost efficiency. And overall, all this helps us to increase our productivity and finally validate our results and to create scientifically validated results. So the big question now is how we can test Nextflow pipelines. And to address this problem, we developed NFTest. And the idea of NFTest is to create a domain-specific language that is as similar as possible to the Nextflow language. And the main idea about NFTest is that we describe the expected pipeline behavior by using different blocks, the when block and the then block. And then we can use different assertions to create and formulate our assumptions of the pipeline. And NFTest provides you dozens of functions with the aim that we can write small, elegant, and readable assertions. And NFTest also simplifies handling Nextflow channel testing and all this Nextflow specific stuff. And behind the framework itself, it provides a command line program to generate boilerplate code so that we can easily start with new test cases without investing a lot of time of writing or rewriting existing code and we can save a lot of time. And NFTest also has the same requirements as Nextflow, so it's easy to install on your local workstation for your development process, but you can also install it on some continuous integration system, on GitHub Action. So on every push, on every commit, NFTest is rerun and ensures that your pipeline is working correctly. And NFTest allows you to test the whole scientific project, so your whole pipeline project. You see here a graph of the fetch NGS pipeline, and NFS allows you to test processes, to write simple unit tests on a process level, but you can also test integration tests. You can also write integration tests. It means you can test your workflows to ensure that the processes are connected in the right way. You can test your Nextflow channel operations. And you can also create end-to-end tests, so you can test the entire pipeline with all the inputs and outputs. And the good thing is that everything is done with the same syntax, with the same concept, so you can easily switch between these different levels of testing. And I think the best way to introduce you NFTest is showing you an example. Here we use a simple pipeline for FastQ preprocessing that use FastB and multi-QC. You can see here the simple pipeline. We have a main Nextflow file where we include the processes. We have here the FastB process and here the multi-QC process. And you can see here we load the new channel from the input parameter. We start the FastB process with this channel that creates filtered FastQ files and also a JSON file. And in the last step, we use multi-QC to create some fancy HTML report. And now in the first step, we want to test our entire pipeline. For that, we create a new file, an NFTest file. And each file is a test suite. And you can see here, this test suite starts with some specific keyword. In our case, we want to test a pipeline, so we started with Nextflow pipeline. Then we can give our test suite a name. We can specify the location of our Nextflow file. And then one test suite contains one or more test cases. A test case starts always with the test keyword followed by a name. And inside of this test case here, we can then define the when and the then block. In our example, we set in the when block the parameters. We have here some test file, some test fastq file. We set also the output directory. And then NFTest uses all these parameters set here in the when block to run the pipeline. Once the pipeline is completed, then NFTest executes here the then block to check your assumptions and assertions. In this example, first we check the status, the state of our workflow, if it's successful, and then we can open the two files and can ensure that the files that were created by this pipeline have the MD5 sum from the expected files. We can then use the NFTest command line tool with the test command to run our test script, and you can see it here. Each test case is executed. We have here everything green, so we see that we have no errors in our tests. And you can see here on this example, with only about 9 lines of code, we have written a test that ensures that our pipeline produces the correct result files. When we now change a parameter of the process, for example here, the Q parameter, and when we rerun this script, then we can see here that our assertion failed because we have a different output file with a different number of reads. So the MD5 sum is different, and we see immediately that we have some breaking change and we need to take some action. We can also test, as mentioned before, not only the entire pipeline, but also the process. You can see here we have simple to replace Nextflow pipeline with Nextflow process. We can then specify the name of the process we want to test, in our case fastp. And in the when block, we can now set the inputs of the process. In this case, we set the first input of the process to a file channel of our fastq test file. And in the then block, we can now check the process state, and we have also access to all the output channels of a process. For example, here we can check if the first item in the fastq channel has the expected MD5 sum. And you can see here that writing tests for a lot of output files could be very annoying because it's a manual task. You have to find the correct MD5 sums. And to overcome this, we introduced snapshot testing. And the idea is to reduce all this manual validation by creating a snapshot that is then automatically checked by NFTest against the current run. And it enables to make regression tests more easily, more efficient, and it also ensures that you have, with a single line of code, a possibility to check that you have reproducible results and that the output of your pipeline is consistent across runs. We can see it here on this sample. We have here then with different assertions where we check different files, and that we can now replace with this line of code where we create, because we create now a snapshot of the fastq output channel, and NFTest automatically creates for us this snapshot file and then checks if it's matched in every subsequent run. And the snapshot is very flexible. You can include different objects, multiple objects, so it's a very nice possibility to write simple and efficient test cases. Besides snapshots, we have other highlights in NFTest. We support YAML, JSON, or also JZip files. We have also some plugin mechanisms so that users or the community can write additional plugins to add additional file formats. For example, you have a faster plugin. And NFTest is also written like Nextflow in Groovy, so it's possible to import and reuse any existing Groovy or Java library, so you can easily use existing libraries to write nice and efficient assertions. And since the last release, we have also process dependencies where you can run one process to generate the input data of another process. And the best way to get in touch with all these features is to read our documentation on our website. And here we have different tutorials. We also have collected resources from the community. We have videos, we have tutorials. So I think it helps to get in touch with NFTest. So one last question, what's the next step of NFTest? So our idea or our aim is that we try to simplify the process of writing, running, and debugging test cases. And for that, we are working on a Visual Studio Code extension that automatically parses your NFTest files. You can see it here. It detects all the tests, and when you then click on this play button near one test case, it automatically executes this test case. You can see the output of NFTest in the terminal window, so it's very easy to run tests with one simple click. And this extension is also integrated very seamless into Visual Studio Code. So you can click here on the typical test icon. You see a list, a tree of all founded test cases in your project. You can run tests on you can run one test, or you can run all the tests at once. So it's very easy and helps you to develop test cases. We hope that we can release a first beta in the next weeks, and this beta will then available on the following GitHub link, but we hope that we can make this process more fun and efficient with such an extension. So before I conclude my presentation, I would like to thank the community. It's fantastic to see how a two-man show project as a side project is now a community project where a lot of people from the NFCO community invested so much time to bring it to the next level, to push it, and it was fantastic yesterday to enter in a room and to see that so much people are using NFTest. And with all this, thank you very much for your support, thank you for your attention, and enjoy the Nextflow Summit.
Thank you. Thank you so much. That was extremely clear. I understood almost all of it. And for context, I ran my first Nextflow pipeline on Monday this week. So very clear. Thank you very much. We don't really have time for questions, but there's very active discussion on Slack. So I recommend you join that.
Thank you.
And thank you very much for your talk. And now Heath is going to talk to us about empowering distributed precision medicine. Thank you very much.
Okay, thanks very much. My name is Heath O'Brien with Sanogenetics, and I'm excited to tell you today about how we've started to use Nextflow in our efforts to accelerate the adaptation of precision medicine. I should probably start by defining what we mean by precision medicine. It's not just a fancy buzzword. There are a lot of genetic diseases that are actually defined at the cellular level. So ALS is characterized by degradation of motor neurons. Parkinson's disease is a defect in dopamine production. And it turns out that there are a lot of different changes at the genetic level that can all basically cause the same symptoms at the cellular level. And over the last decade or so, we've started to see the development of a lot of really powerful therapies that target particular pathways defined by specific genetic mutations. So when I talk about precision medicine, I basically just mean tailoring the therapies to the particular genotype of the individual that has a disease. As I mentioned, there's been a huge amount of progress in this area. Pharma and biotech companies have huge pipelines of really promising drug candidates. But thus far, there hasn't been all that much impact on patients from all this effort. And there are a lot of reasons for that. But the one that we're focused on is a chicken and the egg problem, which is that these therapies can only be tested on people with particular mutations in their genomes. And because none of these precision medicines have made it into clinical practice, genetic testing is often not done as part of routine clinical practice. So there's not a pool of known people available to test these medicines on. This is combined with the fact that we're talking about generally very rare diseases, and now we're subsetting those into even smaller populations. So you need to cast a very wide net to find people who would benefit from these treatments. And traditional clinical trials are generally run out of a small number of trial sites. So you're asking people, if you're recruiting very widely, you're asking people who are, in many cases, profoundly unwell to travel to these trial sites to take part in the pre-screening. And then if they're immediately screened out based on their genetics, it's pretty frustrating for the whole process, not least the participants. How we're trying to contribute to this problem is basically by bringing the screening for the clinical trials to the participants. So we're basically developing a tech platform where participants can be recruited into this system, fill out pre-screening questionnaires online, receive a genetic test kit, and send that off for genetic testing. And we can also integrate with healthcare providers in various ways. And all this can be done without the participant leaving home. And then only if they're with high certainty going to actually be eligible for a clinical trial will they have to travel to a trial site to participate in it. So in addition to working with pharma and biotech companies, we also have a lot of population genetics initiatives that we're working with and patient groups. And we found that they're often very concerned about data sovereignty, so they want to make sure that all the data stays within the infrastructure that they control. And they also want to have a very consistent experience, so the branding and the graphic design and stuff of the portal is very important to them. So we've evolved from just running studies within our platform to doing this as a software as a service where we can actually stand up a copy of our portal set up in a customer's own infrastructure, and then they can run their own recruitment and engagement in part of this, as part of that. I forgot to mention on the previous slide that a key part of what we do is people, regardless of whether they screen into or out of a clinical trial, are kept engaged with educational materials and updates about advancements in the particular disease area that they're concerned with, so that when additional clinical trials come along, we have a pool of people that will potentially be eligible for them and can be screened in much more efficiently. And so that applies to these portals as well. So because we're running now this network of portals, scalability is really important to us. So that I don't just mean scaling up bigger and bigger, I also mean that we've got some pretty small patient groups using this, sometimes in pretty restricted time zones. So we don't want to have a huge amount of infrastructure that's always on for smaller projects. So we've adopted a completely serverless infrastructure. So basically the whole portal is handled through these AWS Lambda functions, which are great for making the website run with pretty easy setup and easy scalability, but they have very limited compute. Execution times are capped, so they're not really useful for data analysis. So we've been using AWS Batch for these longer running jobs, including processing genomic data, et cetera. We found that provisioning different compute environments with different resources for different processing jobs is a real bottleneck because the skill sets required for writing the pipelines don't overlap with the infrastructure skill sets required to deploy them. So this is where we've started to use Nextflow, not so much in our case to handle orchestrating complicated pipelines. We have the event-based execution within the AWS Lambda that handles most of the logic of our data processing. Nextflow allows us to basically easily request whatever resources we need for AWS Batch, so it's proved to really accelerate that process of being able to deploy different pipelines without having to write a bunch of Terraform code to stand up new AWS Batch infrastructure. And yeah, so we're using this for standard genetics things like variant calling, genotype imputation, et cetera, but also for much more mundane tasks where we've really been coming up against the limits of the AWS Lambdas, converting files and copying large data sets and all the things that any bioinformaticians are probably familiar with. So we're quite new with our journey into Nextflow. We basically just started implementing pipelines using it. So we're still learning a lot about it, but we're quite excited about the future potential of what can be done with this. So currently, we're writing pipelines that are then integrated through our system, but we'd love to get to a point where clients are able to analyze the data within their portals in whatever ways suit their needs. So basically writing their own workflows and data analyses. So we're quite excited about the developments with Seqera Cloud and the interactive ability of that so that people can do all their data analysis without having to export data from the portal. One thing we're very interested in is allowing people to develop pipelines using a graphical interface. So we have a similar problem with designing our screening surveys. They involve a lot of complex logic where people can screen out at different stages and move down different paths. So we think there's some similarities there, and ideally we're developing ways for people to reason visually through those graphs, and we'd like to ideally have a seamless interface for writing data workflows as well. The other thing that we think that Nextflow may open up the possibility for is for customers to write workflows that run on other instances of our portal in other infrastructure systems through a networking system, assuming that all the permissions are in place and the consents that potentially workflows can be run on other people's data and aggregated results returned without having to share the underlying data. We think there could be a lot of power there to really start to increase sample sizes to power further discovery in this area. And with that, it just remains to thank the rest of the bioinformatics team at Sano, including Adam and Matteo, who are here, the organizers of this fantastic meeting, and all of you for your attention. Thank you.
Thank you. That was a very interesting talk. So what you were describing towards the end there, it sounded like kind of a federated system where you would potentially be able to connect to data repositories in other places. Can you say a little more about that?
That's the dream, yeah. I mean, we're pretty early stages of thinking about that, but yeah, I mean, I've been involved in some large meta-analysis projects with GWAS and things like that, and there's room, I think, for some large increases in the efficiency of sort of trying to get dozens of groups to run consistent workflows and then aggregating the data. So if there was an easy way that a single analysis could be run across all those data sets, I think it could potentially be really powerful.
That is the dream, yeah. Thank you so much.
All right, thank you.
Thank you. All right. Next up, Louis is going to tell us about genomic imputation.
Hi everyone, I'm Louis Lenezet, and I will present you for the first time today my work on the new NF core pipeline phase impute dedicated to genetic imputation. But before presenting the pipeline in itself, I would like to explain you why genetic imputation is useful and how it works. As many of now that the Nextflow community is growing, some of us are not necessarily familiar with the genetics and how it works. So genetic studies aims to understand how genetic variation influence traits, but also disease. To do so, the first thing we need is reliable and as comprehensive as possible DNA information. When using sequencing techniques, we get a sequence of nucleotides called reads that we align to a reference genome. We then identify the difference between the target DNA information and the reference genome by calling the variants. As you may see, the genetic variation is noted as a combination of two nucleotides called allele, and indeed, humans, as well as many other species, are diploid, meaning that they possess two sets for each chromosome. One is inherited from the father, and the other one from the mother. Therefore, they aren't completely identical. As a consequence, we need to distinguish between a genotype, a combination of allele at a given position, and a haplotype, the sequence of allele along a given chromosome. When studying a target DNA information, we may miss or not be highly confident in the genotype observed. Phasing and imputation are statistical analysis that helps to overcome this problem. Phasing aims to align alleles into haplotypes. We can then distinguish which allele belongs to each parental chromosome. This employs a hidden Markov model which considers the probability of recombination event and point mutation alongside the chromosome. Imputation algorithm, on the other hand, use statistical model to predict the likely unobserved alleles. But the phasing and imputation software, the use of them will be dependent on the data in input. It exists effectively multiple technology to extract the DNA information from biological samples, with a balance between the quantity of information we get, its quality, and its cost. High coverage whole genome sequencing provides the most comprehensive data by sequencing an individual's entire genome with a high number of reads covering each position. It allows to be highly confident in the genotype observed, but its downside, it's quite expensive. Genotyping, on the other hand, focuses on specific markers of interest, providing a cost-effective and targeted approach. Low-pass sequencing is a balance between the two. It sequences an individual's genome at a lower coverage, reducing the cost, but still provide more denser information than genotyping. Phasing and imputation analysis enable bioinformaticians to get the most out of their data. For high coverage data, phasing allows to study the haplotype context of variants and hence its consequences on disease. Phasing shines even more when used with imputation on sparse data to complete the missing genotype. More specifically, for low-pass sequencing, it allows to still detect new variants of interest and then to uncover rare causal variants for disease at a low cost. To help people to use phasing and imputation software easily, I've decided, with the help of Sol Pierotti and Annabella Trigila, to create an NF core pipeline dedicated to genetic imputation named PhaseImpute. The goal for the moment is to develop the following pipeline. It's a bit complex. It consists of multiple linked processes that I will now describe. The first one prepares the reference panel to be used for the next processes. The main step will be to phase it, if not already done, do some quality control, create specific software files, and separate each chromosome to chunk them in small bytes to parallelize the imputation process. This imputation process is the core of the pipeline. It is used to impute target individuals' files, BAM, CRAM, or even VCF, and to output the corresponding imputed VCF. Many different softwares already exist, and our aim is to make them all easily available in this pipeline. I'm helped on this task by Sol Pierotti for the Stitch software and Annabella Trigila for the Kilt. To ensure that the pipeline is working correctly, but also to perform accuracy analysis, I developed a simple simulation process to then sample a high coverage target file into either a genotype or a low-pass sequencing simulated file at different depths. This will be then imputed and compared to a truth VCF file in the concordance process. These steps will either use the concordance tools of Glimpse or a workflow in development by Sol. It generates concordance statistics that will help us to identify the more impactful parameters in the imputation process. Finally, a refinement process proposed by Sol will allow to filter the best imputed position in the imputed VCF and rerun the imputation process to improve the accuracy. The data needed to run this pipeline are the following: the target DNA information, some reference genome file, the reference panel to be used for the imputation, as well as some additional file for specific softwares. For the concordance analysis, you will also need a file with the allele frequency and a truth VCF to be compared to the imputed one for each individual. The main challenges I faced so far while developing this pipeline are the multiple entry points due to the multiple processes. As a consequence, I need to configure multiple CSV input files and to correctly set up the succession of processes. Another nightmare that many other bioinformaticians face in genetics is the presence or absence of the CHR prefix in multiple files. I will need to choose one configuration while aiming to reduce the duplication of file. As an example, I would like now to present you some preliminary results we have on human data with a collaboration with Anthony Herzig from the laboratory of Emmanuel Gemna from the INSERM in Brest. We performed concordance analysis on 16 French individuals using the version 37 of the human genome. To do so, we used the 1000 genome reference panel phased, but also the same panel in addition to 800 French individuals. We simulated 4 different depths, studied the longest and shortest chromosome with or without a genetic map. For the allele frequency, we use the reference panel, and we compute the truth VCF for each individual using BCF tools and pileup. Concerning the imputation tools used for this analysis, we only use Glimpse 1 for the moment. Here are some of the results. On the x-axis is represented the minor allele frequency in percentage, and on the y-axis is the imputation accuracy from 0 to 100%. As expected, imputation accuracy tends to decrease as the allele frequency becomes less common and as the level of coverage decreases. Here, it's a bit more complicated, but it's exactly the same. It's compared here the chromosome 1 in light color and the chromosome 21 in dark color. The size of the chromosome doesn't seem to have a huge impact on the imputation quality, except maybe for lower coverage or rare variant. Similarly, the presence of a genetic map doesn't seem to improve a lot the imputation accuracy. More interestingly, on the other hand, the addition of individuals with a shared genetic background to the target DNA individuals inside the reference panel seems to impact positively the imputation accuracy. Moreover, this effect seems to be even more pronounced for rare variants. In conclusion, the phase impute NF-core pipeline will enable bioinformaticians to easily use imputation software and to test their accuracy. The first results are encouraging, but need to be expanded to other imputation tools and other configurations. I aim to use this pipeline for my PhD thesis to identify genes involved in canine dysplasia, a complex genetic disease, and to perform a comparative study with human data. I want to highly thank my team and our collaborator from the GGB team, the funders of my thesis, and of course the NF-core community, and more particularly Saul and Annabella for their contribution to the pipeline. Thanks for your attention, and I'm happy to answer any question.
Impressive work, thank you. So you have multiple pipelines involved there. How are you going to combine them? And specifically, are you planning to build a new mega pipeline concatenating them, or are you going to run them separately and sequentially? Does that make sense?
We were thinking for a time to add it to Sarek to make a meta pipeline with everything, but it would have been complicated. So the aim is really to have all those connected inside as presented in the global metro map. But yeah, for the moment, we want to stick on this plan, but I'm happy to improve. We are really at the start of the development of the pipeline, so we are open-minded on everything.
That's fair. That's fair. I think there's a balance to find between trying to get everything into one thing where you press the button and everything runs, and readability and usability. So, good luck. It's going to be. Wait. Hang on. Don't go. I still have one more. How does the performance of the pipeline compare to web-based services for imputation, such as the Michigan Imputation Server?
Yeah. So that was one of the aim to use this, to create this pipeline is I'm working on dogs, and there isn't a lot of services allowing to use the new version of the dog's genome. And we asked to have the new panel available, but they asked for an astronomical cost to add the new version. So we wanted to have everything available easily and in open science. So for the moment, we don't have really performance. So the quality of the imputation is broadly the same as what we can find in the literature for the moment. But for the computational resources, I still have a lot of work to be done to compare to the available public services.
That's totally fair. And it will be a great resource for also other groups who want to run on other organisms, so extensible and all of that.
Yeah, that's the aim.
Yeah. Well, good luck. Thank you very much.
Thanks.
And now we're going to have our last talk today, and I'm not saying that these guys stand between you and dinner, but they kind of do. I'm kidding. We have a good half hour, and we have 3 speakers for this one. It's a little special. So we have Luke Paul, Blaue, and Ricardo, who are going to talk to us about porting workflow managers to Nextflow at a national diagnostic genomics medical service. So please proceed. Thank you.
Awesome. Hi, everyone. I'm Luke. I'm a senior bioinformatics engineer at Genomics England, and it's actually great to be here wrapping up the second day of this conference, speaking about how we're migrating to Nextflow. I'm joined by my colleagues, Ricardo and Borje. So I'll be going through a bit of an introduction about our organization, then we'll speak about our unique migration strategy and refactoring work that we're planning after that. We'll be speaking about how we're validating everything we're doing, and then we'll be sharing lessons learned with everyone. Hopefully you get to learn a little bit from our experience as well. So our organization is Genomics England. We were set up in 2012 in England, primarily to deliver quite an ambitious project, which was to sequence 100,000 genomes. The aim for this was to kickstart the genomic medicine revolution in the UK. We actually succeeded in delivering this in 2018, and thanks to our success in doing that, the UK government decided to start offering this as a routine service through the National Health Service. Now we have a new project that we're hoping to start off, known as the Generation Study, and we'll be scaling up to support this, and this is part of the reason why we're moving to Nextflow. So briefly about the organization, we're split into 2 different tribes. There's the clinical tribe, which we fall under. We are fully accredited, and our bioinformatics pipeline is a medical device. When patients need a genetic test, their data is sequenced by Illumina, then it is sent to us, where it's passed through our analysis pipeline. Findings are returned to a clinician, and with their consent, their data is added to a trusted research environment where further insights might be gleaned by researchers who gain access after being vetted. And essentially, if any further analysis is discovered, that further knowledge is added to our diagnostics to help future patients when their data hit our system. So we fall under the clinical part of the organization. So we're migrating to Nextflow primarily because our current system, known as Bertha, while it served us well, it was hyper-optimized for the 100,000 genomes project, and would like to start supporting new use cases. In addition, workflow management development, solutions development is not our core business offering, so we don't really want to do this anymore. And leave it up to the next engineers. We'd also like to start supporting a wider range of compute environments, because right now we're running primarily on our on-prem infrastructure, and we'd like to start using public clouds such as AWS. And this would support our ambition of, for example, completing the Generation Study, where we are aiming to sample 100,000 samples of newly born children across England. So this is the project which we are working towards to replace our current bioinformatics workflow engine. It's a combination of Nextflow and Nextflow Tower and uses AWS functionality for automation. We call it Genie. And with that, I'd like to pass to my colleague, Boaje, to speak to us about the migration strategy.
Thank you, Luke. My name is Boaje Szczerba. I am software engineer at Artigen, and we are working with Genomics England for over a year in this project. And yeah, this is how current sample journey looks in Genomics England clinical services. So at the end and at the beginning of the journey, there's a patient. Then there's a whole genome sequencing of patient sample. Then the sample is ingested in our system, and our in-house workflow manager, Berta Orchestrator, starts 3 workflows. And this workflows is a set of bioinformatics modules. At the end, the result is dispatched back to the clinician and then the patient. It is worth to mention that Berta is a critical medical device, and this is really important when we're looking at the priorities that we had in the project. So our first priority is to get out of Berta as quick as possible, but we need to think about minimize the risk and minimize divergence between old pipeline, old workflow manager and new one, because this migration should be transparent for patients. And the last priority for us was to work in small iterations to validate if our migration is going well and to again, minimize risk. So, at the last summer, we wanted to explore 3 strategies. We wanted to see how we're going to do that. So the first strategy was to full refactor to Nextflow. So in this strategy, we rewrite from the ground up the workflows that are written in Berta, and we're going to leverage all Nextflow features. However, this strategy comes with great risk of divergence because Berta is still in active development, and the overhead updating during the migration is not acceptable. The second strategy was strategy lift and shift. So as Berta is Berta Compute, the set of bioinformatics modules that is used in Berta, this is used in on-premise infrastructure at Genomics England as singularity image. We can leverage that and use this image, this container, right away in Nextflow. So this comes with low risk of a rally. So we know that the same code works exactly in Berta and Nextflow, and it also looks as the quickest strategy. The first strategy that we had in mind is to extract gel libraries from Berta Compute. So Berta Compute is a monolithic software. It's a Python package included in Singularity image. And in this strategy, we wanted to separate that to the smaller packages and import it back to Berta and Nextflow. So the risk of divergence is still low. We have really nice architecture because we can actually use any workflow manager that we want, but still the time to do this intermediate step is a really important factor. So we decided to go with lift and shift strategy. So we're going to use this singularity image with Berta Compute code unchanged in Genie. It is highly conservative. As a next step, we're going to deprecate the orchestration, the Berta orchestration part of Berta system. And then we're going to refactor our pipelines that are already orchestrated by Nextflow to leverage all Nextflow features. So this slide you've already seen, and this is our proposed solution. So actually, we want to replace Berta orchestration with Nextflow and Nextflow Tower and automate this with AWS. So one of the challenges that we faced during this migration is to assure that Nextflow and Berta Compute code has a common interface. To do that, we created an adapter over the Berta compute that basically translate the execution parameters from Nextflow to meet Berta compute module's requirements. It also communicates with some external APIs. This functionality was before included in Berta Orchestrator, and now it's a part of our wrapper. So this is exemplary code that we had right now in Nextflow. It's really easy. So this is just calling a Python library. As you can see, there's no bioinformatics code here right now. This will be the part of the next step. So we see that we are using the same code that are used in Berta. We just call Berta from Nextflow. So the migration status for now, we migrated 2 clinical programs, rare disease, and we created a generation study program. This includes 5 migrated workflows and over 60 components from Berta that are now executed in Nextflow as Nextflow processes. And that was my last slide, and I give back voice to Luke about the refactoring strategy. Thank you.
Thanks, Bajay. So as Bajay described, we're migrating in phases, and this is a later stage where we will be refactoring our migration once we've exited Berta Orchestrator. The initial version of workflows when we migrate is going to be optimized for minimizing divergence and minimizing risk, but less for taking advantage of everything that Nextflow has to offer. In addition, we're going to have 3 distinct workflows per sample to run, just like Berta used to have it. And actually, we had to create another overarching workflow to link these 3 workflows. We're calling it the conductor workflow, which runs before and in between each of the other workflows to get them going. The modules that we've moved over from Berta contain a mixture of bioinformatics logic and API calls, which can make them brittle to run, which is not great for us because we need the highest reliability, given that we need to return data to our doctors in a very short time. And another disadvantage to how we migrated is the fact that we're not using channels actually for our file handling at the moment. We're still using file handling that used to be used in the previous system, which is basically written in Python and contains things like direct OS file calls, which makes it difficult to run on things like S3, for example, using S3 as a storage layer. So we have 2 horizons of delivery, which will be following to future-proof our workflows. I'll be running through some of the ideas we're going to pursue. The first is to split up this big singularity image into multiple images, 1 per team working on bioinformatics code, just to allow them to work independently and in parallel. We'll be doing an audit of the module logic to see if there's any legacy logic which has built up over the years and isn't needed anymore. We'll be switching over to using channels, which will make it much easier to use S3 as a storage layer, which we're quite keen to do. We'll also be following NF Core best practices when it comes to separation of concerns in our modules, such as separating API calls from bioinformatics logic and making sure a module does only one thing. And would also love to use as much of NF Core standards as possible. That includes the way modules are shared and documentation as well, because I think there's a lot of great examples there that we can emulate. As a second horizon of delivery, we'd like to create adapters for third-party services that might be getting swapped out to make it easier for us to do that. We'd also like to explore merging the 3 workflows into one to make orchestration simpler. We'd like to profile our code to see if there's any room for improvement. And finally, we'd like to look through the code, which might have implicit dependencies, and actually expose all of that as parameters, basically to make them more portable, to be able to place them anywhere in the workflow without any issues. And I guess with all of that, this is quite complex work, and since we're dealing with patients' data, a key part of our process is validation and making sure everything works as it should. And with that, I'll pass to Ricardo, who will speak to us about Jasmine, which is a custom automation testing framework that we created to assist us.
Hi, I'm Ricardo, and thanks, Luke, and as Luke said, I'm going to talk about how we are validating our migration. So we have a system that we're calling it internally Jasmine, and it's our testing environment. And the reason for this to exist is because we have very, very complex dependencies that may include hardware, the architecture where we are running, software. We have to keep a good track of which version of each software we have, because as it was mentioned before, it's accredited as a medical device, so we need to be very precise with that. And there are also interactions with humans. So there may be problems with the sequencing. It may be that we detect that the samples don't correspond to the family. So or there was a mislabeling. So all these kind of interactions require to be processed by someone. So that decision needs to be made if there is a deviation on the pipeline. So for that, we use external systems like Jira. So this is just a selection of some of the systems that we have to interact with. So how do we make sure that all these interactions happen correctly? So we have a we use Docker Compose to coordinate several images that correspond to our workflows initially, and also to these third-party systems. And the idea here is that we want to be able to run the pipeline and connect to all these external services locally in our development environment and in continuous integration. That way we avoid having to go and test with full samples. I'll talk about that a little bit later. But one of the things that we found out, we thought like, oh, how hard can it be to set up all this to run locally? Well, it ended up most of the year. Now we are in a good position where we can test things quite efficiently, and we are already finding bugs quite quickly. But yeah, it took a little bit longer than we expected, because there were actually more services than we initially thought. But that's fine. Right now, it's mostly done, and that's a good thing. But the other thing that we have, it's because this is an accredited pipeline, we have a set of sentinel samples that we know that we need to find some mutations, or we need to find some variations that are related to certain clinical results. And this set of tests take them anywhere between 2 and 3 days to run and to be analyzed to make sure that they work properly. And we have a quality assurance team that keeps, every time we do a release, they go and test that things are still working as expected, and if there are changes, that they are also reflected on these samples. However, we are migrating the pipeline. We cannot afford a test time of 2 or 3 days just to figure out that on the 20th component we had a typo, right? We need faster testing. So for that, we created a smaller sample. So we have a fake genome where we caught the first 100,000 bases of the human reference and created an annotation, and in some cases also included variants in this region that we can test just to make sure that the components go through it. We kept the X and Y chromosomes because some of the validations are related to the sex of the patient. And for that, you need a full chromosome. So anyway, that allows us to go from 2 or 3 days to run the pipeline with the real samples to having a continuous integration that can take 15 minutes. Also, we can run it. It takes a few minutes on our development environment. I mean, it's not instant, but still it's way better than having to wait a day to find some errors. So we move from developing directly on the cluster to developing completely locally. The other thing that we have to do to speed up or to be able to make the migration, it's to simplify how we look at the workflow. If we look at the full workflow, it looks quite complicated. I mean, it's roughly linear, but still there are several modules that have to be tested. So instead of trying to test everything in one go and trying to understand everything, we simplified the workflow and we divided in the natural checkpoints that it already has. And then the workflow at the big scale, it becomes quite linear. So you can see that the actual workflow, it's just one step after the other. However, when we look at the detail, we use the ability of using workflows as modules in Nextflow. So that way we can abstract some of the complexity. In this case, this section has three concurrent processes, and then the actual Nextflow code, it looks a little bit more complicated than just having a linear sample, but it's a linear workflow, but it's still a complexity that you have the diagram and you can read the code and you can see that it matches. I mean, it's not trivial, but it's not that difficult. And then in order to test and to make sure that every part of the workflow works, we have this idea of subsections where we only test the linear parts of each one of the workflows. So in this case, we run a test of just two components on the example, and we run the prerequisites. We assume that the upstream part of the pipeline is already working, so we are migrating from the top to the bottom, and we assume that that works, and we can make sure that this subsection is still working. And every time we make a change, we run continuous integration to make sure that whatever change we made doesn't break something that we already know that works. And that has helped us to keep a little bit sane. So as we mentioned, we have our workflows are accredited as a medical device. So that means that if something changes, we need to report it. We need to make sure that everything is exactly the same all the time. So in order to test the new workflow, we need some sort of ground truth. The ground truth is running the old workflow and having the same results. And the same results is not only the files, it's also all the side effects. So as I mentioned, we have these external databases that report to our users and to our providers. We also keep track of all the files that we generate for audit reasons. So if somebody comes and asks why we have these results, we can go and check every single file and make sure that everything runs correctly as expected. So we need to make sure that our new workflow still is consistent with these requirements. So we save the state of the database and this Docker, not only a database, but all the databases and all the files in the Docker Compose set of images, and we run it twice, once with the old workflow and once with the new. And then we check that everything is exactly the same. Of course, it's not trivial. Some files have timestamps, so of course that's not going to be the same, but we have comparators. So that way we can make sure that our system is still the same. Just as a reminder, we are just changing the workflow manager. We're going to change the pipelines in a later stage or the modules, but that's not the aim right now. And for new workflows and for cases where when we have already depreciated Bertha, we can also make a fake state of or unexpected state of the outputs, and set that on what we use right now to compare. We can also use it to feed a known state or unexpected state, and that way we can also validate that things are working correctly. So finally, I want to talk a little bit of the lessons that we learned over the last year. Many of them are like some things that we have to question ourselves. So one of them is how can we make sure that whatever we are running, it's always correct. And one of the problems that we had before is that whenever we updated the pipelines, we couldn't be completely sure that it was the same. So right now, every time we have a release, we have a singularity image that has the exact versions of all the software, so we can tell this sample was run with this version, and this is what we expect. So this is mostly because the workflows are still evolving. So we are a provider for the health system in the UK. So whenever they detect something that they need, we need to update. So this is a moving target. So this helps to keep our systems consistent. For the same reason that the workflows are changing and the expectations are changing all the time, we cannot just shut down everything, refactor everything, and start over. So we need to keep the system working all the time. So for that reason, we are using an iterative model where we are just changing one piece of the system at a time. Eventually, we hope to have a linear system, a more efficient system. But right now, we need to take this approach because we cannot afford to stop. Another thing is, as part of the migration process, we are probably not interested on every single line of code in our team. Of course, there are teams that are working on each one of the modules and all the details, but we don't need to understand all this as part of the migration effort. So we use black box testing. So this testing framework, once you run it, you know what happened. If there is some error, if we find that the systems diverge, then we do analyze the code. But that saves us having to read every single line of code. We only focus wherever we find any discrepancy between the systems. We also want to make sure that we find errors before they go to the end-to-end testing. So the end-to-end testing is this process where quality assurance runs all these Sentinel samples. If they find that a problem with our pipeline, it may take like 1 or 2 weeks since the time we release that code for the testing and come back. By the time we get the results, we have no idea what we did 2 weeks ago. So we want to make sure that we find these divergences as soon as possible. So having this testing environment has helped us. The other thing is how we were wondering how are we going to tackle this problem. We look at the pipelines like this is a mess. It's very big. There are too many things going on. So sitting down and deciding to split it and take this, okay, we advance a little bit, has kept us moving. Like we feel that we have progress, even if we are about to finish. And right now we feel like we are really close to meet the first deadlines, but at the beginning it was very daunting. But having these ideas of sub workflows and subsections, and okay, this week we are testing this, makes it keeps the morale up, and it keeps the focus on something instead of panicking because you don't see the light at the end of the tunnel. The other thing is that it's very important to have a test data set that you understand that you can run locally, especially in genomics with the size of the data sets. Waiting for things to run, it's not fun, and it can also be a little bit demoralizing. So having this tiny data set has helped us a lot to keep the rhythm of the development. And the last thing is how we were really concerned of how to make sure that the old and new systems work the same. So having these databases and file systems in a fresh state each time we run the system has been extremely helpful, because we don't need to worry, which is the ID of the ticket that was generated. We don't care. It's always one. So as final remarks, I'd like to remind you all the things we cover today. First, we had a big migration to do, and we have to decide several migration strategies. Some of them included rewriting everything from the scratch, but given our organizational necessities, we decided to just lift and shift. So we changed as little as possible on each iteration. We also talk about the refactoring strategy that it's happening in parallel of the migration. Some of these changes were necessary to move to Nextflow, and some of them will happen after we have we have depreciated the current orchestrator and we are fully on Nextflow. we also talk about the validation approaches that we have and how we simulate a testing environment that it's very similar to our production system, but in a smaller scale. And we talk about the lessons that we learned. Finally, I want to thank you for your attention. And well, all these people has been involved in our project. This is not just the three of us. It's a large project. And if we have any questions, we are here to answer them. Thank you very much.
Thank you. That represents a phenomenal amount of work, and I want to thank you really for sharing both the thinking that went into the thought process to develop this approach, and also the lessons you've learned so far. I've witnessed I had front row seats to a major sequencing center migrating its pipelines, and it can be a fairly daunting experience. It's really a phenomenal amount of work. So I think this will be a very valuable resource to the community. I believe a lot of people will be watching with great interest as you proceed and continue to go forward. And hopefully, I think a lot of people are hoping you will share the outcome 6 months or a year from now. I don't know what the time frame is, but you've got a lot of people anxiously waiting to hear from you. There were a couple of questions about validation, which Ricardo answered during his part of the talk. A lot of questions came early. A few additional things. Oh, yes. One, do you find a lot of divergences, or is the lift and shift strategy successfully keeping this to a minimum so far?
So, yeah. So we haven't found many big divergences on the modules themselves. We find some on the orchestrator side. So because the workflows were tightly integrated with the orchestrator, sometimes we realize that some of the behavior that was happening on the orchestrator, we have to move it to the component themselves or to the component wrapper. That's where we mostly found divergences. Not that much on the modules, but we still need to make sure that things are consistent.
One question about in the process of revisiting the code, have you found any bugs with the original approach?
Yes. So yeah, the short answer is yes. Probably Voya has a lot of fun with that.
I had a lot of fun with finding bugs in Berta, but yeah, this project also helped us to make our product even better.
And I guess one more thing. You've explained kind of the key factors that informed your decision to go with this approach. If you were thinking strictly from the point of view of accreditation, do you think it would be harder or easier to get accreditation with just a completely new code base as opposed to this approach?
Yeah, so I'm not an accreditation expert, so I'll answer to the best of my ability. But yeah, I can maybe ask our experts at Genomics England for a full answer. But I think from what I know, accreditation is based a lot on the usage of the pipeline. So when you're writing something from the ground up, because it's written on, like if it's completely new code, you basically need to create completely new documents describing what the code does, and then get that audited essentially. And I guess in our case, because a lot of the algorithms that do a lot of the bioinformatics analysis will remain the same, the auditing requirement is believed to be lower overall. Okay. So that's how we this approach has helped us in that regard as well. But that bit of the work is still outstanding as well, so this might change once we have a chat with our auditors.
Fair. Mm-hmm. Well, good luck. Thank you very much, and we'll look forward to hearing from you on how it goes. Awesome. Thank you so much.
Thanks, everyone.
Now, that was the last talk, so I'm going to liberate you in a moment. But first, I'd love to get another round of applause for all of our speakers from this last session. And now I would like to invite you to go to dinner. We'll relax a little bit, and then we'll meet you all for dinner at Shoko. The address is printed, I think, on the little thingy. It's on the website. We'll see you there at 8 p.m., and I do believe you need to bring your badges again. So we'll see you there for drinks and dancing, apparently. See you there.